professor in Sun University, Naples, Italy. She happens to be a reviewer of many reputed national and international journals. She is a dynamic lady who holds many administrative responsibilities like coordinator, warden, counselor, and also a member of various important committees of University of Allahabad. She shows her excellency by presenting 150 papers in national and international conferences. More than 10 books are being published by her. She had many laurels. She worked on 10 national and international projects. Not only this, she also acquired guarding 19 PhD students and 10 postdoctorate fellowships. She also had one patent on her account. Now, to give momentum to this session, I hand over the session to a respected chairperson, Professor Santi Sundaram ma'am. Ma'am, kindly chair the session. Hello, am I audible?
Yes, ma'am.
Yes, ma'am. Ma'am, Shanti ma'am, please chair the session. It's a humble request. Ma'am, can you hear me, ma'am? Shanti ma'am?
One minute.
Excuse me, ma'am. Ma'am, can you hear me? Shanti ma'am?
Yes, ma'am. You are not muted.
दबाया हुआ है, हम नहीं कर पा रहे इधर से.
Ma'am.
Hello.
Yes, ma'am. You are audible, ma'am.
Yeah, ma'am.
Are you able to hear me? You see? Hello.
Yes, ma'am. Hello.
Hello.
Hello, ma'am. You are audible.
Can you hear me?
Ma'am, kindly continue.
Okay. Okay. So, good morning, everyone, and welcome to the day of this national conference.
मैम आपका माइक पास में रख लीजिए थोड़ा सा अपने. आवाज धीमी आ रही है मैम आपकी.
हाँ, बहुत धीमी आ रही है.
मैम अपना माइक अपने पास रख लीजिए, हेडफोन का माइक. शांति मैम. हेलो.
So, Prakash Yadav, who's currently principal scientist, medical and process chemistry at CTRI now. I'm proud to invite him as a speaker for this session, being an alumni of University of Allahabad, from where he did his bachelor's and master's from chemistry department, PhD in the Department of Chemistry from Awadh University, and his topic of communication of medicinal plants and purification of bioactive medicinal products. Dr. Prem Prakash Yadav is both of the DART Fellowship from Germany, Göttingen University.
हेलो,
Yes, ma'am.
आवाज नहीं आ रही है ना मैम की?
I think some network issues.
Hello, can you hear me now?
Yes, ma'am. But your
Hello, can you hear me?
yes, ma'am.
I would, okay, I again would take this opportunity to thank the organizers of this international conference for having me to be the chairperson of this session. I invite Dr. Prem Prakash Yadav, who's going to be giving his lecture right now, who is right now the principal scientist in CSAR CDRI from Process Chemistry Division. To invite my Allahabad University alumni, who and MSc from Allahabad University and PhD University Faizabad on the area of investigation of medicinal plant and chemical transfer.
The voice is missing.
Hello, sir.
Hello.
Ji.
Yeah.
Yes,
I'm here.
you are audible.
I think there is some connection problem.
Yes, sir.
So
Sir,
Shall I introduce?
you are co-host now. You can share your screen from your side.
hello.
Shall I introduce, sir, if you wish? Anuradha, ma'am.
Yes, ma'am.
Shall I introduce,
Yes, ma'am.
sir?
First, you introduce, sir, because
Oh yeah, there is some problem. Now I'm glad to invite our speaker, PP Yadav sir, senior scientist, medicinal and process chemistry department, CSIR Lucknow. He received many awards like CSIR CDRI Incentive Award, DAAD Postdoctorate Fellowship to work at Goethe University, Germany, SRF, and also qualified grade 2001. His research interest is identification of bioactive chemicals from natural resources and development of novel strategy for the synthesis of medicinal active heterocyclic. He had 44 research articles, 3 review articles, 1 book chapter in a reputed national and international journals having high impact factor. 8 PhDs are awarded and 5 pursuing under his able guidance. He too have 2 patents on his account. I cordially invite you, sir, for your lecture. Sir, please continue.
Thank you, Dr. Sumita, for nice introduction. I heartfeltly thank Dr. Shanti, who has some network issues. So I'm thankful to you, ma'am, to initiate this introductory session, and I'm thankful to Principal of SS Khanna Girls PG College, Professor Lalima Singh, and conveners of the conference to give me this opportunity to present to you my views regarding drug repurposing in this pandemic times of COVID. And as you all know, this is a pandemic time, and to have students engaged in scientific activity, scientific and academic activities is very challenging. So I want to extend my thanks to principal and conveners of this international PEA conference because they are doing very hard work to connect students and scientific community, academic community, to stay connected and informed about recent literatures, recent happenings, and what need to be done in the situation of pandemics. So, being human beings, we learn from our past experiences, and this is a very right time to say that yes, we have to come out of some set protocols. We have to come out of box, think differently to deal with this pandemic situation. And health sustainability as the theme of this conference. And I like this theme, and I wish health sustainability for all of us in future. And we need to be prepared. And the message I want to convey through this e-seminar is that we need to be well informed, then we need to be proactive, and then we need to be very much participatory, and we need to interact with each other to help society at large. So with these few words, I would like to share my slide. So shall I start sharing from here?
Yes, sir.
It's audible and clear?
Yes, sir.
So my slides are visible?
Yes, sir.
So is it in full screen mode or not? Can you see it in full screen mode?
Yes, sir.
Yes, sir. Yes, sir.
Okay. So I'll be talking about I would request all of participants to please mute and listen to the talk, because it's disturbing in between. If there is a lot of voice, I may not be able to focus. Okay, thank you. So I'll be discussing recent advances in drug repurposing for combating SARS-CoV-2, and the situation has arisen because of very recent virus pandemic, and I'll be discussing it in detail. So
Don't take.
Sir, kindly unmute yourself.
yeah.
Yes. Yes.
So we know that drug discovery is much needed affair towards attaining health sustainability, and I will be describing about drug discovery perspectives, how we approach traditional drug discovery research and how we achieve it. We know that drug discovery has evolved through ages, and it's not only now we are talking about it. Actually, since human evolution, we have learned from our ethnic traditional systems. We learn to live together with the environment, nature, and then we learn to cure diseases from nature also. And as you can see from this slide, that from ancient to medieval to modern era, we have evolved from traditional systems of medicine to more personalized systems, where we have target-based drugs, screening of large synthetic and natural product libraries, nanomedicines and targeted delivery systems, phytopharmaceuticals and nutraceuticals being talked about. They are actually advanced versions of what we have uh, used in past in throughout our ancient historical knowledges from traditional Ayurvedic, Unani, Siddha uh, systems of medicines. So phytopharmaceuticals, nutraceuticals, and Ayush and other traditional systems of medicines being used in other countries are actually uh, purified versions of what we have used in past. So there are challenges posed in this drug discovery chain, and these challenges of a recent era are drug resistance, which has emerged for antibacterial drug resistance has been a major problem, and emerging lifestyle and CNS diseases, and added to it is unknown pandemics like COVID-19, which we don't presume to happen and it comes to us. Secondly, affordability of medicine, because medicine prices and therapeutics cost has gone very high because of involvement of so many regulatory things. So affordability has been an issue and this has to be worked out. Drug regulations, time and cost of development, these are all issues which not only affects uh, development of affordable drugs, but it also affects access to medicines. So if we go through a modern drug discovery flow, we basically start from drug target identification and validation. And here we take help of chemical biology for target identification validation, molecular understanding, and chemistry methods, being it chemical biology approaches, and synthesis of target molecules. So first we identify drug targets, validated drug targets, and then we identify ligands for these drug targets. Followed by this, based on this rational designing or phenotypic screening, we screen library of compounds against this identified targets, and we arrive at hit molecules, maybe micromolar ranges. Then hit to lead identification is performed, where we diversify our hits based on their physicochemical properties, DMPK properties, and finally we achieve selective nanomolar active molecules with suitable for in vivo studies. Followed by lead optimization studies where structure activity relationships, pharmacokinetic optimizations, where we need to check its absorption, distribution, and metabolism, and then suitable formulation for suitable for the route of delivery is developed, and then preclinical candidates are being identified through these lead optimization steps. Followed by preclinical efficacy, safety, and toxicity studies in GMP, from a GMP batch of the preclinical candidate, and these all studies require GLP conditions. So here in recent past, this GMP and GLP requirements have been stringent, and we have to perform these preclinical studies in a GLP condition. And after successful completion of preclinical efficacy and safety and toxicity establishment, we can go for investigational new drug submissions, and after clinical trials, approval by the regulator. So this is a long process. It's not a one-day job, or it's not something which we can anticipate today and deliver tomorrow. So this slide shows how success rate and time in drug discovery is involved. So success rate of a drug discovery is pretty low. As you can see, when we start from a 10,000 molecule library, if we test 10,000 molecules at the drug discovery stage, discovery phase, only one molecule gets approved out of it. And time required for the discovery phase is around 4 years. Then preclinical trials, it is around 3 years. 7 years it takes for the clinical trials, first 2 and 3 phases. And then followed by regulatory review, which again takes 1 year, and then finally it gets approved. If we talk about the global average cost of this development, it's around $2.5 billion. And if you talk about global average time required for new drug discovery to development, it's around 12 to 14 years. So it's a huge time. 12 to 14 years, virus is not going to wait for us. So every now and then we see pandemics in different parts of the world. Since mobilization has become so easy and accessibility to different remote places and different countries has become very easy, so uh, spread of these diseases is very easy nowadays. This we have seen in case of coronavirus spread this time, that it has spread to around more than 218 countries now. So it's at very fast pace. Within around 6 months, it has spread to around 200, more than 200 countries. So you can imagine how fast it is. It One reason is because of access to international travel to many of us, very fast modes of transport. Even in India, if we say we have initially we have it in pockets, then we have throughout country now. No part of India is free of coronavirus on the map. So to deal with this pandemic, this drug discovery pathway is not suitable. We have to have, we have to evolve with systems which are pretty fast, which can deliver drugs in very fast time. Development of vaccines is again one thing which is not that fast. Development of vaccine, because there are certain issues with development of vaccines, because they have to be safe at the end, and their safety concerns are major. So there has to be stringent clinical trial requirements before it can be delivered to masses. So vaccines cannot be given to patients in very short span of time. I don't think we can see any vaccine coming out with before 1 year. It will not be available for common people for use. Maybe it's available for use in clinical trial settings. So what happened after this coronavirus pandemic came? So most of the countries have started getting information, getting to know. Regulators have meetings to decide upon what to do and what they have done in past. So drug repurposing has been one uh, rescue in this situation. So you can see notification by CDSCO, Central Drug Standards Control of India, on 19th March 2020. It was released. India has no policy regarding uh, drug repurposing before this pandemic. So it's good to see that drug regulators have thought well in time, and they have come up with, these are basically three major points which they have mentioned in that notification, so that organizations or startups developing drugs or vaccines can approach Drug Controller General through the Public Relations Office for any information guidance related to regulatory approval for any repurposed drug or any other candidate. The central authority will give priority to firms or research organizations with a protocol for repurposing of existing drugs or vaccines for the treatment of COVID-19. The last point is very important as far as drug repurposing is concerned, because you have very large time window for these studies to be completed. So depending on the individual cases, the data for animal toxicity study, stability study, and clinical study of the drug or vaccine can be abbreviated, deferred, or waived based on the type of vaccine, nature of drug, and nature of material which we are going to repurpose. So major, we have seen in the previous slide that it's around 7 years which took to complete this preclinical to clinical work, preclinical toxicity, and this one. So we can save this much of time by uh, going through these regulations. And so as a welcome decision. So what is repurposing? It's also called repositioning, redirecting, reprofiling. And drug repurposing involves identifying new use of approved or investigational drugs that are outside the scope of original intended or approved medical use. Say, for corona case, we have several of the drugs which are being, like anti-malarial drugs have been repurposed, anti-HIV drugs have been repurposed. So their intended use is in malaria or HIV. However, they have been repurposed to be used in COVID condition, in COVID infection. So alternative strategies in the current era are reducing development time, cost, and all advantages over there. It's basically polypharmacology driven, identifying existing compounds through biological plausibility, in vitro, in vivo, in silico studies, or serendipitous clinical observations. Like in these cases, people use uh, clinical observations and repurpose these drugs, provisions of repurposed drugs to patients through compassionate use or clinical trials can be sought. So there is, I think somebody wanted to ask something. Okay. So history of pandemics, if we see, it's from 1700 to 2020. I have, you can visit this website to see this categorization. And here balls represent the amount of deaths incurred during these pandemics. So you can see that there are several pandemics in past, like third plague around 1855, like Spanish flu. Around 40 to 50 million people died. HIV/AIDS still continuing, 25 to 30, 25, 35 million people died. And here it is COVID-19, which is around 608,000 deaths incurred so far, and it's still continuing. Swine flu, SARS, Ebola, and MERS related, you can see over here. So it's continuing. It's not that COVID has come today. There are viral pandemics in past, and if we compare their deaths, we can see that Black Death or bubonic plague has been the maximum, has accounted for maximum death, 200 million deaths incurred. Spanish flu, smallpox, plague, then uh, there are so many to note, and COVID is certainly gaining its pace and it's moving forward in this race of pandemic deaths. We are fortunate to have it being halted at this step so far. I hope with the effort of international community, we'll be able to reduce the number of deaths in case of COVID. So coming to coronaviruses of group of highly diverse envelope positive sense, single standard RNA viruses. And there are two subfamilies of coronavirus family, coronavirus and coronavirus. These were first discovered in 1960s, and mainly they are of zoonotic origin, transmitted from animals to humans. And there are four genera from genomic classification: alpha coronaviruses, beta coronaviruses, gamma and delta coronaviruses. So SARS-CoV-2 belongs to beta coronaviruses family. So 7 types of coronaviruses infect human beings, and 4 out of these 7 infections are self-limiting, mild, and only self-limiting respiratory tract infections are observed, like in these 4 cases. There are 3 coronaviruses which cause fatal pneumonia, severe acute respiratory syndrome, SARS-CoV, around 2002 to 3, it occurred in China. MERS, Middle East Respiratory Syndrome, MERS-CoV, it occurred in Saudi Arabia, 2012 to 13. And then SARS coronavirus 2, which is 2019 till now, and 218 countries that has spread to. These 2 viruses didn't spread too much to other parts. However, SARS-CoV-2 has spread to around 218 countries, and the exact reason why it spread so fast to so many countries is still question, and still there is no exact answer. However, it is believed that initial symptoms they don't come out in time, and people are infected without symptoms, and we have seen a considerable shift in travel pattern of international community. So there is a frequent travel to Chinese part of the world where this coronavirus first appeared. There is again one important fact to note. Everybody was interested in the beginning that from where it came from, this uh, coronavirus. Has it come from animal, mammals, birds, or what? So this theory has to be proved, and it is surprising to see that the uh, geneticists or microbiologists, they have come up with uh, within 2 to 3 weeks of emergence of the SARS-CoV-2, and they have mapped the genome of coronavirus 2 and analyzed it with the existing genome mapping of bat coronaviruses, SARS-CoV and MERS-CoV and related coronaviruses to map its origin. And what they found that may have originated, it has origin in bats, but its ultimate mammal host or intermediate mammal host is not sure, but it is presumed, it is speculated that pangolins have the most similar genomic sequence of SARS-CoV isolated from uh, these pangolins. So pangolins are the immediate intermediate host which infected humans, and it was said that from a food market of China it is spread to humans, and so where pangolins were sold it is said, but it's not confirmed yet. So MERS-CoV also originated from bats, and intermediate host was camel, and in SARS-CoV intermediate host was palm civets. So these have been published literature where you have all genomic informations regarding phylogenetic origin of coronavirus, where you can find it. So why this is important to know that what case fatality and reproduction factor is for these viruses, because SARS-CoV-2 has very high infectivity due to certain differences in its receptor binding domain of ACE2. It binds to ACE2 of the human cells, and there is certain differences in receptor binding domain of SARS-CoV-2 and SARS-CoV and MERS-CoV. So you can see case fatality is still less, 2.3%, but it has infected larger populations, so it needs to be seen over the time. After pandemic is over, then this case fatality will be clear. This reproduction factor is around 2 to 2.5, which means that it is it has more infectivity as compared to SARS-CoV and MERS-CoV, and that's why more people possibly get infected from an infected individual, per infected individual. So an infected individual is able to transmit this disease to more number of people than in case of SARS-CoV and MERS-CoV. So this we all know. We listen in our phone ringtones that's being transmitted through different routes, human to human transmission through respiratory droplets. It has been detected in urine, saliva, fecal samples, anal swabs, and even in cerebrospinal fluid. Okay, and it can also transmit through contacts and fomites, non-living objects where droplets have fallen. So that's why we need to keep distances, use masks, and I think this virus is going to change the community behavior of whole population, because as I heard from my grandparents that during chicken pox and other this communicable diseases pandemic in past, they used to have containments. Like if you have smallpox, you will be confined to a room. Nobody will be able to visit you, touch you, something like that. So, but that situation can be tackled in individual domains. Here, if an infected person is outside on the roads, in population, in community, then if everybody is following the rules, everybody is wearing mask, it's fine. If everybody is not wearing mask, it's not fine. So community behavior needs to be changed to break this chain of transmission. So symptoms we all know: fever, fatigue, kidney function decreases, diarrhea occurs, sneezing, runny nose, sore throat, dry cough, shortness of breaths, circulatory system got affected. And most interesting of all these symptoms, there is one more symptom which has been added in recent past, that is loss of taste or smell. And it is believed that through nasal mucosa, it invades into the uh, CSF, it reaches CSF, and it disturbs our loss and taste centers in the brain. And it is also believed that this may be one of the reasons in uh, ARDS, acute respiratory syndromes, because lung function is controlled through brain. So it is also possible that this virus is affecting our lungs through infecting our CNS system. So coming to the diagnosis, and you know that diagnosis has been a challenge, and every day governments keep on increasing the number of tests. And why it is so important? Because unless and until we know that some person is infected, he or she may not uh, be able to contain it. So to be able to contain infection, we need to know people around us infected, so that they can be contained and the infection can be curtailed, spread can be curtailed. So there are three test categories which are being possible for this virus: nucleic acid test, antigen test, and antibody test, where most reliable one is nucleic acid test, because it's very sensitive test, where you get viral RNA from the nasal and mucosal swab, isolate the viral RNA, do RT-PCR, reverse transcriptase does the synthesizes DNA, and then copying occurs in presence of a probe. This probe is fluorescent, and so copying occurs. So from one DNA, from one copy, you are going to make thousands copy with this fluorescent probe, and this fluorescence is measured at the end. So a very small amount of RNA is sufficient to give you the results, and these results are very informative. However, it cannot tell you that if you had infection in past, it will tell you today's scenario. Whereas antigen test is also performed from swab samples, and this virus is collected and chemically broken down, and antibody mounted on plates are being, these viral proteins are loaded, and then fluorescent antibodies are clubbed, so that you get the fluorescence. If you get the fluorescence, it means there is viral antigens present. So these tests are not very reliable like nucleic acid test, and there are sensitivity issues, and they depend on the time of infection. Antibody test is also nearly the same, where you get viral proteins, viral proteins and antibodies. Samples is loaded over there, and fluorescent is detected at the label antibodies. So best one is through nucleic acid tests. Now coming to the structure of coronavirus. Why structure of coronavirus is important? Because if you don't know how virus is going to behave, we may not be able, what virus looks like and how it is going to behave, we may not be able to find out cure. And to ultimately find out, cure, and know about the virus, we need to know the structure first. So several structural biology reports have been published, and it is shown that it contains structural proteins, that is envelope protein, E spike protein, S membrane, and nucleocapsid proteins. As marked over here, spike proteins are these which protrude out of the surface, and it forms a corona-like structure. That's why they are called coronaviruses. So S, M, and E proteins, they together form the envelope of the virus. M protein and E proteins, they are membrane proteins and envelope proteins. They are structurally, they facilitate assembly and release of the virus. S proteins are embedded in the envelope, giving this crown-like appearance. And this S and M proteins are also transmembrane proteins that involve in virus assembly during the replication. This S protein is again important because it is involved in attachment to the human ACE receptor, and that's how this infection occurs. We'll see in coming slides. And protein first is RNA forming nucleocapsid inside the envelope. So COVID replicates through these proteins. So looking at the SARS-CoV-2 genome functional units, it's around 30,000 base pair long positive sense RNA genome, where you have two different regions. One is the structural protein region, and another major region is ORF1 and ORF1A and ORF1B, which are non-structural proteins, NSPs. So here you can see that spike region S, E encodes the envelope proteins, M encodes these membrane proteins, and N encodes these nucleocapsid proteins. So they are engaged in viral genome, structural proteins assembly. Then spike proteins are very important because they are responsible for viral entry into the host. Then we have RNA polymerase NSP12 to 17, and these units are involved in RNA polymerization and modifications for multiplication of viron. This region from NSP2 to NSP11 is encoding proteases, which are required for proteases which cleave this polypeptide synthesized through RF1 and give final amino acids required for synthesis of membrane or other parts. So viral assembly proteins can be seen. So ultimately these accessory proteins, some accessory proteins are there. You can see over here, these accessory proteins and these accessory proteins, they are accessory regions. They are required to antagonize human interferons as an immune response of the virus. So genomic similarity of SARS-CoV and SARS-CoV-2. What we can see is that why it is important. This study in Nature paper compares genome organization of SARS-CoV-2, SARS-CoV, and bat-originated coronavirus SL-CoV ZC45. So it's still in the bat population of bats. It's not infecting humans as of now, but it has very close similarity to SARS-CoV-2. And you can see that most of the genome is conserved, and you can see that most of the genome looks like to be similar, including structural proteins and ORF1 and ORF1A and ORF1B domains. However, there are certain differences in the sequences of spike proteins and other structural proteins. So SARS-CoV-2 was most closely related to bat with around 82.3% amino acid identity and around 77.2% amino acid identity with SARS-CoV. So this gives us a clear view that something which has been found effective in cases of SARS-CoV may be thought of to repurpose in case of SARS-CoV-2 upon due diligence and studies regarding its binding pattern or receptor binding domains or active forms of protein. So in this slide, it's highlighted that amongst the SARS-CoV-2, SARS-CoV and MERS-CoV, what important proteins, receptors, what common proteins, receptors and proteases are involved. So you can see they both, SARS-CoV-2 and SARS-CoV, they infect, they enter to host cell through a receptor ACE2, angiotensin convertase enzyme 2, and they both have spike proteins similar. SARS-CoV-2 has around 76% similar spike protein to that of SARS-CoV-2. Then they all have 3CL-Pro and PL-Pro viral proteases. They all engage C3L-Pro and PL-Pro viral proteases to assemble their proteases, assemble their final polypeptides. Viral polymerases, so their genome is polymerized through RNA-dependent RNA polymerases. All of them, all these three viruses, they use RNA-dependent RNA polymerases to replicate, make copies of the genome. There is one more interesting thing, that host proteases, TMPRSS, serine proteases. This is engaged in MERS and SARS-CoV-2 to affect a cleavage of a unit of S protein to help in viral internalization into the host cells. So viral entry is assisted through host proteases. So all these things, if we look in comprehensive detail, TMPRSS2, RDRP, C3L-Pro, PL-Pro, S proteins and ACE2, these are potential drug targets for SARS-CoV-2 also, as compared to SARS-CoV and MERS-CoV. So here we can see that the spike proteins, this is viral spike proteins. This is ACE2 receptor, angiotensin convertase enzyme 2 receptor on the cell linings of lung mucosa, and this virus binds to ACE2 receptor, followed by activation. This receptor, after binding it is activated by TMPRSS2. This is a host-derived serine proteases. It binds and cleaves this receptor to activate this complex and thereby facilitate internalization or endocytosis of viral particles. So this is a complete picture how virus enters to host cell. So if we are able to interrupt these spike proteins to bind to recognize this receptor, or if we interrupt this receptor and spike protein interaction, or if we can inhibit this proteases activity, we'll be able to control, or we will be able to defer viral entry into the host cells. So this is the first target, and if we can see the whole process, You can see it over here. So viral particles bind to ACE receptor and then internalized through endocytosis. RNA genome is released. Then translation of viral polymerase translation occurs. Then this negative sense RNA is released, and then genome replication occurs. After that, viral genome is assembled through viral genome is copied to numbers by reverse RNA dependent RNA polymerases. These proteins, nucleocapsid, spike, membrane, and envelope proteins are being translated into viral structural proteins, and then assembly of these viral particles occur, then formation of mature viron, and then exocytosis lead to release of more viral particles to mucosa. So if you can see these processes, these are numbered according to the points of interventions. We can intervene at these steps during viral life cycle. So as glycoproteins, AMPRO, 3C-like proteases, papain-like proteases, PL2-PRO, RNA-dependent RNA polymerases, they are all targets, viable targets to be pursued. So if we see from the literature and all, we will find that what happens in case of other viruses, or what we can gain insight from drugs which are being used in SARS-CoV, MERS-CoV, and other related viruses, that monoclonal antibodies and convalescent plasma can be used to inhibit this ACE and spike protein interactions, so that it inhibits viral entry to the host cell. Then we can inhibit the serine proteases TMPRSS2 to activate this ACE2, and which leads to internalization of viron. So camostat mesylate is used. Then we can inhibit proteolysis. So after translation, we get polypeptide chains synthesized. However, to get desired polypeptides for particular activity, you need to digest these polypeptide chains into required polypeptides, and this proteolytic cleavage is done by 3CL-Pro and PL-Pro proteases. So these lopinavir and ritonavir, which has been earlier used as anti-HIV drugs, as proteasome as proteolysis inhibitors. So they are being repurposed. Then remdesivir comes, where you have RNA-dependent RNA polymerase inhibition. So remdesivir and other related compounds, they inhibit RNA-dependent RNA polymerization to so that viral genome is not multiplied and stopped at this stage. And then followed by repackaging into the vesicles, and then exocytosis, viral release to the cells. So targeting viral entry by small molecules, if we see this structure of the SARS-CoV-2 spike protein and ACE2 receptor, there are 3 sites: N-terminal, C-terminal, and Hinge region. And this shows a picture of SARS-CoV-2, and green shows SARS-CoV RNA receptor binding domains and ACE2 receptor. So we can identify the receptor binding domain from here, and we can target the specific residues which are required for receptor binding in SARS-CoV. And interestingly, in SARS-CoV and SARS-CoV-2, receptor binding domain sequences are not very similar. And that's why we can say that infectivity of these 2 viruses is different. So in the first case, we can see that the small molecules targeting receptor binding domain and in its interaction with ACE2, chloroquine and hydroxychloroquine. They have been used in malaria treatment and inflammatory diseases, and they have been repurposed for this use, and they have been used for more than a decade now, and they were the easiest choice for this repurposing. For a country like India, India is the largest producer of chloroquine and hydroxychloroquine. So it's very interesting to see that these compounds were effective, though they were not effective in severe cases, and they were recommended for only prophylaxis to uh, not to get infected from, like in healthcare facilities, people use these drugs to avoid any infection. Interaction with membrane fusion of the spike protein S2 inhibition is by umifenovir, and umifenovir is a drug which is approved for influenza, respiratory viral infection in Russia and China, and this drug was repurposed for COVID-19. Similarly, we discussed that TMPRSS2 is serine proteases can be targeted, so proteolytic processing inhibitors and chemostat and related analogs, amidine analogs at this site also. They have been repurposed, and they are actually clinically used in Japan for treatment of pancreatitis. So repurposing of these drugs, because they have been already approved by regulatory authorities, so they must have undergone critical clinical trial regimens. So they must have been found to be safe for particular use, and they need to be re-verified depending on dose and period of use under COVID conditions. And after regulatory review, they will be approved for use as therapeutics for SARS-CoV-2. So M-Pro, and it is also called 3CL Pro. If you see proteases, 3CL Pro proteases, actually what they do is serine, thiol, and this histidine. Histidine is activating this thiol to attack to the amide bond of polypeptides in this fashion, then generating this tetrahedral species, followed by hydrolysis to give amines and thiolated peptides. So free amino acids or cleaved amino acids you get through proteolytic cleavages, and this mechanism has been proved through various mimicking studies. And there is one, this is a crystal structure, 3D structure of 3CL Pro bound to a peptidomimetic, and here it shows that this peptidomimetic bind to cysteine 145 of this CL Pro to show that how it is effective. Actually, it is acting on Michael acceptor region of this peptidomimetic. So if a molecule is having a Michael acceptor region, it is prone to be attacked by this, affected by this 3CL Pro. So the SARS-CoV-2 and SARS-CoV share very high identical sequences, and that's why HIV proteases have been repurposed. So lopinavir, ritonavir, cobicistat, and darunavir, they have been repurposed to treat COVID infections. Similarly, PL Pro is again a proteases, which is important, and it works in synthesis of viral polyproteins in a coordinated manner. And it has an additional function of stripping ubiquitin and ISG15 factor from host cell proteins to help cope to evade the host innate immune response. And that's how it is having additional benefits where it can help regulate the immune balance under these COVID infections. So disulfiram, mercaptopurines, and 6-thiaguanine, these compounds are also under repurposing for PL-PRO inhibition. RNA-dependent RNA polymerase is remdesivir. Before this coronavirus pandemic, it was in late stage development for Ebola and SARS, and it is approved for emergency use in COVID in many countries, including India. So next is favipiravir. It is approved for influenza pandemic earlier. It is an approved medication, and it has no patent protection as of now. So generic version is approved for use in COVID-19. Both remdesivir and favipiravir are prodrugs, and they are getting metabolized in cells into active triphosphate metabolites, which were found to be active. Again, another galidesivir, it's developed for Ebola pandemic in West Africa and repurposed for use in COVID-19 as RNA-dependent RNA polymerase inhibitor. There are certain host targets which are also targeted to get repurposed drugs. There is one biotech company, Benevolent AI, which proposed that AP2-associated protein kinases as a host target to fight SARS-CoV-2. This was through a machine learning program, and AK1 is a key enzyme of receptor-mediated endocytosis. So you can see that baricitinib is responsible for receptor-mediated endocytosis of this viral entry, and thus they have predicted that these kinases will be effective to reduce ability of virus to enter lung cells. And baricitinib and other kinase inhibitors are being repurposed based on this approach to target SARS-CoV-2. So it's more than, I think, around 200 now. These treatments are under investigation for COVID-19. Antivirals, antiparasitic, antibiotics, antibody, immunologicals, others, they are being repurposed. These are all drugs which have been approved by one or other regulatory agencies. So these are some of the compounds which have been repurposed. Others which we have already discussed. So for the people involved in synthesis and chemistry oriented task, I just kept 2 slides to show the importance of how process chemistry is helping community. And if you see, this is a remdesivir synthesis, remdesivir commercial synthesis process, which was adopted by Gilead earlier, which was later on modified. Here, actually, it's a chiral molecule, and most challenging part is with chiral molecules is stereoselectivity. So in this case, they started from ribolactones and to generate this hydroxyribonucleic, and they did cyanation through TMSCN mediated reaction, followed by HPLC separation of this alpha beta mixture. And then at the last step also they have to do HPLC separation. So this process has flaw that it uses 2 HPLC purification, chiral HPLC purification steps. It's very easy in the lab conditions to do chiral HPLC purification. And also it is an indirect way to introduce nitrile function to the ribosyl moiety. So these 3 things were the limitations of this synthesis. To avoid this synthesis, they later on changed this process through this flow synthesis method, where you inject different compounds in different concentrations. These compounds move through pre-cooling loops, then reaction loops, and then first step is performed, and then second step reaction is performed, and then finally you get the product under controlled conditions in a continuous flow. So by replicating this process into a flow reactor, they have synthesized around 250 kg of material with more diastereomeric purity as compared to the other reaction where you get mixtures with um, undesired diastereomers. So undesired diastereomers were very less in this case, and yield and purity is around 99.9%. So that's how this process is being used for commercial production of remdesivir even in India now. So just to brief you about CSIR contribution to fight against COVID-19. CSIR as a whole is also engaged in rapid and economical diagnostic, digital molecular surveillances, development of new drugs, repurposing of drugs, and other logistics. So to summarize these contributions, these are the drugs which are being repurposed through CSIR efforts, and remdesivir, favipiravir is being repurposed by IACT and IIIM Jammu. Omifenavir has been repurposed by efforts at CDRI, and it has been approved for phase 3 clinical trials by DCGI. Other drugs being pursued at CDRI are niclosamide and nitazoxanide. We are already in process to get these compounds through, and IACT and others, they are also modified. They are also dealing with other drugs for repurposing. These are certain APIs available with Indian manufacturers which can be repurposed without any further delay. So this is all about omifenavir, which is being repurposed by CDRI. So these details we have already discussed. There are certain advantages. It's being used as antiviral in Russia and China, and approved medicine for influenza. So it's very easy to be repurposed, and the beauty with this molecule, why we have selected is because it's not produced in India. So we have FTO, freedom to operate in India, with this molecule, and this will be tested in phase 3 clinical trial in collaboration with PGI and other institutions. So there is an effort by Government of India to create task force on repurposing of drugs, T4 for COVID-19, and this is a website. This is nclinnovations.org/COVID-19. It's a group including CSIR and other task stakeholders, and you can go to that web page and find out very interesting information related to repurposing of drugs, and you will find each and every detail about the repurposed drugs in India and their status as of now. So more than 1,600 clinical trials, 150 drugs, and at least 3 vaccines in advanced phases of trials. So this is all summary for the presentation. Personal protective equipments have become a necessity now. So just on a lighter note, this is to say this is a 17th century PPE kit, and this is 21st century PPE kit, where you protect most of the part of the body and keep yourself away from viruses. CDRI, I would like to dedicate few slides to my current institute where I'm working. So 114 interdisciplinary scientists are involved in R&D activities, and major areas: medicinal chemistry, parasitology, microbiology, endocrinology, CNS, they are engaged in discovery research, then drug target and mechanism, and then preclinical and clinical research. This is to highlight that we have GLP test facility for safety pharmacology and toxicology, and a SAF facility which is being used by too many institutions, universities, colleges. These are the list of drugs which are being developed in India since independence. You can find 20 drugs being developed so far in India, 20 new drugs. And out of these 20, 13 drugs have been developed in CDRI. So it's proud to say this. And some of the success stories are Centrumon or Meloxifene, which is sold in the brand name of Saheli. Recently, Chaya has been launched by ICMR in the National Family Planning Program. And it's being repurposed also for mastalgia and fibroadenoma. Alpha beta RT ether, this process was developed at CDRI and is the most selling process from CDRI, alpha beta RT ether, and all the injections by these companies, they are being made through, Themis is being made through this process done at CDRI. Recently, plant-based products have been approved. Delbergia Sisu derived reunion and Spinacia oleracea derived joint fresh. This is a nutraceutical and this is a standardized extract Ayush mode. So they are used for anti-osteoporotic agents and for bone health. So finally, the message of Ebola, SARS, and CoV-2 is that we are going to see these things emerge. The only antidote is being ready, and that's what we have to do, prepare. So COVID-19 has prepared us for future viral pandemics, accelerated drug discovery by inclusion of artificial intelligence, drug repurposing as a viable strategy. It has been proved during this pandemic, and development of safe vaccines at faster pace is something which is coming up. We are seeing very fast development of vaccines. Thanks for patiently listening, and thanks for inviting me to this interview. I will come in comments to this. Thank you very much. Hello.
Can you hear me? Thank you very much for the excellent talk, sir, which you have delivered. It was very descriptive, very helpful to all of us to know about the various structure and the symptoms of SARS-CoV-2, the genome functional units involved, especially are trying to discover drugs and vaccines. The repurposing of drugs is something which we have all learned recently, and that's a very interesting aspect of drug discovery. As you mentioned, that Ebola, SARS, HIV drugs are being repurposed for SARS-CoV-2, and we hope that your institution, CDRI, which has always stood out for the society discovering new drugs, is going to continue and have great success in finding a drug for COVID-19. It was a very illustrative, very informative talk, and we really appreciate you for taking time out and telling us all about the drugs that is being worked upon recently, because everyone now is curious and are very keen to find a useful drug or vaccine so that we can come to an end of the pandemic. Thank you so much, Dr. Prakash, for the excellent delivery. Thank you.
I'm delighted. Thank you very much.
Anshua ma'am, hello.
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 So thank you, ma'am. I think I have conveyed the thanks to the speaker, and I would like to say thank you to you also for inviting me to chair this session. Thank you very much. Hello.
Hello.
Can you hear me?
Yes, ma'am.
Thank you very much.
Thank you, ma'am. Archana, ma'am, kindly conduct the
Okay.
session.
Okay. Good afternoon, everyone. Sir, thank you so much for very informative talk. There are some questions asked by the participants. First question is asked by Aisha Dev. What are post-COVID-19 preventive measures for recovered COVID patients? After how long should he or she start daily life?
Actually, it's
Sir,
very
am I
tough
audible?
question. because we have seen that it is being revised time and again since the beginning of COVID-19 infection. You must have seen that we first didn't knew that what is asymptomatic, symptomatic. Then we got to know that there are asymptomatic people who can also cause infections, who can spread infections. And then, so to deal with this kind of a situation, we must ensure at least 14 to 20 days of quarantine of people who are infected, and wearing mask even after quarantine period is over, so that you don't spread it unintentionally. And
Yes.
it's not for infected people or for people who have gained this disease, who have infected with this virus. It's for everybody that we should ensure safety in public places. We must wear masks to curtail this spread.
Thank you, sir. Second question is, sir, if a person is not affected by COVID-19 and he or she takes proper dose of hydroxychloroquine just for precautionary measure, is it safe or harmful?
Yeah, it's very interesting question, because I have seen people piling up chloroquine and
Yes.
hydroxychloroquine with
Yes,
them
of course, sir.
without knowing what purpose they are going to solve out of it. And
Yes.
it's very much required to be answered here, because chloroquine and hydroxychloroquine, both these drugs, they are used to treat parasitic infections.
Yes.
And as you are aware that for parasitic infections, you don't need to take them for long time. And they are not the only medicines which are taken for malaria chemotherapy, because you have other medications, safe medications. Chloroquine and hydroxychloroquine, they are both known to cause serious side effects when doses are not, appropriate doses are not taken.
Yes.
And they cause severe damage to liver. So many countries, they have stopped using it even for prophylactic use in healthcare workers also. So I don't think it's good not to get virus, then go away with our liver. So we have to be safe from virus, as well as we have to keep our liver healthy. So to keep our liver healthy, we don't need to take unnecessarily any medicine, and specifically chloroquine and hydroxychloroquine, because hydroxychloroquine is relatively safe, but they both affect liver.
Thank you, sir. The next question is, some people say that if a person is tested for this disease 3 or 4 times in the same day, then sometimes the results are positive and sometimes results are negative. Is it correct or not?
No, it's not correct. If a person is infected, there is residence time.
Yes.
So this virus shows its symptoms somewhere from 4th day to 12th day. So you don't know, in some people on the 4th day of infection, you will get symptoms. Some persons you will get symptoms, you will not get symptoms, asymptomatic patients. But test, if you perform nucleic acid test, RT-PCR test, RT-PCR test is going to give you results the day you are infected, because it is taken from mucosal swab or nasal swab, where the primary infection occurs. And so even small viral particles are sufficient to multiply the genome and then test it. So frequent positive and negatives is not possible unless and until you are infected. But of course, several sensitivity of test matters. Since it is a lengthy process, it takes around 1 day to test and report the result. It's a task intensive process. So there may be some handling issues or something, reagent issues, because first you have to degrade virals to break this protein capsid wall to expose your RNA. So we take buffer and add it to the swab and viral is exposed. Viral genome is exposed. Then viral genome is being multiplied through RT-PCR. So PCR reaction is performed. Then these fluorescent tags are being involved in this PCR reaction, and then you detect this fluorescence. So if you do any mistake at any of these steps, this is not going to give you positive. Correct. So correct this.
अभी हमें किया नहीं है.
Human errors are possible. So it needs to be repeated. That's why you must have heard that if 3 times a person's sample comes negative, then only they say that he or she is cured. It's not one time.
Yes. Thank you, sir. Sir, next question is, can COVID-19 disease be transmitted by blood transfusion?
Blood transfusion, of course it can
Hello.
be. Yes, it can be.
Okay. Okay, sir. Thank you, sir.
Okay, welcome.
I hand over the mic to Dr. Sumita Sehgal.
Thank you, sir. Thank you, sir, for such a vibrant lecture and informative one.
Thank you.
Can you hear me? Am I audible?
Yeah, I can hear you.
Yeah. So now I think I just want to give you a vote of thanks to all. It's my privilege to have been asked to propose a vote of thanks on this occasion. I, on behalf of SS Khanna family and myself, give sincere thanks to our chairperson who had given permission and had spent valuable time with us. I extend my warm gratitude to our eminent speaker for giving such an informative and interesting lecture. We are indebted to you, sir. Thank you, sir. I also accord my thanks to our participants, both of our country and abroad, who made this session a big success. I also convey my deep appreciation to our principal, ma'am, for her perennial support. Last but not the least, I want to thank each and every member of the organizing team for holding such wonderful seminar on such a relevant topic which suits the present scenario. I hope you all have enjoyed this academic feast. A huge thanks to one and all. Now I request you all to kindly switch on your video for group photograph. Please, participants, switch on your videos. And Radha ma'am, kindly take it. And Radha ma'am,
Yes, ma'am.
have you taken it?
Yes.
Thank you. So this is all for today's session. I thank you once again. I hope our next session will begin at 2 p.m. We have oral presentation over there. So till then, I think goodbye from me and from all our organizing team. Thank you very much. Over and all.
Thank you, sir.
Okay.
Thank you,
Thank
sir,
you very
once
much.
again. Thank you very
so
much,
much.
sir.
Okay.
Thank you, sir.
So shall I leave now?
Yes, sir.
Okay. Thank you very much.
Okay, once again, sir, thank you. And Radha ma'am, is it over?
Yes, ma'am.
Okay, we will meet at 2 o'clock.
Yes.
Any query of any participant, kindly deal it right now. Okay, I think everything is clear. Okay, shall we leave?
Yes.
Okay. Bye, everyone. Goodbye from my
Bye.
side.
Bye.
Bye.
Ma'am, Sippy ma'am, kindly start. If about
Ma'am, we have been joined by Dr. Justin Masih, sir, but Dr. Nija Kapoor ma'am has not joined yet. That's why I'm waiting.
okay. Now she has joined.
She has joined. I think she's connected also.
Yes.
Kindly make Justin sir and Nija ma'am as co-hosts so that we can communicate with them.
I have asked this to Nishi ma'am.
Fine. Okay, let's start with the session. So, good evening to everyone. Welcome to the technical session 2 of day 4 of this e-conference. In this session, we'll be listening to the oral presentations submitted under Environmental and Health Sustainability section. I welcome our two distinguished guests, Dr. Nija Kapoor ma'am and Dr. Justin Masih sir, who will be chairing this session. Welcome ma'am. Welcome sir.
Thank you very much.
Dr. Nija Kapoor ma'am is the head and associate professor at the Department of Zoology, CMP Degree College, Prayagraj. Ma'am has specialized in the field of fish and fisheries and environmental biology. She is the recipient of prestigious awards like Bharat Excellence Award, Best Indian Golden Personalities Award, Rising Women of India Award, Gold Medal for First Position in Science Faculty in MSc, and Distinguished Teachers Award by SBSRD. Dr. Kapoor has teaching experience of 23 years and 25 research publications to her credit in the journals of high repute. Ma'am is a member of various scientific bodies, and apart from her academic capabilities and excellence, she is very generous, simple, and affable person. We welcome you, ma'am. We welcome you to this gathering.
Welcome ma'am.
Thank you.
Welcome sir.
Dr. Justin Masih sir is an associate professor at Department of Chemistry, Ewing Christian College, Prayagraj. Sir specializes in the field of inorganic chemistry, and he has published more than 15 papers in national and international journals. Dr. Masih has also edited a book entitled Chemistry, Role and Challenges. He has been awarded Research Fellowship by Indian Academy of Sciences, Bangalore. Sir is instrumental in setting up a spectroscopy laboratory in his department, and his research areas of interest includes heavy metal toxicity, bioaccumulation, and biodistribution of heavy metals, water pollution, heterogeneous catalysis, etc. We welcome you, sir. We welcome you to this gathering.
Thank you very much, ma'am.
Now, with your permission, sir, ma'am, the permission of the chair, we shall proceed for the presentations. So, shall we?
Yes, ma'am.
Okay.
Okay.
Yes.
Thank you, ma'am. Thank you. Before we start, I'll just read out the instructions for the paper presenters. The organizing committee has resolved that all the presentations will be in oral mode only. The presentations have been categorized into two sections, one for research scholars and others for faculty members. Due to variation in time zone of different countries, the international paper presenters had to choose one of the option, either go through the live online presentation mode, or they had to send the recorded presentations beforehand in the prescribed format. The presentation will be of total 8 minutes, which includes 5 minutes of presentation, followed by 3 minutes of discussion. The evaluation will be done on two parameters, organization and quality of the content, as well as delivery of the content. Now, I invite Dr. Anuradha Singh ma'am to continue for the further proceedings and allow our first participant, the international participant, Dr. Abdilghani Milani, to present his paper. Over to you, Anuradha ma'am.
Ma'am, I am sharing a screen of Dr. Milani.
Okay.
Kindly introduce
okay.
him. Meanwhile.
Sure. As our first speaker, His abstract number is ES11F. ES11F. Abdul Makani Milani is an assistant professor at PMR Department, Faculty of Medicine, University KM of Urgela, Algeria. The topic of his presentation will be well-being improvement of chronic low pain back patients treated with balneotherapy in southeastern Algeria. Milani sir, kindly unmute yourself.
Ma'am, screen is visible?
No, presentation is not visible right now.
Okay. Okay. Please let me know when the screen...
Sure. I'll let you know.
Now it is visible?
Yes, it is visible now. So Milani sir, you can start. Kindly unmute yourself, first of all. Dr. Nishi, could you please help him? Milani sir, first of all, please unmute yourself.
Yes.
Yeah.
Yes.
Yeah.
Do you hear me?
Yes.
Yeah, you are audible.
So, President, ladies, gentlemen, good afternoon. My name is Abdul Ghani Miliani. I am an assistant professor of physical and rehabilitation medicine at the University of Ouargla, Algeria. So it's my great honor to be with you today.
Yes.
Continue, sir.
Sir, I am sharing your screen.
So you can continue. We can see your presentation.
Can you continue?
Hello, Dr. Milani.
Yes, I am here. Can you continue presentation?
Yes, your screen is visible.
So it's my welcome everybody. It's my great honor to be with you today and to participate in this event. So I will talk about well-being and for improvement of chronic low back pain patients treated with balneotherapy in southeastern Algeria.
Hello?
Hello.
Yes.
So in introduction, I can see that improvement of quality of life and well-being is one of sustainable development goals. Balneotherapy or spa therapy is one of the therapeutic arsenal recommended by the major clinical guidelines for the management of chronic low back pain. It's useful to improve pain, function, and quality of life. The region of Ouargla in the southeast of Algeria has many thermal springs that are still largely intact. Around these spa resorts, there is the Balneotherapy and Rehabilitation Center of Ouargla, situated in the heart of the palm groves of the Algerian desert. This thermal water comes from a deep borehole reaching 51 degrees. With of
Hello.
Hello.
Yes, sir.
So with a flow of 200 liters per second, its mineralization is chlorinated and sodium sulfated type. The aim of our paper research is to assess the contribution of this baniotherapy in well-being improvement of patients suffering from chronic low back pain. Our method, this pilot study included 10 participants from patients treated at the Baniotherapy and Rehabilitation Center of Ouargla, Algeria. All of them present a common chronic low back pain evolving from more than 3 months. The sample was divided into 2 groups to comparative purposes. The first one is the intervention group, group A, receiving physical therapy combined with spa therapy. And the second one is the group B, control group, receiving only physical therapy. Physical therapy represents the classic rehabilitation program based on relaxing massage, electrotherapy, and exercise program. On the other hand, spa therapy contains some thermal cares as thermal water bath, general underwater mobilization, and free swimming pool. For the response of treatment was assessed using the new scale, the Oswestry Disability Index, which represents the most important scale used in determining improvement in health-related quality of life. It is a self-assessing questionnaire, contains 10 items. These items are pain, personal care, lifting and moving objects, walking, sitting, standing, sleeping disorders, sex life, social life, and traveling. This index was translated and validated in different languages of world, including Arabic, which used in our study. The questionnaire was given at the beginning and at the end of treatment. An improvement in Oswestry is considered clinically irrelevant. It is greater than 30%. Sensitivity to change of this index is estimated by the standardized response mean and effect size. Our results, please, the slide of results before. The table number 1 summarizes the characteristic of the patient included in the beginning of study. The mean age was 31, and the disc conditions were the cause of the chronic low back pain in 70% of sample. Figure number 1 illustrates the score evolution of the Oswestry Disability Index into the 2 groups during the 4 weeks of study. Of course, there was an improvement in both of the groups, but the gain obtained in the score of Oswestry into group A was higher than group B. It was 25 versus only 16% for group B. In addition, we noted that 60% of patients had a clinically relevant improvement of Oswestry index in group A versus just 20% in group B. The table number 2 reports the sensitivity to change of the scale into the 2 groups. And here also we noted the superiority of group A on B estimated by the standardized response mean and the effect size. Next slide, please. Discussion. In our study, there is clinically relevant improvement in 60% of patients in the intervention group. These results are close to those observed in a French study carried out by Foresti and al. in 2017, interested by the same subject. where the rate of clinically relevant improvement was 76%. In the scientific literature, we find some similar studies comparing the effect of the combination of spa therapy with usual treatments versus a control. The characteristics of these studies are summarized in table number 3, where is noted that the effect size in the intervention group is frequently higher than the control group. Next, please. In conclusion, I can say that baniotherapy using thermal water of Wurgla may be more effective in improvement function, well-being, and quality of life for patients with common low back pain. These preliminary results must be complemented by other large-scale studies which make it possible to assess the long-term effect and the durability of this combined therapy. In Algeria, according to the available thermal potential, baniotherapy should be more recommended in order to achieve the essential development goals. So I finish with this. Next, please. I finish with this photo of baniotherapy and rehabilitation center of Wurgla in Algeria. So thank you for your attention.
Thank you, sir.
Yes, yes, ma'am, continue.
You are the most welcome.
Hiya, your voice was there. Now I request our chairperson to ask any question. If they have any query, they can talk to you.
No, ma'am, I don't have any query. Thank you.
Have you considered any age group for your patients? Have you considered any age group? Between which age group you have studied?
Thank you for the question. The age mean was 31 years. Between it is
between
any age?
Between what is the age group of your patient? Have you done therapy our 80 years old, our 60 years old, or what was the age group?
Yes. It is the majority of our population, our Samal, is the adult, were adults, so between 29 to 43.
Okay.
Yes.
Thank you. Okay.
You are welcome. You are most welcome.
Okay.
Thank you. Our next participant is, his abstract number is ES07R. Anuradha ma'am, ES07R, Vishal Kumar Singh. Vishal Kumar Singh, along with the paper of Himani Chaurasia and Professor R.K. Singh. They are from Bioorganic Research Laboratory, Department of Chemistry, University of Allahabad. And the title of the paper is Design, Synthesis, Docking, TopCat Analysis and Molecular Dynamics Simulations of Daryl pyrimidine derivatives as non-nucleoside reverse transcriptase inhibitors against HIV. Vishal Kumar Singh, are you there?
Screen is visible or not?
Screen is visible, ma'am. Screen is visible. Vishal Kumar Singh.
Kindly unmute yourself. Vishal, kindly unmute yourself.
Yes, ma'am.
Yes.
Am I audible, ma'am?
Yes.
Yeah.
Okay, thank you, ma'am. A very good afternoon to all of you. I am Vishal Kumar Singh, pursuing my research work under the supervision of Professor Ramendra Kumar Singh in the Department of Chemistry, University of Allahabad. The topic of my presentation is design, synthesis, docking, TopCat analysis, and molecular dynamic simulation of diaryl pyrimidine derivative as non-nucleoside reverse transcriptase inhibitor against HIV. Next, please. This is a structure of HIV. HIV is a retrovirus belong to the family of retroviridine containing single-stranded RNA as the genetic material. There are two types of HIV strain, HIV-1 and HIV-2. Out of these, HIV-1 is more common as compared to the HIV-2, and it is transmitted through the several body fluids. Next, please. These are the physiochemical data of the diaryl pyrimidine derivative and reference drug etravirine, followed by the Lipinski rule of 5. According to Lipinski rule of 5, the molecular weight of the compound should be less than 500. The number of hydrogen bond acceptors should be less than or equal to 10. The number of hydrogen bond donors should be less than or equal to 5. The lipophilicity indicated by the log P should be less than or equal to 5. Total polar surface area must be less than or equal to 140. Physiochemical data revealed that none of the molecules show the major violation from the Lipinski rule of 5. The compound 6 having the lowest value of lipophilicity, which show that the compound 6 have better viral cell penetration ability. Next, please. These are the schematic outline of the compound 1 to 8. Next, please. These are the docking interaction of the diaryl pyrimidine derivative with HIV-RT protein, which revealed that the compound 6 have the greater number of hydrogen bonding and greater number of pi interaction as compared to the reference drug etravirine. Next, please. These are the docking score, which is obtained from the docking simulations such as PLP1, PLP2, dock score, ligand interaction energy, LUDI2, LUDI3, binding energy, and predicted EC50 value, which is also very helpful in prediction of stability of the complex formed between the compound 6 and HIV-RT protein. Next, please. These are the docking interaction of diaryl pyrimidine derivative with non-nucleoside inhibitory binding pocket of RT allosteric site. Next, please. Next. These are the TopCat analysis of diaryl pyrimidine derivative and the reference drug etravirine, which also revealed that the compound 6 have the lowest value of lipophilicity. That's why I have taken the compound 6 for molecular dynamic simulation. Next, please. These are the various type of graph what I have obtained from the molecular dynamic simulation. The graph 1 and graph 2 indicate the root mean square deviation and root mean square fluctuation of the compound 6 with HIV-RT protein shown by the red color, which is similar to the root mean square deviation and root mean square fluctuation of the reference drug etravirine with HIV-RT protein, which also favor the stability of compounds. Next, please. Next graph is the strength of compactness. The compactness of compound 6 with HIV-RT protein is shown by the red color, and the reference drug etravirine with HIV-RT protein is shown by the black color. The compactness of compound 6 with HIV-RT protein is lower as compared to the reference drug. Therefore, it is more stable as compared to the reference. Next one is the number of hydrogen bonding. The number of the hydrogen bonding formed between the compound 6 and HIV-RT protein is shown by the red color, and the hydrogen bonding formed between the reference drug and HIV-RT protein is shown by the black color. Next one, please. These are the comparative data of the compound 6 with HIV-RT protein and with the HIV-RT protein and reference drug etravirine with HIV-RT protein, which show that the compound 6 have anti-HIV potential as similar to the reference drug etravirine. Next, please. These are the snapshots what I have taken during the molecular dynamic simulation after each 10 nanosecond. Next, please. Next, please. These are the anti-HIV screening of the diaryl pyrimidine derivative against HIV-1 and HIV-2, which revealed that the compound 6 is active against both HIV-1 as well as HIV-2 with the selectivity index of 32.65 and 32.32. It is also interesting to note that the compound 5 is active only against the HIV-2. Any compound having the SI value greater than or equal to 5 may be considered as a good drug molecule. Next, please. These are the cytotoxicity evaluation of diaryl pyrimidine against human embryonic kidney cell. The data showing the variation, the cell availability of the compound, cell availability at the different concentration of compound. Next, please. Next one is the conclusion. From the molecular dynamic simulation, from the docking simulation and anti-HIV screening, we can see that the compound 6 can behave as a better NNRTI as compared to the reference drug etravirine. Thank you. Any question?
Am I allowed to ask something?
Sure,
Yes,
sir. Sure, sir.
Vishalji, first of all, many congratulations to you for this wonderful work that you are doing.
thank you,
And
sir.
of course, under the able leadership and guidance of Professor R.K. Singh, I'm sure that in the
Thank you,
days to come, you will be able to complete and pursue your work. Can you take me back to the slide where you were talking about hydrogen bonding?
Yes, sir. Analysis of hydrogen bonding. Ma'am?
1 minute. 1 minute. Sure.
So, Vishalji, there are a couple of things which I wanted to ask you.
Yes,
One
sir.
is that what is the biggest advantage of TopCat analysis, and why have you used TopCat analysis in this particular reference?
Okay.
The second thing is,
yes, sir.
The second thing is, how are you able to justify the stability of a compound based upon the number and type of hydrogen bond that you have referred in your presentation?
Yes.
Let's talk about the TopCat analysis first.
Yes, sir. TopCat analysis. By the TopCat analysis, we can predict the we can predict the type toxicity of our compound, and toxicity. It is only the prediction. For the cytotoxicity, we perform the cytotoxicity screening in another way, which are shown in next slide. And next one is the sir stability.
What kind of toxins? What kind of toxins are you able to analyze using TopCat analysis?
Yes, sir. It is a computational software. During the carcinogenicity, sir, and mutagenicity.
Yes.
And this actually, sir, we check the stability of the complex firstly by the Discovery Studio software. It is give the stability of the compound in any certain pose, not in all poses. But by the molecular dynamic simulation, we can check the stability of compound during the fluctuation. So that the molecular dynamic simulation is the correct way to check the stability as compared to the DS dockings.
And referring to the kind of hydrogen bond, what kind of hydrogen bond do you think is providing greater stability to the compound in consideration?
In electrostatic hydrogen bond formed between the ligand and HIV-RT protein.
Electrostatic hydrogen bond.
Yes.
So the ligand is behaving as electron donor or acceptor?
Donor.
Electron donor.
Yes.
Fine. Thank you very much, Vishalji. Thank you very much.
Thank you, sir. Thank you.
Next, we have Jyoti Sharma, abstract number ES08R. Jyoti Sharma, and she's from Amity Institute of Biotechnology, Amity University, Noida, Uttar Pradesh. Our topic is strategies for enhanced removal for pharmaceutical compounds from water by microalgae. Jyoti Sharma. Kindly unmute yourself. She's in the list.
Ma'am, let me know her screen is visible or not.
Abstract. Okay, I'll tell you, ma'am. Whenever it's visible, I'll let you know. Right now, it's not visible.
Okay.
Yes, now it's visible. Jyoti Sharma, kindly unmute yourself. Nishi ma'am, please help her. She has written the chat box. She's not able to unmute. Jyoti Sharma, try to unmute yourself from your end. Nishi ma'am, are you there? Hello. Nishi ma'am.
Yes, Jyoti, kindly unmute yourself.
Hello.
Yes.
Good afternoon, everyone. Ma'am, this is not my slide, ma'am. This is not my presentation.
Okay.
Represent.
Thank you, ma'am. Good afternoon, everyone. This is Jyoti Sharma from Amity University, Noida. The topic of my presentation is strategies for enhanced removal of pharmaceutical compounds from water by microalgae. Next slide, ma'am. As we know, water pollution is increasing day by day. Presence of pharmaceutical compounds in water has become a serious environmental issue lately due to their extensive ecotoxicities and chronic and acute harmful effect on living organisms. Though the detected concentration of pharmaceutical compounds is generally very less, that is from nanogram per liter to microgram per liter, but even at such low concentration, they exert harmful effects. The conventional treatment methods such as wastewater treatment methods are ineffective in removing these contaminants from water, and the chemical treatment methods are, though they are able to treat them, but they are associated with several drawbacks such as generation of large amount of sludge, which is a secondary pollutant, and it is even more harmful than the primary pollutant. Microalgae, on the other hand, can be considered as an ideal candidate for treating these pollutants as they are autotrophic, highly adaptable, economically and ecologically comprehensive, and they do not generate any sludge. So they can effectively assimilate nutrients from wastewater like nitrogen and phosphorus for their own growth, thus end up treating the wastewater. The removal efficiency can be enhanced by various processes like acclimation and bioremediation by microbial consortia. Acclimation can be achieved by changing the living environments of algae. We can do it by either providing high doses of organic compounds, salts, water, changing pH and light intensity. By changing that, we can achieve the enhanced removal efficiency of algae. Also, by making consortia of algae with bacteria or cyanobacteria, which is highly effective to improve the degradation efficiency. It can be done by natural substrate colonization or artificial inoculation. Next slide, ma'am. Thank you, ma'am. Various previous results study shows that the removal efficiency has been enhanced by the process of acclimation and microbial consortia. Acclimation results in higher tolerance and degradation capacity due to enhancement in various processes. In case of microbial consortia, the synergistic interactions can be occurred between co-cultivated microbes, hence resulting in an overall improved degradation capacity. I would like to conclude that the discharge of pharmaceutical waste should be regulated. The existing technology for treating pharmaceutical waste are ineffective and associated with various drawbacks. So microalgae can present interesting advantages and can be considered as an ideal candidate for potential treatment of pharmaceutical contaminants. Also, we can enhance the overall removal efficiency of microalgae by various methods that I have discussed, like acclimatization and forming the microbial consortia. Thank you, ma'am.
Yes, sir. Any questions?
Yeah, I just wanted to, first of all, Jyoti Sharma, ma'am, many congratulations for this wonderful work that you are doing in the field of environmental conservation, more or less. You mentioned a couple of things, out of which one sounds very interesting, and I'd like to just draw your attention and maybe be educated more on that. You spoke about microbial consortia.
Yes, sir.
What microbes would you suggest will constitute the larger part of the consortium, ma'am?
So basically, my PhD work is I will try to make consortia of microalgae with cyanobacteria or microalgae with another microalgae, like combining 2 or 3 microalgae together, and then checking their degradation efficiency, whether they are increasing or not.
Okay. And
We can also try it with bacteria also.
Okay, what part of the microalgae would you think that would be more instrumental in causing the degradation of pharmaceutical waste? What kind of interactions do you suggest that the microalgae or the microbial consortia will have with the pharmaceutical waste such that it causes effective degradation?
Sir, actually, the microbial consortia forms the synergistic interactions among them. Bacteria is giving the nutrients to microalgae, whereas microalgae is giving oxygen to bacteria. So, due to this synergistic interaction, they helping their growth and thus end up degrading more compounds.
Okay, fine. Thank you. Thank you.
Okay, sir. Thank you, sir.
Our next speaker would be Mahesh Kumar Gupta, abstract number ES09R from Department of Chemistry, University of Allahabad. His topic is removal of methylene blue and commercial brilliant blue R250 dyes from contaminated water with the help of banana peel activated carbon. Mahesh
And
Gupta,
let me know slides are visible or not.
I'll take it.
Mahesh, kindly unmute yourself.
Okay, thank you, ma'am. Ma'am, am I audible?
Yes, ma'am. Slide is visible. Yeah.
Okay. A very good afternoon. Kumar Gupta. I am pursuing PhD under the supervision of Professor P.K. Tandon, Department of Chemistry, University of Allahabad. Today I am presenting here my research article is on removal of methylene blue and commercial brilliant blue R250 dyes from contaminated water with the help of banana peel activated carbon. I am introducing here the water is the most essential element on Earth. In the current scenario, freshwater resources and their quality are depleting quickly due to rapid industrialization, urbanization, human activity, and other zoological and environmental changes. These activities are also lead to contamination of groundwater, organic pollutants. WHO estimated that around 2.2 billion people do not have access to safe drinking water, and around 4.2 billion people do not have safe sanitation facility. Clean drinking water spread waterborne disease like diarrhea, cholera, thyroid, and hepatitis A, etc. Every year, around 485,000 deaths takes place because of contaminated drinking water. Next, please. Textile industry produce wastewater is a large quantity that contains variety of contaminants that include acidic or caustic dissolved solid noxious compound and many natural or synthetic dyes. Most of these dyes are carcinogenic and mutagenic in nature and additionally noxious to human being, aquatic species and microorganisms. Removal of these hazardous contaminants from water bodies have become environmental necessity. Now, the removal of dyes from contaminated water with the help of banana peel activated carbon, remediation of water has been attempted with the help of novel green method. It is the ash obtained by burning banana peel. Banana peel is readily available in abundance as a waste product and hence is very economical and environmental friendly biomaterial. The main objective of the work present to be the study the efficiency of banana peel activated carbon is the degradation of commercially available mutagenic and carcinogenic dye from the wastewater. Next, please. For the remediation of water, I am using here the banana peel activated carbon from banana peel. By synthesizing this, the banana peel were obtained from the local market of Prayagraj Firstly, they were washed with distilled water and removed any adhering impurities and then dried under sunlight. Washed and dried banana peels were burned in porcelain dish. The obtained ash was activated with concentrated sulfuric acid, and the excess acid was removed by repeating washing with distilled water, and the solid black colored ash was filtered. Ash was left overnight for drying, and this material used for the purification of water. Next, please. Now, the method of water decontamination. The required amount of banana peel carbon was added to the desired strength of methylene blue and CVB dyes. After stirring the solution for the pre-selected time, the solid mass was filtered. The remaining amount of these dyes, methylene blue and CVB in filtrate, was measured spectrophotometrically at 663 nanometer and 585 nanometer respectively, with the help of prepared standard graph plotted between absorbance versus concentration. Next, please. Now, I'm going to results and discussion. The effect of variation of factors on removal of dye, like time of treatment, pH, initial dye concentration, amount of adsorbent are changed one by one, keeping other variables constant to find out the optimum condition. From the given figure 2, we can see here graph A and D. When we increase the time of treatment and the amount of adsorbent, the removal efficiency is increases, but at equilibrium it will constant. From figure B and C, at pH 6.5 and 5.5, the maximum removal efficiency when the solution concentration is 10 ppm. Next, please. Now, I'm discussing here the characterization of banana peel activated carbon. Changes in the resulting material before and after activation with acid. The treatment were studies by different techniques such as XRD, SEM-EDX, and FTIR measurement. From the figure 3, we can see here the XRD spectra presence of a broad peak in the range of 20 to 30 degree demonstrate typical amorphous nature of amorphous carbon. From figure 4, 5, and 6, this shows the SEM-EDX spectra of banana peel carbon after the treatment with methylene blue and CBB dye. The surface of the peel carbon to be an rough surface having carter-like pores, as they are partially wrapped by dyes. After adsorption, the pores got thicker in size and blocked with these dyes. Now, the FTIR spectra of banana peel, figure 7, 8, and 9 shows that some peaks disappear or decrease in intensity in banana peel after treatment with dyes. After the loading of dyes, appearance of new bands and disappearance of bands along with shifting of the bands indicate that the dye is adsorbed on the surface of banana peel carbon. Next, please. Now, in conclusion here, my work, the present study with gives a method to prepare the value-added product from one of the agricultural waste and the presence of a cost-effective alternative to existing commercially available adsorbent. It has been shown that the amorphous nature obtained after the acid activation of the carbon of banana peel is an effective adsorbent for removal of dyes from aqueous solution. At room temperature, only 20 milligram of the prepared material was able to remove 99.8% cationic dye methylene blue and 99.3% anionic dye CVB from contaminated water, having 10 ppm dye in 30 minutes and 45 minutes respectively. Thanks to all of you.
The house is open for discussion.
Hey, Mahesh ji, thank you very much for this wonderful presentation, and you have really done a remarkable work in the field of decontaminating water.
Yes, sir.
So many congratulations to you.
Thank you, sir.
Mahesh ji, there are a couple of queries which I had, just my own personal clarifications.
Yes.
The first one is, the first clarification is that what is the toxic effect of methylene blue and CBB dye on water such that it needs to be removed?
Yes.
What is the toxic effect?
Yes, sir. Many dyes. This is the from textile industries. Here, the textile pharmaceutical use this and infected to intestine and other type of disease.
मैं
So that
मूल रूप से ये जानना चाह रहा हूँ कि ये जो 2 डाइज की बात आप कर रहे हैं कि इनको हम लोग पानी से हटाएं, तो इनका प्रभाव किस तरह का पड़ता है मानव शरीर के ऊपर या पौधों के ऊपर या मिट्टी के ऊपर कि इनको हटाना और इसको अध्ययन करना आवश्यक बन जाता है?
जी सर जो कलरिंग मटेरियल है इसको सर पीने की वजह से सर दिक्कतें सर आती हैं, ऊतक में भी सर कुछ प्रभाव पड़ते हैं और सर बीमारियां इससे होती हैं। तो सर से मतलब क्लीन ड्रिंकिंग वाटर के लिए इन कलरिंग मटेरियल का रिमूवल सर जरूरी है। इसके लिए सर मैंने ये ग्रीन सिंथेसिस से बनाना पी यूज किया।
अच्छी बात। इसका मतलब कि ये जो पर्टिकुलर डिकंटामिनेशन है पानी का, ये उन्हीं क्षेत्रों में ही लागू है जहां पर कि टेक्सटाइल इंडस्ट्री है और इंडस्ट्री जो है वो लिक्विड एफ्लुएंट को बिना ट्रीट किए ओपन एनवायरनमेंट में रिलीज करती है ताकि वो सो लिहाजा आपका काम जो है मूल रूप से ये है कि ये पानी साफ रहे, पीने योग्य रहे।
यस सर।
दूसरी बात मैं महेश सर ये जानना चाह रहा था
यस सर
कि बनाना पील आप इस्तेमाल कर रहे हैं या बनाना पील एक्टिवेटेड कार्बन आप इस्तेमाल कर रहे हैं?
सर मैं बनाना पील एक्टिवेटेड कार्बन इस्तेमाल कर रहा हूँ।
इसका मतलब ये हुआ कि बनाना पील को आपने पहले उसको कैरमलाइज किया होगा, जला दिया होगा और उससे जो कार्बन प्रोड्यूस हुआ उसका आपने इस्तेमाल किया, राइट?
जी सर।
तो क्या आपने बिना इसको जलाए केवल बनाना पील को इस्तेमाल करके डिकंटामिनेशन करने का प्रयास किया सर?
यस सर। सर
क्या
मैंने
हुआ सर उसका?
सर वो प्रभाव ये हुआ कि सर जो बिफोर बर्निंग में सर ये उतना ज्यादा रिएक्टिव नहीं मुझे मिला जितना सर आफ्टर एडजॉर्बमेंट बनाने के बाद ऐश के रूप में यूज किया, उसकी वजह से सर वो वाटर बिल्कुल क्लीन हो गया। इसका
मतलब कि जो बनाना पील से जो कार्बन प्रोड्यूस हो रहा है वो कार्बन का इसके ऊपर डिकंटामिनेशन में ज्यादा रोल है ना?
जी।
तो ये कोई और दूसरा बायो मटेरियल आप जैसे मान लीजिए आप संतरे का छिलका लेके उसका एक्टिवेटेड कार्बन तैयार करें,
यस सर।
क्या प्रभाव आप क्या सोचते हैं, प्रभाव सेम होना चाहिए या प्रभाव कुछ अलग होना चाहिए?
सर प्रभाव तो सेम होना चाहिए बट सर मेरा जो मटेरियल था मैंने सर कोकोनट, क्वायर, बगाशी और राइस हस्क के कार्बन को भी सर की मैंने ऐश बना के स्टडी की, but my material is better than that adsorbent.
ठीक। Thank you so much. Thank you.
Okay, thank you sir. Thank you.
धन्यवाद।
Thanks to all of you.
Next speaker is Ms. Rakshanda Jabbar. Rakshanda Jabbar, I have unmuted on my side. Unmute yourself from your end. Her abstract number is ES10R. She is research scholar from Department of Botany, University of Allahabad, and her topic is impact of COVID-19 lockdown on water quality of Indian aquatic environment.
Ma'am, slides are visible?
No. I'll tell you. Whenever it's visible, I'll let you. Right now, we can see the slide of Mahesh Gupta only. Yes, now it's visible. Rakshanda, Ms. Rakshanda Jabbar. Unmute from your side. Hello, Rakshanda, can you hear us?
I request to Nishi ma'am kindly assist.
She's unmuted. She's unmuted. Hello.
She's unmuted.
Miss Rakshanda?
Yes, ma'am. I'm audible?
Yeah, you're audible. Go ahead.
Okay, ma'am.
Ma'am,
good afternoon, everyone.
Hello.
No.
Anuradha ma'am?
Yes.
Anuradha ma'am?
Yes.
Slide has been changed. No, it's okay. Fine. Just lower bit. Now you can continue. Rakshanda, you can continue.
Okay. Good afternoon, everyone. I am Rashna Jabbar, and my topic of presentation is the impact of COVID-19 lockdown on water quality of Indian aquatic environment. I am a research scholar under the CMP College, University of Allahabad, under the supervision of Dr. Anita Singh. Next slide, ma'am. Next slide, ma'am.
Is the problem and objective of this study. I'm showing first slide. Second slide.
Second slide, ma'am. In this presentation, I have focused on the two major problems, that is increasing water pollution as well as. Later slide, ma'am. Can you please? In this presentation, I have focused on the two major problems that are water pollution increasing as well as how the coronavirus pandemic impact the economies. My objectives are to study the impact of COVID-19 lockdown on water pollution in India and to develop a method for mitigating the pollutant from the water bodies. Pandemic COVID-19 imposed total lockdown, which has restricted the anthropogenic activities and banned on all commercial as well as industrial activities across the world. These are my assessment, which I've shown you, and these are basically telling that how before and after the pandemic, the water quality has changed. These are the assessment which I have taken from the source related to Wimprot, Smart Water Magazine, and Economic Times. It shows how the Ganga and Yamuna water quality has changed, and we can able to easily assess them that they are changed. Next slide. Next slide, ma'am. Water quality has changed from the river before and after, as well as the drain. The drain system is also closed during the lockdown. Result. Here I've taken the indicators which are responsible for evaluating the good water quality of Ganga. These are dissolved oxygen, BOD, biological oxygen demand, and fecal waste. That how they have changed. Dissolved oxygen has increased, as well as biological oxygen demand is decreased, whereas fecal waste is also decreased. Next slide, ma'am. Next slide, please, ma'am. Yes. Result of a study I've continued further. According to Muhammad Arif, there is reduction of pH, biological oxygen demand, and COD, in which is the signal of a good water quality. According to Ali P. Yunus, there is improvement in surface water quality in terms of suspended particulate matter. In the Vembanad Lake, the longest freshwater lake in India, various news reports as well as social media posts indicate that life seems to be returning to the river. According to Dean Singh Rawat, there is reduction in the organic pollution level, which is responsible for destroying the river's self-cleaning property. Conclusion and future perspective. The problem is that how the lockdown is considered as fruitful, but in the recovery of water bodies. But lockdown hurt the economy of India badly, and the government unlocked all the human activities one by one, as a result of which various industries as well as agriculture sectors are open and increased the pollutant in the water bodies. Therefore, the method should be adopted to mitigate the pollutant from the water bodies, for which bioremediation is considered as one safe, self-expensive, less expensive, eco-friendly method for removing toxic chemicals from the water bodies. Short-term lockdowns can upgrade the standard of environment, but it hinders the economy. Today is the need of global concern to advance the sustainable development with the low environmental impact, and that is sustainable economy in coexistence with ambient environment. Bioremediation is a process which involves in which living organisms degrade toxic chemicals compound into less toxic or non-toxic substances. Thank you, ma'am. Thank you, ma'am.
Any questions?
May I allow to ask any question?
Of course, ma'am. Of course.
Yes, ma'am.
Congratulations, Miss Rakshanda. वन क्वेश्चन, ये जो आपने वाटर सैंपल लिया था तो वो आपने कितने पॉइंट से लिया था, किस जगह से? इसमें असल में जो स्लाइड्स आ रही हैं ना वो प्रॉपर डिस्प्ले नहीं हो पा रही हैं, जो आप बोल रही हैं वो ये कोरिलेट नहीं हो पा रहा, उसमें वो स्लाइड नहीं आ रही है, दूसरी आ रही है, इसलिए मैं उसको स्किप कर गई हूँ लगता है। तो आपने वाटर सैंपल जो लिया था वो कितनी जगह से लिया था, कौन से पॉइंट से लिया था?
Ma'am, I have not taken still the water sample because I am just enrolled in the CMT degree college. Okay, ma'am. Ma'am, I've just enrolled in the CMT degree college, and therefore I'm just reviewing the paper. And the data I have taken from the source, and that is the Times of India and from the Google.
Okay. Thank you, sir.
Thank you, ma'am.
So shall we move forward?
Yes,
okay. Next presenter is Ria Johnson. Her number is ES11R and she's from Plant Physiology and Biochemistry Division, Department of Botany, University of Calicut, Kerala. Her topic is cross-adaptation tolerance of antioxidants in cowpea seedlings to sodium chloride and PEG stress. Ria Johnson.
Ma'am, let me know the serial number.
Serial number is ES11R.
No, ma'am, serial number, not abstract.
Serial number 5. Serial number 5.
Serial number 5. One minute.
By the time Ria Johnson, you may connect. Ria Johnson, unmute from your side.
Serial number 5.
This slide which is displayed. Okay, Ria Johnson. Okay.
Yeah, now it's visible, ma'am. Ria Johnson.
Yes, ma'am. Am I audible?
Yes.
Okay. Okay. So myself, Ria Johnson, and I'm a research scholar from Plant Physiology Biochemistry Division, Department of Botany, University of Calicut. So my topic is cross-adaptation tolerance of antioxidants in cowpea seedlings subjected to NaCl and PEG stress. Moving on to my introduction. So moving on to my introduction is that global climate change and increasing human population is a big challenge to the global food production system. And as an outcome of this global climate change, the environmental stress factors might induce several damages to the crop plants in the coming day. When it comes to the environmental stress factor, it's being two types, that is biotic as well as abiotic. When it comes to biotic, they are the living organisms. And when it comes to the abiotic stresses, they especially include salinity, drought. But it's not limited to salinity and drought. They also include highlight, high temperature, and chilling stress too. So these somehow the salinity and drought have a major effect on the case of reduction in growth as well as yield loss of the plant. Now, when it comes to salinity, it is energetically cheap osmoticum because it requires lesser accumulation of the organic compound as that compared to that of synthesis of this organic compatible solute. But in the case of this PEG stress mainly cause dehydration. That normally they cause when their plants are exposed to these dehydration stress, they strongly rely on synthesis of compatible solutes for the maintenance of the cell turgor. Next, please. So next is moving on to my objectives. My main objectives include the ROS production, then lipid peroxidation, membrane stability index, and antioxidation machinery. So these are the major factors which have been analyzed by myself, and these all include the biochemical and the antioxidation machinery. Next slide, please. So next is materials and method. So what I have selected here, the crop is cowpea, and we know that cowpea is also called as Vigna unguiculata, the scientifically termed, and the variety we have selected is KBC4, which is an unreleased variety. And the seeds were collected from Regional Agricultural Research Station, Pattambi, Kerala. And when it comes to cowpea, they belongs to the class Dicotyledone, order Fabaceae, family Fabaceae, subfamily Faboideae, tribe Phaseolae, genus Vigna, and species Unguiculata. This Vigna unguiculata, or called cowpea, it's also been called as poor man's meat because it's rich in protein, and it's also involved in the case of nitrogen fixation. So it's highly important. That's why I have selected the cowpea. Now, in the case of growth condition, we have selected seeds of uniform size. Then they were surface sterilized. They washed with mercury chloride, and then and then again washed with distilled water. And then on the right side, you have see the picture that it's been grown in a culture bottle, which is containing the adsorbent cotton, and then soaked with 1/4 of the Hogland medium. Next slide, please. So next, what we have selected is the seedlings. They were grown, the bottle, they were grown in a controlled condition of particular temperature, light intensity, and relative humidity, and all the analysis were done on the 8th day of the seedling. Now, there are two types of stress we are going to be subjected to cowpea. That is, one is NaCl-induced salinity, and the next one is PEG-induced drought. But it's not limited to drought. They also induce osmotic stress. So in the case of NaCl, it was 75 millimolar, and in the case of PEG stress, it was 15%, which was fixed as a concentration, which had the potential to impart 50% of growth retardation. Next slide, please. Now, when it comes to stress tolerance potential, the biochemical assays include lipid peroxidation, membrane stability index, antioxidation machinery, which include the non-enzymes and the enzymatic ones. In the case of non-enzymatic, it includes total phenolic, ascorbate, and glutathione, but in enzymatic, we have included SOD, that is superoxide dismutase, catalase, ascorbate peroxidase, and glycol peroxidase. Next slide, please. Now, what we have done is, moving on to our result, we have lipid peroxidation and membrane damage. So towards the right side, the graph, first one represents the MDA content, that is malonyl dialdehyde, which represent the damages which been caused to that of the membrane. So it's the ROS production, that is a reactive oxygen species. And we can see that NaCl treated seedlings were accumulating more MDA as compared to control. But when comparing to that of the PEG stress, it was higher, that is of around 62%. Then again, in the next slide, again in the next graph, we can see the membrane stability index. It was also showing that PEG has a higher rate of membrane damage. So the lesser membrane stability index represent higher membrane damage, which was lesser in the case of the NaCl. Next slide, please. In the case of ascorbate content, we can see the higher accumulation of ascorbate was slightly, that is 289%. A significant increase can be seen, and it was 289% in the case of NaCl-induced salinity stress, whereas 45% in the case of PEG-induced drought stress. And we can, or this can be indicated, can be concluded that the accumulation of this ascorbate is due to the enhanced activity of ascorbate synthesizing enzymes, and the subsequent decrease in the catabolizing ones. Next slide, please. So next slide is the glutathione. In the case of glutathione, we can see that the NaCl treated seedlings has higher accumulation of glutathione, that is around 47% as compared to that of control, whereas in the case of PEG, it was just around 25%. And according to Gibson, 2005, it's been said that increased accumulation of glutathione content regulate the osmotic stress, which has been caused by the ROS production, and then that leads to the preservation of the biological molecules and the membranes. Next slide, please. Then in the case of total phenolics, again, we can see that the same, again and again, we can see the same pattern, that is NaCl induced a higher accumulation of phenolics. And phenolics, they act as a non-enzymatic antioxidant during the stress, and they're mainly involved in the growth and the development. And its increase was around 45% in the case of NaCl, whereas in the case of PEG, it was 24%. Then total soluble protein was still higher in the case of NaCl, and significant decrease can be seen in the case of PEG-induced stress when compared to that of NaCl. Now, this protein content, high increase in the protein content, could reflect a strong defense response to the cellular damage which is caused by the NaCl ROS production. Next slide, please. Next, we have SOD, that is superoxide dismutase and the catalase activity. So superoxide dismutase and catalase, the main function is what they do is they just, whatever the ROS generated, the superoxide and the hydrogen peroxide, SOD first converts the superoxide into that of the hydrogen peroxide, and then the catalase ascorbate peroxidase and glycol peroxidase convert that hydrogen peroxide into that of water and the oxygen. So this is the main enzymes which convert and detoxify the ROS production. So here we can see that NaCl has a reduction in the case of SOD activity, whereas PEG, it's higher in the case of SOD activity. And again, in the case of catalase, we can see an increased cat activity was observed in the case of seedlings, which are subjected to PEG when compared to that of NaCl. Moving on to the next slide, we have ascorbate peroxidase and glycol peroxidase. In the case of ascorbate peroxidase, we have an increased ascorbate peroxidase in the case of NaCl when compared to that of PEG stress. And I have already said that NaCl, the ascorbate peroxidase and glycol peroxidase, they are mainly involved in detoxifying the hydrogen peroxide into water and that of oxygen. Moving on to the next, what we have is summary and conclusion. So from the above, we have concluded that the cowpea seedling, when subjected to NaCl and the PEG stress, they results in accumulation of ROS, that is reactive oxygen species, and thus they cause an oxidative stress. When it comes to oxidative stress, they leads to membrane damage through the increased rate of lipid peroxidation, the MDA, and then the decrease the membrane stability index in the PEG seedlings. Now this increased rate of enzymatic and the non-enzymatic antioxidants, they effectively scavenge the excess RS accumulation in NaCl as well as in PEG stress, but it was little bit less in the case of the PEG stress. When it comes to the non-enzymatic, the phenolics, they accumulate because they are mainly involved in the function of that intermediate, act as an intermediate in lignin biosynthesis, and they're also involved in preserving the plant cell by building a physical barrier. So this induction of phenolics accumulation observed in the study proves that oxidative stress. Now, overall, we can conclude that the seedlings were able to avoid salt stress much better than that of the PEG stress. When it comes to salt, it mainly causes ionic imbalance and toxicity, but the PEG stress caused two types of stresses that to cope up with the dehydration stress and the osmotic stress imposed by the PEG stress, but it was able to be grown in a condition of high salinity. And thus the tissue, they have used the mineral component of the nutrient medium to attain a level of ion homeostasis, but they were unable to cope up with the dehydration stress imposed by the PEG. So from the above, I have concluded is between NaCl and PEG stress in cowpea, the tolerance response has been shown by NaCl rather than that of the PEG stress. So in our field condition, we can, in a high salinity or high salt concentrated area, we can grow cowpea rather than that of rather than in a dehydrated area. Thank you.
The chairpersons can have their question.
Am I allowed to ask a question now?
Of course, sir, you are there.
Thank you. First of all, I'd like to congratulate Madam Ria Johnson for this wonderful work that you've been doing, and I'm sure that the direction in which you're moving is definitely going to bring forth a lot of good result. There's just one query. Is Ria Johnson listening to me?
Yes, sir. Yes, sir.
Okay, wonderful. Ria, you have done all your work with sodium chloride, right?
Yes, sir.
So I wanted to know what is going to be the impact if you replace sodium chloride with, say, potassium chloride or lithium chloride?
Actually, I haven't worked with potassium chloride, but according to my results, why I have selected NaCl is because salt is rich in our nowadays the soils are rich in salt because of the irrigation and as well as the anthropogenic activity. Mainly these are the two reasons. And to cope up with these, we have to somehow develop the crop. We have to make that crop tolerant to the stress, or
No,
otherwise we can.
I got that part. See, my contention of asking you this question is that there is something that sodium chloride is doing. You are using sodium chloride for this particular piece of research.
Yes, sir.
My query is, whatever sodium chloride is doing, can that be done with a stronger electrolyte or a weaker electrolyte? And if yes, then would you consider using that electrolyte? And if your answer is going to be no, then there must be something very distinguishing or something very specific that NaCl is doing, such that lithium chloride or potassium chloride or rubidium chloride or any other magnesium chloride, etc., cannot be used. So that is my question. Is not with any other part. My question is just pertaining to the use of NaCl, particularly for your piece of research.
So actually, I haven't moved to potassium chloride, but it's a good question. So, if possible, I would wish to do that part of work too.
Sure. You are pursuing your PhD, right?
Yes, sir.
Ria, my gut feeling, my suggestion as a senior in this field is that, I mean, even if you're not taking that part, that part in your PhD, still just for your general information, if you can keep that in your mind, I think it will help you in your Viva OC as well.
Yes, sir, of course, sir, I will do that.
So much.
Thank you, sir.
Thank you, sir. Ma'am, do you have any question?
I think Neerja ma'am is in waiting room.
Out of the link, I think so. So moving on, our next presenter is Ms. Mansi Sharma, ES12R. She's research scholar from Biotechnology, JP Institute of Information Technology, Noida, and her topic is exploring of anti-cancer and other pharmacological potential of Musa paradisiaca peel extract. Mansi Sharma.
And slides are visible or not?
No, not right now. Yeah, now it's visible.
Ma'am, should I start?
Yes.
Yeah, sure.
So good afternoon, everyone. I'm Mansi Sharma, scholar in JP Institute of Information Technology. I'm looking forward to talking with you today about the topic exploring anti-cancer and other pharmacological potential of Musa paradisiaca peel extract. Next slide, ma'am. As we go with introduction, when we talk about cancer, as we're exploring the anti-cancer activity of the banana peel, we need to talk about cancer as well. So it's an unchecked cell growth, a disease in which the abnormal cell starts growing rapidly in an uncontrolled manner, and they destroys the other body tissues as well. There are many therapies available in the market for the treatment of cancer, such as the chemotherapy, the UV radiation therapy, the surgery, and immune therapy, and many others. Apart from the usage, these therapies have several side effects as they are highly expensive, they are invasive, they have toxic side effects. So scientists all over the world, they're exploring new therapies, new treatments for the cancer, which can be harmless, non-invasive, economic, patent compliant, and target therapies. This review basically focuses on the treatment of cancer through the natural products such as banana peel, and which has shown to be much more effective based on the past reviews. The scientific name of banana is Musaceae paradisiaca. It's a tropical plant, and it protects itself from the oxidative stress caused by the strong sunshine and high temperature by producing large amounts of antioxidants.
Hello. Mansi.
Hello.
It's been noticed that banana peel extract of human cancer and normal cell lines. This present investigation is undertaken to evaluate the antioxidant and anti-cancer power of banana fruit peel and to identify the responsible compounds for those activities. Next slide, ma'am. So as we know this, I'm telling you that this survey, it's totally based on the literature review. So the basic mechanism, assays and methods used for the peel extraction and exploring the anti-cancer and antioxidant properties of banana peel. For the extraction, we use ethanol as it's a polar solvent. It readily mixes with water, breaking down water-soluble molecules. The second is determination of antioxidant activity, which can be done with the DPPH radial scavenging assay. This is a free radical scavenging, a mechanism for screening the antioxidant activity of plant extracts. In this assay, the violet colored DPPH solution is reduced to yellow colored product. The second is the MTT assay. It is using for exploring the anti-cancer activity of the banana peel. It's a standard colorimetric assay. It also predicts the degree of cytotoxic effect. In this essay we use the compound diphenyl tetrazolium bromate. Why we use this essay for exploring the anti-cancer activity? Because the plant extract, they shows best activity with petroleum, ether, chloroform. The second is the antioxidant assay using the ferric thiocyanide methods. While we explore the anti-cancer activity, we have to apply it on the cell lines. So different cell lines culture conditions are provided to them, such as the cell growth. It has to be kept in the cell growth medium. Next slide, ma'am. Based upon the previous results, I've listed here the surveys of few people. Firstly, one Shruti et al. In 2019, she reviewed that the ripe banana yellow peel with dark patches on skin produces substances called tumor necrosis factor, which is the ability to combat normal cells. The study revealed that darker the patches, higher will be the immunity enhancement quality. Hence, riper the banana, the better the anti-cancer quality. Ahmad in 2015, he stated that banana seemed to have highly significant cytotoxic activity when tested over the cell lines. In the same year, Dharman and his team, they found that the hexane extract of the banana peel extracts the highest toxicity activity on the HCT116 and MCF tumor cell lines. Secondly, Jyoti Marathi reviewed that the banana plant extract contains high level of tannic acid, inhibited the tumor cell proteomics activity, which may contribute to the claimed anti-cancer activity. Mogwell investigated the green banana peel and the yellow banana peel, and they resulted out that the extract of the green banana
Hello. Hello, Mansi.
Yes, ma'am. Next slide. We discussed that the banana extracts, it have higher amount of scavenging activity. This activity in them is due to the antioxidant present in them that helps them to protect cells from the damage caused by the free radicals. These free radicals are unstable molecules that are made during normal cell metabolism. Cytotoxicity of the ethanolic peel extract on human breast cancer carcinoma cell line MCF7 was detected by measuring the percentage of cell viability using the SRB method. SRB is sulforhodamine B assay kit. It is used for testing the cell viability. Two assays were used to assess the tumor genesis effects of the banana extract. Cytotoxicity of the extract was measured using the MTT assay, as we have discussed, which shows the cell viability via mitochondrial activity in living cells. Green banana peel displayed high antioxidant activity. It was measured by the B-keratin bleaching method, DPPH, free radical, and linoleic acidic emulsion. The mechanism aspects of the fruit bioactive components may prevent cancer through various modes of action, including antioxidant activity, inhibition of cell proliferation, induction of apoptosis, inhibition of cell invasion, and subcellular signaling pathways. Next slide, ma'am. Basis on the review lead to a conclusion that the new therapeutic in the form of natural products such as banana have appeared to be significant as it is capable of targeting tumor and thus results in decreasing graph of severe side effects. Phytochemical compounds from the extract of plant roots, bulbs, bark, leaf stems, and others have shown promising potential as anti-cancer drugs or for serving as a lead component compounds in the synthesis of new drugs. Therefore, targeting tumor angiogenesis using natural anti-angiogenic agents that inhibit the growth of new blood vessels which supply the tumor with nutrients and oxygen represents a promising therapeutic approach against tumor growth and metastasis. Thank you, ma'am.
Question?
Ma'am, it's done.
Is there a question? I'll request chairperson to.
I don't have a query, ma'am. Thank you so much.
Ma'am. Okay, so moving on, we have our next presenter, Ms. Mubashra Afroz, research scholar from Department of Chemistry, University of Allahabad. Her abstract number is ES13R, and the topic is Impact of COVID-19 on Indian Atmosphere. No. Mubashra Afroz.
Ma'am, screen or
Yes, ma'am. Yes, ma'am. Mubashra Afroz, unmute from your side.
Yes, ma'am. I have unmuted.
Oh, fine. Start.
I'm audible?
Yes.
Okay, ma'am. Good afternoon, respected teachers, chairpersons, and all faculty members. This is Mubashra Afroz, and I am pursuing PhD under the supervision of Professor P.K. Tandon, Department of Chemistry, University of Allahabad. And my topic is Impact of COVID-19 on Indian Atmosphere, Case Study, Lucknow. Next slide, please, ma'am. Introduction. A novel coronavirus COVID-19 or SARS-CoV-2, which is an acronym for Severe Acute Respiratory Syndrome. Coronavirus second is a third pandemic in recent 2 decades after SARS-CoV-2 2003 in China and MERS-CoV 2012 in Saudi Arabia. This coronavirus affects humans through zoonotic transmission, and its first case was reported in Hebei district of Wuhan, China in 2019. Its global transmission, repeated emergence, and its infection and mortality rate, World Health Organization declared its pandemic in 11th March. Here is the Sustainable Development Goals. These are the collections of 17 global goals. Ma'am, first slide, please. Yes, ma'am. First slide of introduction. Yes, ma'am. Sustainable Development Goals are a collection of 17 global goals set in 2015 by United Nations General Assembly, including social, environmental, and developmental dimensions to achieve a better and more sustainable future for all by the year 2013. Due to this pandemic situation, no country is in a position to meet this path. However, to stop the spread of coronavirus, many countries have put a lockdown. In this lockdown situation, most of the human activities were paused, which affects the environment, and give it a healing time. India is a developing country and a pollution hub due to its rapid industrialization. According to World Air Quality Report of 2019, India has 20 cities in world's third most populated city, in which maximum number of polluted cities are of Uttar Pradesh. My study area is Lucknow, which is the state capital and also a highest populated city of Uttar Pradesh. Lucknow ranks 11th position in recently published Indian Air Quality Report of 2019 with a particulate matter concentration of 90.3 microgram per meter cube, which is higher than the permissible limit of 60 microgram per meter cube. Here in this work, I have contemplated data of 5 pollutants of air quality index, which are nitrogen dioxide, sulfur dioxide, carbon monoxide, particulate matter 2.5, and ozone, to observe the improvement in air quality before and lockdown. All data are collected from Central Pollution Control Board online portal. Additionally, I have compiled a satellite image of NASA to show an overall decrease in concentration of nitrogen dioxide and sulfur dioxide in atmosphere. Next slide, ma'am. Next slide, ma'am, please. Here are the graphs of my analysis. Average concentration of all 5 pollutants are taken and dates were randomly selected of different weeks of different months. where first initial 4 dates are 20 January, 10th February, 28th March, and 20 March, which shows pre-lockdown condition. And the next 3 dates are of 10th April, 30th April, and 20 May, which shows lockdown condition. And the last 2 dates are of 10 June and 30 June, which shows post-lockdown condition. Also, data of same dates were collected from previous years to make comparison. Here is the first red line, which shows particulate matter concentration permissible limit, and this mustard line shows concentration of pollutant this year, and the blue line shows concentration of pollutant previous year. The particulate matter concentration, here we can see in graph that the particulate matter concentration before lockdown was high as compared to after lockdown. Also in comparison to previous year, this year condition was normal. And the second graph shows concentration of nitrogen dioxide, which is in permissible limit, but its pre-lockdown concentration was quite high in comparison to post-lockdown. And also this year concentration is less in comparison to last year. Possible reason behind this would be a pause in transportation. And the third graph shows concentration of sulfur dioxide. Here also its concentration is in permissible limit, but no significant decrease was observed during lockdown. Possible reason might be running of industries, because sulfur dioxide is mainly emitted from industry, which are continued during lockdown condition. However, in comparison to last year, this year concentration was little less. And the fourth graph shows concentration of carbon monoxide. Its concentration is in permissible limit, but in comparison to pre and post lockdown period, its concentration decreases. In comparison to last year, its overall concentration was increased. Graph, next slide, ma'am. Here is the fifth graph, which shows permissible limit in 180 microgram per meter cube. Here the concentration of ozone is in permissible limit, but its concentration was high after first lockdown, and then concentration decreases after second lockdown. This abrupt increase or decrease in its concentration is dependent on the other pollutants, because ozone is a secondary pollutant. It is formed by the photochemical dissociation of nitrogen dioxide. Here is the table which shows the percentage reduction of pollutant concentration before 3 months of lockdown and before 2 months of lockdown. Here we can see in table 50, particulate matter concentration removal rate decreases concentration, and nitrogen oxide concentration decreases, sulfur dioxide concentration decreases, carbon monoxide concentration decreases, but ozone concentration increased. But in between but most significant decrease was shown in nitrogen dioxide concentration. Next slide, ma'am. Here is the satellite image of Aura NASA, which I had collected from NASA Earth Observatory. Here the upper two images show the concentration of nitrogen dioxide in upper Indian atmosphere. The dark pixel shows a concentration of nitrogen dioxide in Indian atmosphere, which was in right image shows less. However, the below two images shows sulfur dioxide concentration in Indian atmosphere. Red pixel shows high concentration, and on right side we seen that its concentration has been decreased. Conclusion, ma'am. Ma'am, next slide, please. Ma'am, next slide, please. Conclusions. COVID-19 pandemic originated in Wuhan has restricted human activities in India since. Ma'am, next slide. Ma'am, previous one, please. Has restricted human activities during mid of March, due to which its impact has been seen on environment. The air quality of Lucknow showed a trend of a decrease in concentration of pollutants except for ozone. However, nitrogen dioxide shows a maximum decrease, and particulate matter shows a significant decrease in its concentration and reached up to a permissible limit, which was earlier higher than the permissible limit. If the satellite image of nitrogen dioxide and sulfur dioxide shows a decrease in its concentration from the previous year, undoubtedly we can say that COVID-19 brought a fearful, devastating situation for human beings, but emerged as a blessing for natural environment. The situation today is a reset for nature to which mankind has to observe, analyze, and accordingly reorganize ourselves. Thank you. Next slide. Ma'am, I've done.
It's okay. A request to all the participants, please restrict to your time limit. Don't exceed beyond 5 minutes. Do we have any questions from the chairpersons? Do we have?
No question. No question.
Thank you.
So next participant is Akash Kumar Singh. He is 14. He's from School of Environmental Sciences, JNU Delhi, and his topic is carbonaceous aerosol pollution during winter season. Delhi, India. Oh, Mubashra, please mute your mic. Akash Kumar Singh.
Good afternoon, everyone. My name is Akash Kumar Singh, and I am a research scholar, PhD research scholar in a School of Environmental Sciences, Jawaharlal Nehru University, Delhi. Today's title of my presentation is Carbonaceous Aerosol Pollution During Winter Season in Delhi, India. I am starting this, ma'am, next slide. So I am starting this presentation with a brief introduction to the topic. Aerosol are well defined as a suspension of solid, liquid in gaseous medium in the form of dust, fume, smoke, spray, smoke suits, etc. So a number of chemical species are found associated with the different various size of particle fraction, and among that chemical species, carbonaceous fraction of atmospheric aerosol are becoming the matter of great concern in the world because it play important role in the formation of smog, degradation in visibility, global warming, melting of glacier, and adverse effect on health. So carbonaceous fraction of aerosol is generally classified into black carbon or elemental carbon and organic carbon. Elemental carbon is also known as black carbon. So elemental carbon is a primary pollutant and it is emitted as a result of incomplete combustion of fossil fuel and biomass burning. It does not undergo any chemical transformation and it has property to warm the atmosphere by absorbing the light. While OC, organic carbon, is composed of a number of aliphatic or aromatic compounds and it is released into the atmosphere by combustion as a primary organic carbon, or it can be produced as a secondary organic carbon by photochemical reaction in atmosphere. It is a scattering in nature and it cools the atmosphere by reflecting the sunlight. So emission from vehicle movement, industrial activity, biomass burning, coal combustion, or fossil fuel burning are some important source of carbonaceous aerosol in the atmosphere. Ma'am, next slide. Therefore, the main objective of this study was to investigate the variation of OC and EC in PM1 size fraction in the industrial area and residential area of Delhi. So for this purpose, aerosol sampling was performed in the residential area and industrial area in Delhi with the help of cascade impactor, and the sample was analyzed with the help of thermal optical carbon analyzer. So this is the cascade impactor. It collects aerosol particle in 4 different size fraction in the range of more than 10 micrometer and 10 to 2.5, 2.5 to 1, and at the bottom part it collects all the particle less than 1 micrometer. Next slide, ma'am. So coming to the result and discussion part, figure 1 shows the particulate matter concentration deposited in 4 different size fraction. So a clear difference in the mass concentration of particulate matter in 4 size fraction was observed between the 2 different size, and among the 4 different size, it's a 4 different size. Among the 4 different size, highest deposition of particle was observed in PM1 size fraction. While figure 2 shows the percentage contribution of fine and coarse mode particle at industrial and residential site. Here, the fine mode particle dominated the coarse mode particle at both the sampling site, and its average contribution was more than 70% at both the sites. Next slide, ma'am. In case of organic carbon and elemental carbon, a clear difference in the mass concentration of OC and EC was observed between the two different sites. The concentration of OC and EC was found higher in the industrial area as compared to the residential area. In case of organic carbon, in industrial area, it was approximately 1.5 times, and in case of EC, it was approximately more than 2 times higher concentration was in industrial area as compared to the residential area. So we have also measured the OC-EC ratio, and it was found more than 2 at both the sampling site during the winter season, while the low OC-EC ratio were observed in industrial area as compared to the residential area. Next slide, ma'am. So coming to the conclusion part, a clear difference was observed in the mass concentration of organic carbon and elemental carbon at both the site. The highest concentration of organic carbon, elemental carbon, and particulate matter deposited in all the 4 size fraction was observed in the industrial area as compared to the residential area. Among the 4 size fraction, highest deposition of particle was observed in the particulate matter of PM1 size range. So overall, fine mode particle dominated the coarse mode particle during the study period. The higher concentration of OC, EC along with PM can adversely affect the atmospheric property and health of the people. Therefore, efforts would be made to control carbonaceous fraction of aerosol along with the fine and coarse mode particle. Thank you.
Thank you, Akash. Now the session is open for discussion.
Yes, ma'am.
No queries, ma'am. Thank you.
Oh, thank you.
Thank you very much.
Okay, our next speaker is Ms. Anila Juhi from Department of Botany, Vinoba Bhave University, Hazaribagh, Jharkhand. And our topic is phytochemical studies in some varieties of soybean and their hybrids. Anila Juhi, ES15R.
And screen is visible?
I'll tell you. Yes.
Hello, am I audible, ma'am?
Yes, yes. Continue.
A very good afternoon to everyone present over here. I'm Anila Juhi and I'm a research scholar at Vinoba Bhave University, Hazaribagh, Jharkhand. Today I'm here on this platform to share my findings on soybean. So on this platform, on this forum, I would presently emphasize on sustainability first. So a sustainable health and care system, this is achieved by delivering high quality care and improved public health without exhausting the natural resources or causing severe ecological damage. There are naturally present bioactive compounds in plants and they are called as phytochemicals, and they are studied extensively for their role in human health. Studies have shown that they can have an important role to play in the prevention and management of several human diseases. So basically, phytochemicals, they are non-nutritive bioactive plant substances which have beneficial effect on human health, like the phenolics, alkaloids, and flavonoids. Now, more focus is on crop legumes because it already has less amount of fats. It's an excellent source of protein. It has micronutrients and it has phytochemicals as well. And the chemical compounds, it has been found to protect the human tissues from the free radicals, and so they are gaining more and more attention in the pharmaceutical sector as well. Next slide, ma'am. With the methods, first of all, 5 varieties of soybean were obtained through the courtesy of NBPGR, New Delhi. They were the BRAC, Punjab 1, Pusa 9814, GS 9560, and NRC 7. And intervarietal hybridizations were performed, out of which the 11 intervarietal hybrids were obtained, and they were analyzed along with their parents. Ma'am, the previous one only. So they were analyzed along with the parents for the total phenolic content, total tannin content, total flavonoid content, and the antioxidant activity. Phenolic content was done using FC reagent, using gallic acid as the standard. Tannin content was determined by FC procedure using insoluble cross-linked PVPP. Total flavonoid content was calculated using aluminum chloride reagent, and antioxidant activity was done by DBPH method. Next slide, ma'am. Now the results. Here you can see the graph. There are the set of 11 bars, and in each there are a set of 3 bars. The middle one is representing the F1 hybrid, And on either side of it, the parents are present over here. So the total phenolic content in all the 11 F1 hybrids along with their parents are present in the screen. In phenolics, we saw there was increase in 6 hybrid varieties, while there was decrease also in 2 of them. Next slide, ma'am. In flavonoid also, there was substantial increase in one of the crosses, and there was few varieties which showed a reduced level of flavonoid from the parents as well. Next slide. With the tannin content, most of them showed increase in the total tannin content than either of the parents. Next slide. This was for antioxidant activity. The increased as well as intermediate value of antioxidant activity was seen in case of all the 11 F1 hybrids along with their parents. Next slide, ma'am. So what we found from the graph that there were higher levels of both phenolic contents and the flavonoids in some of the F1 hybrids like PNNR, NRJS, JSNR, BGJS, and PUJS. Out of which, except for BG cross JS, all showed intermediate DPPH radical scavenging activity also. And the hybrids like PUPN, JSNR, JSPN, BGJS, and PUJS, they were having higher levels of tannin content as well as total phenolic content. However, there was variability in the contents of other phenolic classes also. This was in accordance with the findings of other authors concerning phenolic contents and antioxidant activity, which have been done on common beans and other leguminous species. However, there was no correlation between the contents of other examined phenolic classes and antioxidant activity, which was observed. Next slide, ma'am. So now, what's the inference we are getting from all these experiments? So basically what is there? Humans are consuming thousands of different phenolic compounds through plants. Phenolics also have redox properties which allow them to act as antioxidants, thereby reducing oxidative damage. Phenolics may inhibit carcinogenesis by affecting the molecular events in the initiation, promotion, and progression stages. Flavonoids also have attracted increasing attention due to pharmacological and antioxidant activity. These may be used as additives in the food system also. So the potential source of antioxidant compounds have been searched in several types of plant materials like vegetables, fruits, oil seeds, spices, herbs, cereal crops, and so on. So what we concluded that our results suggest that besides the protein and oil contents, phenolic content should also be considered as an important feature of soybean. Some of the pharmacological effects of soybean could be attributed to the presence of these valuable constituents. And it seems that total phenolic content alone or in combination with other phenolic constituents, it becomes a potential candidate as a selection criteria for antioxidant activity in the soybeans. These are some of the references which I have studied. And then, finally, I would like to acknowledge Dr. Vineet Singh. He is from SGIPT, Indore, for helping me performing all these experiments. And definitely, I'd like to thank you, SS Khanna Girls Degree College, for giving me such a good platform to discuss my work. Thank you, everybody.
Thank you, Anila. So, are there any questions?
Can we take in place of soybean, can we take any other material or pea in place of soybean?
Ma'am, apart from soybean, these type of studies are going on with other leguminous species also. And arhar, in that also, the scientists are trying to work out this thing that there is increase in antioxidant activity as well. Common bean, most of the beans, means in one or the other bean, they are trying, but soybean is succeeding in this race as compared to other leguminous species. It's already having good protein content, good oil content. So if it gets an upper version of antioxidant activity also, that will be just a boon for us.
Okay, okay. Thank you so much. Thank you. Now, Dr. Anuradha, you can proceed. There are a number of participants who are waiting for their presentations.
Thank you, ma'am.
Next is Miss Fauzia Hamid, but I couldn't find her in the participant list. So, Miss Fauzia Hamid, are you there? No, she isn't. The next participant is Mr. Ayan Banik.
Ma'am, I think Ayan Banik is also absent.
So no, he's also not there. So we'll just frequently move to the next participant, Mr. Ashwini Sharma.
Ma'am, Ayan Banik's screen is visible or not? Please let me.
I don't think he's there. Ayan Banik is not
Actually,
there.
he requested me to show his slides.
Ma'am, sure, we can do it, but I think we should first of all entertain other participants. We'll do it afterwards.
But should I just show?
Oh.
I request to chairperson kindly see his slides. His topic is scientific disposable of face mask and PPE kit during the COVID-19 pandemic to ensure safe public health and sustainable environment.
Slide is not visible.
He requested me to show his slides only.
Is he going to make his presentation, ma'am?
No, sir. He's not here, but he requests me to show his slide.
So we can see his slide at the end, ma'am.
Okay.
Let us listen to the participants who are here first, and thereafter we can see his slide.
Next.
We have Mr. Ashwini Sharma. I have unmuted you. You just unmuted from your end. He's research scholar from Department of Chemistry, CMP Degree College, and his topic is clean and affordable energy, environmental sustainability.
Am I audible, ma'am?
Slides, ma'am. अभी नहीं दिख रही है मैम.
अभी नहीं दिख रही slide.
What is his abstract number, ma'am?
He hasn't been allotted the abstract, but I think so. Ashwini Sharma, are you there?
Yes, ma'am, I am audible.
Oh. You are audible.
Okay, ma'am.
Let's just start sharing.
Slides are visible or not?
No.
No.
I'll tell you whenever I'll tell you.
His abstract number is not mentioned in the list.
Yes, ma'am. Now it is visible?
Yes, now it's visible. Ashwini, you can start.
Okay. Good afternoon to all. Myself is Ashwini Sharma. I am a research scholar in CMP Degree College. I am pursuing research under the supervision of Dr. Archana Pandey. Today, I am delivering a presentation on affordable and clean energy, which also comes in environmental sustainability. First, we have to, next please. No, ma'am, please, previous. Okay, thank you. First, we have to understand what is sustainability. Sustainability is the development which completes our needs without compromising the ability for the future generation to complete their own needs. Or we can say it's sustained to continue over the time. So environment sustainability in a similar way is a responsible interaction with the environment to avoid depletion or degradation of natural resources and allow for long-term environment quality, because we use water for use, air to breathe, and in other ways we complete our needs from the environment. So we have to sustain our environment for a long time. So sustainability is natural environment, healthy communities, economic vitality. These are intercorrelate with each other. We have to sustain this all, not only the environment. Next slide, please. What are the issues? Why we are here to understand the sustainability. First, I want to tell we people are very selfish in nature, because we never try to give nature. We always try to give from take from the nature, from the, take from the food, from the air or the water. We never try to take. In a similar way, we can use a proverb that is, as long as there is no problem in our life, till then we never understand that problem. So now the problem comes on us, we are trying to solve these issues. So the big issue come what the continuous rise in CO2 emission over the last 30 years and growing global demand for stable electricity, heating, cooling, transportation represent a major challenge worldwide. Around 3 billion people are rely on wood, charcoal, coal for heating and cooking. Energy is the biggest distribution for climate change. Around 60% of greenhouse gases emits, around 46% of CO2 carbon dioxide increase globally. Hydropower is the energy which gives 60% worldwide at a very competitive price. Or bioenergy in the nowadays is used, which gives worldwide a 10% of its energy for the production and transportation. Next slide, please. So we get, there is a question, how we can ensure this clean energy. I'm talking about the clean energy or affordable energy. So how we can solve this? The answer needs a global response, which is why the United Nations has chosen energy as the focus of Sustainable Development Goal number 7. What is the goal number 7, or what is United Nations Development Program, established in 1951, and it is take a step in 2015 while looking about the sustainability for in a various ways. Next slide, please. By the world leaders, they took a 17 goals. Goal number 1, that is no poverty. Second, zero hunger. Third, good health and well-being. Fourth, quality education. Fifth, gender equality. Sixth, clean water and sanitation. 7, affordable and clean energy. 8, decent work and economic growth. 9, industry, innovation, infrastructure. 10, reduce inequalities. 11, sustainable cities and communities. 12, responsible consumption production. 13, climate action. 14, life below water. 15, life on land. 16, peace, justice, and strong institutions. 17, partnership for the goals. These are 17 goals mixed by the world leaders in the United Nations Development Programs. And here we are going to discuss about the goal number 7, that is affordable and clean energy. Next slide, please. In order to reach Sustainable Development Goal 7 and decarbonize the global energy system, solutions like clean energy sources, green heating, and energy efficiency measures must be implemented. The decarbonization and economic growth are not mutually exclusive. The most prosperous societies of the future will also be the ones that are most sustainable. Next slide, please. Now, what is clean energy? Clean energy or renewable energy, or we can say a green energy, which is used nowadays, that is produced through methods that do not release greenhouse gases or any other pollutants, so that our environment could be pollution free. Next slide, please. Types of clean energy which we are using. Solar energy, we all know about the solar panels. Wind energy, that is from the windmills. Geothermal energy. Bioenergy is that energy that we use from the plants which generate from the extract of the plant waste materials. Nuclear energy and hydrogen fuel cells, etc. Next slide, please. So a Sustainable Development Program takes a target to perform in a section, because we are performing and we are doing in areas work, but we don't have targets to perform. So United Nations Development Program took the action to end up by 2030. These targets are universal access to modern energy, which means we have to ensure universal access to facilitate the affordable clean energy and energy efficiency. Second, increase global percentage of renewable energy. We have to share the energy that is clean over the globally, so that we can make, please next, so that we can sustain our environment. Now we are doing clean energy and making in a worldwide, but now we have to double the improvement in energy efficiency by sharing it, or by rate improvement globally. Next, promote access to research, technology, and investment in clean energy. That is, we have to intercorrelate internationally to provide or facilitate the research in the clean energy and technology that we use in a modern time, so that we can make efficiency better of clean energy, and give investment or promote our money in a modern technology, so that we can use this type of clean energy in a very vast areas. By 2030, we have to expand and upgrade energy services for developing countries. It means that we have to expand our infrastructure and modern technology for supplying the green energy or clean energy in developing countries, or list developing countries, small islands, small areas, so everyone can use this green energy. That's why we can complete our targets or make our environment very sustainable. So these are intercorrelated to each other. Whether it is health, it is our economy, or it is our environment, because we are living in an environment which is just like our home. If we can't live in our home which is very polluted, so we can't live in our environment. So now it's time to take a step ahead, and we have to contribute each and every people, because we know that people have many potential to perform, but we are not doing it. So now we have to aware all the peoples how to make their environment sustainable, or how to use this clean energy and affordable prices. We have to make so that they can use this thing. Thank you, and stay safe and stay healthy.
Thank you, Ashwini. Ma'am, do we have any question?
No, thank you. Very nice presentation. Thank you so
Thank you,
much.
ma'am. Thank you, ma'am.
I have a query, ma'am. अश्विनी, बहुत बहुत बधाई, बहुत अच्छा प्रेजेंटेशन आपका रहा, और आपने में रिसर्च अपना शुरू किया है, बहुत ही बधाई के पात्र हैं आप। एक छोटी सी चीज मैं आपसे जानना चाह रहा था, और वो ये है कि आपने क्लीन एंड अफोर्डेबल एनर्जी की बात करी है। मैं ये जानना चाहता हूँ कि आप क्या समझते हैं सस्टेनेबल एनर्जी के विषय में? सस्टेनेबल एनर्जी आपके हिसाब से क्या चीज?
सर, सस्टेनेबल एक एनर्जी ऐसी है, जैसे हम लोग अभी कोई भी एनर्जी यूज कर रहे हैं, जैसे कोल से यूज कर रहे थे, पोल्यूट कर रही थी। मेरे साथ सस्टेनेबल एनर्जी ऐसे, जैसे हम लोग प्लांट्स यूज करते हैं, उसके वेस्ट मटेरियल्स हैं, वेस्ट मटेरियल से हम लोग क्या करते हैं, या तो उसको बेकार में हम लोग फेंक देते हैं, चारा बना देते हैं, गाय खा जाती है, लेकिन वो पोल्यूट कहीं ना कहीं हमारे इन्वायरमेंट करती है। जैसे हमने एक राइस स्ट्रॉ का पढ़ा कि वो पोल्यूट करती, पंजाब दिल्ली में काफी उसका जो धुआं था काफी पोल्यूट कर रहा था, तो उसका हमने सोल्यूशन निकाला कि उससे हम एक प्लांट लगाया जिससे हम उससे बायोफ्यूल बना सकते हैं। इन अ सिमिलर वे ऐसे ही हमारे पास कोकोनट हस्क होता है जिसको हम वेस्ट फेंक देते हैं, वो भी, लेकिन उसका भी यूज बायोडीजल बनाने में होता है। ऐसे सनफ्लावर ऑयल का जो वेस्ट ऑयल होता है, सिमिलर वे इस सस्टेनेबल एनर्जी का मतलब यही है कि जो एनर्जी हम लोग यूज करें विदाउट polluting our environment, और हमारी needs complete हो सके।
Thank you very much.
Thank you so much.
Thank you, sir. Next is Mr. Sapresh Devidas, but he is not in the list. So we'll move towards Dr. Suchilitsa Das, who is an associate professor from Department of Basic Science, Modern Institute of Technology and Management, Bhubaneswar. The topic is a study on green initiatives in MSME sector. Ma'am, kindly unmute yourself.
Actually, ma'am, I have not the slides of Dr. Das. So I request to Dr. Nishi Seth, kindly make him co-host so that he can share his slides. I request the name of participant, ma'am.
Dr. Suchilitsa Das.
Suchilitsa Das.
I have unmuted her.
Okay, ma'am.
Hello. Can you hear me?
Yes, ma'am.
Okay. And we don't have your slide, so you'll have to share it from your end. And you are being made focus.
Ma'am, I cannot look the slide. Look at the slide, so no slide is coming there.
Okay. Nishi ma'am, could you please see? Yes, she has made you co-host now, ma'am. Now you can share.
You can share now.
I will share, sir. I will share, ma'am.
Yes.
Hello.
I will share the PPT.
No, you have.
You will share. You'll have to share, ma'am.
Okay.
You'll have to share because you have the PPT.
Okay. Good afternoon, everyone present over here. I am Dr. Suchilipsa Das. I am going to present on the topic a study on green initiatives in MSME sector. Micro, small and medium enterprise MSME sector provides maximum opportunities for both self-employment and jobs. In India, it is projected that MSMEs contribute roughly 70% to the industrial pollution. In order to reduce the industrial pollution and cost, they have to adopt green technologies such as installing solar panels and wastewater treatment. Objective of the study: to study the application and impact of solar energy for electricity and heat production in the MSME sector, wastewater treatment in the MSME sector. Methodology: the paper is descriptive in nature. The data is used from secondary sources, from periodicals, annual reports, referred journals, books, and websites. What is MSME? That is the updated MSME definition. Type of enterprises, such as micro enterprise, investment is up to 1 crore and turnover is up to 5 crore. So like this is the investment for small enterprise and medium enterprise. And these are the different types of enterprises coming under this MSME. Use of renewable energy, that is solar energy. MSME are losing out due to high energy cost they pay for the electric bill by using the electricity, including coal, coke, electricity, diesel, steam, as well as inefficiencies in the system. Adoption of renewable source of energy, such as solar energy, to produce electricity is not harmful to the environment, because when solar electricity is produced from the sunlight falls on the solar panels and electricity is produced. So there is no emission of carbon dioxide gas. But in thermal power plant, when fossil fuel is burned and coal, diesel, kerosene are burned, so carbon dioxide is released and polluting the environment. And also in the MSMEs also, for production of electricity, different types of fossil fuels and oils are also used for production of the motors by the generators, electricity, and that is also producing carbon dioxide, which is polluting the environment and causing global warming and climate change. So this combination of fossil fuels produce greenhouse gases such as carbon dioxide, nitrous oxide, which cause global warming. Because these are the greenhouse gases which can absorb the heat radiated from Earth's surface and capturing it and increasing the temperature of the Earth's surface and the atmosphere. So global warming causes climate change such as the heat wave, hurricanes, cyclones, and heavy rain, thunderstorm, heavy snowfall. So like this, these are the different climate changes. Whereas application of solar panels for electricity production produces no greenhouse gases like carbon dioxide. So here we can see, so this is the graph here. So that is the electrical energy consumption, actual consumption and estimated consumption. So up to that is the year 2020. So that is the increasing trend in the electrical energy consumed. And here the electrical injection consumption estimated from 2020 to 2030. So here in both the cases, so electricity consumption is increasing in the trend. And also here we can see that different types of sources of energy are used for production of the electricity. And this is the coal, which is producing too much of carbon dioxide for 1 unit of electricity production, and then natural gas. And here we can see also that is the solar. And in solar case, so a very small amount of carbon dioxide is released, but that is not during the production of the electricity, that is during the production of the solar panels. Impact of use of renewable energy or solar energy. So solar photovoltaic panels will ensure a constant power supply, because when the electricity is produced from the solar panels, that electricity can be saved in the batteries, and then it is passed to the inverter to convert it into from direct current to alternating current, and then it can be used. Since it is this electricity is produced during the daytime, it can be stored in the battery, and during the night time if required. So since there is no sunlight, that electricity can be used. Then power holidays are followed in some state governments like Telangana. Affect the production of these industries, they can be resolved by the use of the solar energy. Reduces dependence of imported energy, such as the import of the fossil fuels, coal, oils, etc., reduces energy bill, and a great return can be earned by the investment. Helps to slow global warming because these greenhouse gases production decrease by the electricity production by the solar energy, hence climate change due to very less carbon dioxide emission. Wastewater treatment. Industrial waste from the manufacturing processes can be in the form of solids, liquids, or gases and can be toxic or non-toxic in nature. Green technology can also be a part of this production process in MSMEs, such as treatment of wastewater. In the wastewater, one can find a growing number of pollutants, which may be inorganic or organic in nature, which have a destructive impact on the environment. These pollutants can be removed by wastewater treatment. Recycled water produced from the wastewater treatment can be used again for industrial manufacturing processes instead of pumping out again by using the electricity. Challenges of our MSME. MSMEs lack awareness of the environmental impact of their activities and the growing need for green skills. So different types of processing are occurring, and these processing produces different types of waste to the environment, and also they are using the fossil fuels for heating and burning, etc., and production of the electricity also. So that is also polluting the environment. So they lack this awareness of environmental impact of their activities and the growing need for the green skill. MSMEs lack knowledge on clean alternatives and integrated environmental methods and management practices. Lack of qualified personnel having sufficient understanding of new green skills and limited access to finance. So the finance should be provided to all the MSMEs with a required amount, and so they can use the green technologies for the pollution reduction in the environment and the good production in the MSMEs. So the conclusion of adoption of green initiatives such as use of renewable energy solar can reduce the environmental pollution and high energy cost in the MSMEs. Wastewater treatment or recycling in MSMEs should be followed to conserve the water, consume less electricity by pumping out fresh water again, and retain the environment pollution free. So this is all about my this presentation. Thank you.
Thank you, ma'am. Do we have any questions, or we shall move?
No, ma'am. Thank you.
Okay. Thank you. Thank you, ma'am. Our next speaker is Dr. Kumar Gaurav, Assistant Professor from MIT Institute. His topic is wastewater treatment and electricity generation simultaneously through microbial fuel cell. So it's a humble request. Please try to wind up within 4 minutes because we are running out of time and we have a tight schedule. We have next session also. So it's a humble request, sir. Kindly unmute Dr. Kumar Gaurav. Hello.
Slides are visible?
Sir, Kumar sir, kindly unmute yourself.
Now
Yes,
I am
slide is
audible.
visible. Yes, you are. So please try to wind up within 3 to 4 minutes
Okay,
so that we
thank you.
can have all the participants on.
Okay, ma'am. Thank you. Thank you. First of all, I thank you to the organizing committee for that one giving me opportunity to present my work, the wastewater treatment and electricity generations to microbial fuel cell. Okay, next slide, ma'am. Actually, microbial fuel cell, which is the electrochemical device, or you can say the bioreactor. And this microbial fuel cell, which is the renewable source of energy, because in that ones we can use different type of the substrate which are the renewable or sustainable. So in the microbial fuel cell, the chemical energy which is converted into the electrical energy with the addition of the microbe. In the microbial fuel cell, there are the two main chamber, anodic chamber and the cathodic chamber. Anodic chamber and cathodic chamber, which are separated by proton exchange membrane, or this proton exchange membrane may be the Ultrix membrane or the you can say the Sardis systems. Next slide, ma'am. So this is the schematic diagram of microbial fuel cell. Left hand side, which is the anodic chamber, and right hand side, which is the cathodic chamber. In anodic chamber, which is mainly the anaerobic, and the cathodic chamber, which is the aerobic. So microbes which can utilize the substrate, mainly if we take the glucose which is converted into the carbon dioxide produces the electron and the proton. So this electron is transferred to the anode and transferred to the external circuit and produces and it reacts with the proton. And during this oxidation of this glucose, it produces the proton. And this proton is transferred to the proton exchange system, and it may be the proton exchange membrane like the Ultrex, Nafion membrane, or we can use the Salt-based system. So this proton get with the electron, produces the water, and so due to the flow of the electrons, so there is a production of the electricity. Next slide, ma'am. So in this type of microbial fuel cell, we take the microbial culture, the Alcligin faecalis. So we procure this microorganism from National Chemical Laboratory, Pune. And in that ones, the anode, like this is the electrode, which is made up of the graphite, and cathode, which is the copper plate. And before using that ones, there is need to sterile this one. So we have used the absolute alcohol and further irradiation for 30 minutes, so this electrode are the sterile. Next slide, ma'am. So in that ones, we have prepared the salt-based system using the potassium chloride, and we have used the agar. So in that ones, in the enolite means the anodic chamber, we have inoculated the microbes, we have put the wastewater, so that in the process of there is the COD reduction, BOD reduction, and the TDS reductions. So for in the example, in the initial value of the COD, that was the 220 milligram. So during the process of the growth of microbes, COD is reduced with the times, and it comes around the 44. And the output voltage during the COD reduction, that was the 314 millivolt. The same is during the BOD, means biological oxygen demand. Again, the initial BOD was the 160. And the initial voltage, that is called as the output voltage is the 64 volt. And during this time, the BOD is reduced with the times, and BOD becomes 160 to 38. And the output voltage that we get the maximum, that is the 318 millivolt. And we have also measured the TDS of the wastewater, that was the very high, that is the 580, and it becomes the 162. And the output voltage, that is the 320. So this was the reduction of the different type of the wastewater, and this is the reduction, and we get the maximum voltage, that is the 320 millivolts. Next slide, ma'am. So that was my conclusion. So microbial fuel cell can use for the dual purposes, for the electricity production, as well as the wastewater treatment. So during the wastewater treatment, we have the optimized their different conditions, reduction of the COD, BOD, and the TDS. So COD was reduced around the 80%, BOD was reduced 76%, and TDS, which was reduced 22%. So in that ones, this microbial fuel cell, which is the green technology, efficient technology, which can be used for the electricity production as well as the wastewater treatment. Okay, so due to the time limitations, I will compile. Okay, thank you.
Thank you, sir. Thank you. So do we have any question? Or we shall move towards this. So, our next speaker is Ms. Manju Tiwari from Kendriya Vidyalaya, New Camp, and she'll be speaking on Corona and reconstruction. Manju Tiwari, please try to limit your presentation within 3 minutes. Hello.
Yes, ma'am.
Manju, ma'am. Ms. Manju Tiwari.
Thank you, ma'am. First of all, thanks to SS Khanna Degree College that it has made so scientific environment in such short time, and really it is very. It has a very scientific impact on my mindset also for accepting the knowledge, because being in teaching, we are nowadays away from experimentation and all this. So, actually, during this lockdown period, daily my mind was busy in knowing about Corona, knowing about its impacts. And my topic is Corona and economy reconstruction. And what I felt that whatever millions of scientists in this world are doing, experimenting. That is just effort to support mankind, to save them from corona happenings. No, next slide, please. So I put a different view for impact of corona on this globe. We already have such a huge record of tests and reports, recent reports. We already know that now vaccines are coming to be in 3-4 months in market. I think in mid-August will be available for the vaccine in market. But daily some new researches, some new findings we are coming to know. For example, that antibodies which are held in our it is also not existing for a long life. It is having very small life. In 2 or 3 weeks, it dies. So corona is a very unprecedented type of virus. We all know human mind is always curious for surroundings. He is utilizing natural resources. And really corona contributed for us so many lessons for life. And also it has presented so many lessons for this whole globe, for national relations, international relations, family relations. So I had been keenly observing such impacts. In my paper, I am presenting the socio-economic and psychological impact on the society due to COVID. So it is a different type. But because I'm having science knowledge, I'm postgraduate in physics. So I was very happy that I was able to understand whatever papers were presented. And I'm also happy that we are the facilitators for producing scientists, producing warriors. Because for saving our country, we need an army also. For saving the health of our society, we need medical research also. For example, one of my students is a professor in Singapore University in chemical technology. Really, we feel very good that our students are gaining so good positions. And we always try them to be having curious for the environment. And this corona that has turned the whole world, and its impact is continued. And I think it would take many years to become normal, and we have to reach the new normal. Next slide, please. Next slide.
It's my humble request
Next slide,
on the
please.
behalf, ma'am, it's my humble request on the behalf of organizing committee. Kindly conclude your presentation because we have to start our next session.
So you slide them fast. I'll do fast. You change the slides.
Okay, actually, the guest speakers with us, so we can schedule your presentation later technical session. Okay, ma'am.
Okay, whatever you
We will
decide.
schedule in later technical session.
If slides are changed over fast, I'll do fast. I was waiting for the next slide.
Yes.
So objective of this paper is to analyze the government official statements, and it is very good that our government is adjusting well with the situations, and day by day it is issuing the proper orders to tackle with this situation. And we also know that human intelligence is incomparable. In this world, no intelligence can equalize human intelligence. But this human intelligence has become harmful now because of its curiosity, because of to do new researches. And corona has unprecedentedly come. And we also know that corona is a new virus which jumped directly to human, to to make it host rather than animals. Animals are free from corona, but animals are serving as carriers for corona, but they are not affected by corona. So really it is very good that all the scientists adjusted well in finding out vaccines in such short duration, and also whether it is at national level or international level or social level, social services are also doing well for the humanity purpose. And education system, from this seminar only I come to know many new MOU which is taken up by Government of India.
Thank you, Dr.
Thank you.
Manju, ma'am. Yes. Due to the paucity of the time, rest of the presentation will be scheduled on 38th, and we will be informed regarding this. And now I would like to request Dr. Neerja Kapoor, ma'am, kindly summarize the session and give your feedback and suggestion to participants. Over to you, ma'am. Neerja, ma'am.
Okay. Good afternoon, everybody. First of all, I would like to give my sincere thanks to Dr. Anuradha Singh and Dr. Jyoti Baijal, conveners of this international webinar, as well as Dr. Sikpi Singh, to give me opportunity to be a resource person of this session. Secondly, I would like to congratulate Principal of the college, Professor Lalima Singh, and whole organizing committee for organizing such a wonderful webinar. As far as presentation is concerned, I will not comment on presentation separately. I will not comment, but on each paper separately, but I will put some views and general comments on the paper presentations. There were so many presentations, very nice presentations of different varieties. Several plant products have also taken. Different disease has also taken, such as cancer and HIV also taken into consideration. During this lockdown period, 19, some papers are also there for presentations. Some presentations are on water quality of Ganga and Yamuna, aerosol pollutions in Delhi and in Lucknow, phytochemical studies in soybean, affordable and clean energy, in which 7th goal, sustainable goal, has been mentioned. Green initiatives has been taken. Corona and economy reconstruction has also taken. There are so many presentations, and it is very difficult to judge which one is better or which one is best, but we will try our best to judge these presentations. And congratulations to all the participants and all the organizing team once again for such nice presentations and for organizing such a wonderful webinar. Thank you.
Thank you, ma'am, for give your valuable inputs to this session. Next, I invite Dr. Justin, sir. Please share your views with participant and conclude the session.
So first of all, I'd like to thank the organizing committee and especially Dr. Anuradha Singh, Dr. Sikpi Singh, and the principal of SS Khanna Girls Degree College for organizing this webinar. And these moments of interaction are very precious because these are times when we can meet each other, share our thoughts and views with each other, learn from each other. And I want to tell you that you all have organized this webinar in a very strategic manner and very well organized, very well planted everything. And all the paperwork that you've been doing is excellent. All the communication that you've been doing is excellent. So there's something that all of us have learned out of not only this session, but even through the entire process of organizing this webinar. So I want to congratulate the entire organizing team. And I also want to thank and congratulate all the paper presenters who have presented our work. Face-to-face presentation can never be replaced with an online presentation mode. But nevertheless, it is a very beautiful moment. It's a very learning moment for all of us. And I want to congratulate all the speakers who have presented your papers. And both ma'am Nirja and myself, we are elated and we are very grateful, and we hope for the very best for all your future endeavors. So thank you very much once again.
Thank you, sir. We appreciate and acknowledge to both of our chairperson our deep sense of gratitude towards your contribution to this session. Now, I would like to invite Dr. Rahman to propose formal vote of thanks. Dr. Rahman.
Yeah, thank you, ma'am.
Please mute yourself. Yes, sir.
Yes, thank you very much, ma'am. A very good evening to all present in this technical session. I feel great honor to propose vote of thanks on behalf of SS Khanna Girls Degree College family and my own behalf. Extended a very heartily vote of thanks to one and all present in this session. I wish to express my gratitude to Dr. Nirja Kapoor Madam, Head Department of Zoology, CMP Degree College, Allahabad, for chair this session. I am delighted by your presence, Madam. I would also like to express my sincere thanks to Dr. Justin Mashi, Associate Professor, Department of Chemistry, Irving Christian College, Prayagraj, for accepting request to chair the session. Thank you, sir. I would like to express my sincere thanks to all the participants in this technical session for present your important work and sharing your findings and opinions with us. We hope each one of you will be returned with a lots of pleasure in mind. I am especially grateful to my international participant Abdul Ghani Milani, who has participated in this session and shown their interest for our seminar. At the end, I like to thank my co-session coordinator, Dr. Anuradha Singh Madam and Dr. Spi Singh to have been with us. And I request to all kindly mute yourself and on the video for a group photographs. After that, we will move to the next technical session. Thank you.
Thank you so much.
Thank you, Nirja ma'am. Thank you, Justin sir, once
Thank you,
again.
ma'am. Thank you, everybody. Thank you.
And
Thank you. Okay.
Now I hand over to Dr. Jyoti Baijal and Ms. Subodhika Sharma to our next technical session.
तो लीव कर देते हैं. हाँ, नेक्स्ट टाइम का. अभी तो हम लीव कर देते हैं.
Okay. Jyoti ma'am, kindly unmute yourself. Yes. Now the guest with us.
Good evening, all the guests and the participants. Myself, Dr. Jyoti Baijal, along with Ms. Subodhika Sharma and Dr. Nishi Seth, extend you warm greetings and welcome you all in this third technical session. We now over to you, Ms. Subodhika, to accord a formal welcome to our chairperson and Mr. Thomas P. Valenti. Subodhika.
A very good evening to distinguished dignitaries, respected faculties, and all those who have joined us. I welcome you all to session 3 of day 4 of the 5-day International E-Conference on Sustainability Challenges and Transforming Opportunities amidst COVID-19. For this session, under the theme of legal sustainability, we have with us Professor Shakil Samdani as the chairperson. Professor Shakil Samdani is currently the Dean of Faculty of Law, Aligarh Muslim University, Aligarh. He is also the coordinator of Aligarh Muslim University Centers, member of Academic Council AMU, and member of AMU Court. Professor Samdani has a teaching experience of about 30 years with specialization in family laws, public international law, human rights law, and Islamic jurisprudence. Professor Samdani has appeared for interviews on various TV channels like Islam TV Europe, NTV Europe, BBC World, Aaj Tak, Star IBN 7, NDTV, Zee News, Zee Salam, ETV, Sahara, ANI, India TV, Republic TV, etc. Professor Samdani has also been the examiner expert paper setter for organizations such as the Union Public Service Commission, UP Public Service Commission, Rajasthan Public Commission, Jharkhand Public Commission, and educational institutes like the Ambedkar University, Agra, Kashmir University, Srinagar, Rohilkhand University, Jamia Millia Islamia, New Delhi, Invertis University, Bareilly, Central University of Kashmir, Indore University, etc. Professor Samdani was appointed the Estate Officer of AMU from 2015 to 2018 by the Government of India. He is the coordinator of Sir Syed's Bicentenary Celebrations AMU since January 2017. Professor Samdani has won several awards such as the Legal Excellence Award by Pathshala Classes Aligarh 2019, the Legal Expert Award 2018, the Lifetime Golden Achievement Award 2020 by Social and Legal Research Foundation Varanasi, Allama Iqbal Award 2019, Award in the Field of Law and Social Work 2020, Award of Excellence by Islamia Group of College Deoband UP, and Abru-e-Urdu Award by National Council for Promotion of Urdu Language 2018. Sir, we are truly honored to have you amidst us. I now invite Professor Samdani to introduce the speaker for this session. Thank you.
The speaker. Madam.
Yes, sir.
I have to introduce the speaker.
Yes, sir. Yes, sir.
You never told me. I have to preside it.
Yes.
Oh. Okay. I'm
so. Don't worry.
Okay. The speaker is good afternoon, Professor Lalima Singh, our distinguished guest, Tom Valenti of Chicago, dear participants, ladies and gentlemen. Tom Valenti is a Chicago-based conflict resolution specialist offering mediation, arbitration, and facilitation services and training globally. A certified mediator, Tom has conducted numerous mediations involving civil, commercial, interpersonal, and workplace matters. He has mediated and trained extensively both nationally and internationally in jurisdictions all over the world. Tom is a member of several bar associations, including the American, Chicago, Illinois, and Indian Bar, and is co-founder and former board member of Mediation Beyond Borders. Sir Tom Valenti has a passion for mediation, highlighted by many qualifications. Tom, sir, holds in the field. Tom is a dedicated and talented mediator, AA, AAA, and ABNA neutral. He is also a neutral arbitrator and mediator for the National Arbitration Forum and Center for Resolution. A member of Chartered Institute of Arbitrators, Tom Valenti is also an arbitrator on public panel at FINRA, and is an approved arbitrator and mediator for several governmental and regulatory bodies, including the Circuit Court of Cook County, the National Arbitration Forum, and is the advisory board of the Association of Mediation Assessors, Trainers, and Instructors. Extensively trained himself in all aspects of dispute resolution, Tom's accreditations include conflict transformation skills, training, focusing on systematic dynamics, ABA International Family Mediation Training, including Hague Convention child abduction cases, Circuit, Cook County Annex Arbitration, and mediation training programs. Tom also holds a mediation certification at the Institute for Conflict Management and is a board member and trainer at the International Academy of Dispute Resolution. Sir Tom's interests include the Athens Migration Dialogue Project, of which he is the co-leader. The project helps local communities to design, organize, and conduct dialogues that address difficult and dangerous issues such as migration and violence in school. He also led the Hurricane Katrina project in New Orleans, LA, and Biloxi, MS, which involved training area NGOs to help them improve their skills and develop community mediation programs. Having a keen interest in the Middle East and cultivation of mediation system across the world, Tom also worked with the Middle East Peace Initiative. This is very important because peace is very important for everybody, for every country of the nation, for every community. Collaborating with the Pluralistic Spirituality Center project, he oversaw the development of an Israeli Arab and Jewish co-mediation approach and service to mediate community dialogue in an Israeli Jewish and Palestinian peace village, Neve Shalom Wahat al-Salam, NSWAS. Finally, training others to mediate is central to Tom's belief that alternative dispute resolution is a wonderful force for good. Working with the International Academy of Dispute Resolution, INADR, a charitable organization created to develop an understanding of the benefits of mediation among law students. Tom has traveled to the UK, Dubai, India, and Europe to deliver mediation training for students. He is also an International Mediation Institute's mentor to youth. Sir, I, on behalf of the SS Khanna Degree College, Prayagraj, formerly it was known as Allahabad, I welcome you, sir, and I invite you for your talk. And I am very happy, I am very satisfied that you have involved yourself in mediation, conciliation, reconciliation, and you are trying to achieve peace through your intelligence, through your abilities, through your words. And I once again welcome you on behalf of SS Khanna Girls Degree College, Prayagraj.
Thank you. Shall I proceed?
Yeah. You can
Okay.
proceed, sir. You can proceed.
Well, thank you to the college, the organizing committee, and everyone for asking me to participate in this very interesting series of workshops that you have all participated in this week. I miss my time in India. I usually get to travel there a few times. I've never been at the college, but if we get past this pandemic, it would be nice to be with all of you in person someday. And so in this era, what I thought would be interesting for us to talk about, and for me, rather than you listening to me talk all day, I would prefer that we make this interactive, that maybe Subodhika can help me in the chat, and I'm happy to have people pose questions during the session, and so that we make this more of an interactive discussion than just a lecture. I find I can keep a little bit of an eye on the chat and watch it myself, but if one of the organizing people from the session can help do that, I think we'll find this to be a much more interesting session for everyone today. And so feel free to use the chat to pose some questions at any time. And I'll interrupt my session to do that. We have a lot to cover today. We won't probably cover it all because I want to make sure that you have time to ask me questions. So what are our goals for today? What I wanted to accomplish for all of you today was to introduce the subject of ADR, or alternative dispute resolution. What's the impact of COVID-19 on it? How do we all fit into the picture? And I can go on with some specific examples of its use and then address any concerns any of you may have relative to that. All of you come from different backgrounds. You could hear from the gracious introduction by Professor Samdani, and thank you for that, sir. I have a broad experience in many different areas, and because of that, I'm able to look at this COVID-19 and see how is it going to impact disputes. No matter what our job is or where we are in life, we all have disputes. If you have a brother, sister, mom, or dad, you know, or a child, a son, a daughter, grandma, whatever, we all know that life is full of disputes. As a faculty member, as an employee, as an administrator, we deal with conflict every day. This particular pandemic has made probably conflicts more prevalent in all of our lives, because it's hard to live in this atmosphere. It's hard to work in this atmosphere. It's very difficult to even relate to other people when we're locked down, and the restrictions that are put on us maybe even change from day to day, if not hour to hour. So let's first talk a little bit about what is ADR. Usually we think of, if we have a conflict, we think of court, right? That's where we usually go. We find that the courthouse is often the first choice for people to resolve a dispute. Those of us who practice in this area of alternative dispute resolution have looked at other ways without litigation to use other techniques to solve conflicts. And those conflicts can be person to person, it can be employer-employee, it can be business to business, it can be neighbor to neighbor. And when we think about all of these, we can think of it in our local community. You can think of it as on your campus, or you can think of it between you and me. I'm sitting here in the USA and you're in India, and perhaps we each have a business, and we can have a dispute that's cross-border. And so there's lots of disputes that go on and are particularly amenable to alternative ways to do that. So what are the alternative forms of dispute resolution? These are just a list I prepared, and I think most people who do what I do would accept this as the list of common forms besides litigation: negotiation, facilitation, conciliation, mediation, arbitration, and then something I have an interest in, which you heard about a little bit already, which is community disputes. Let's take a quick look at each one of these. The negotiation is probably the easiest one for anybody to do and understand, because it's just you and I maybe have a dispute, or maybe I'm just buying something from you or you from me, and we negotiate. We may have a conflict because I think my old car is worth lots of money and you think it's not worth so much, and I'm trying to sell it to you. So we have a bit of a conflict about price or condition, and we have a conversation or a negotiation on reaching a solution for the transfer or sale of a car. So negotiations happen all the time. Some of us negotiate with our children about what time they should be home in the evening, what clothing they should wear, what schools they should go to, and things like that. So negotiation is part of all of our life. Facilitation is a way that someone like you or I, it's very informal. A neutral third party works with both sides to reach a solution, some kind of resolution of their dispute. We always assume that the people in a facilitation want to resolve something, or want to maybe even accomplish an understanding. When we were doing work with the refugee issues in Greece some time ago, we weren't there to solve the problems that nationals have with refugees. We were there to help them understand the viewpoint of each one of them. As a facilitator, we're not there to propose a solution or an answer to the problem. We're there to help people have a conversation or a dialogue about the things that may be perceived to be differences between people. We see this perhaps even now in COVID, we see lots of different opportunities for facilitation to be used to help people understand what's going on in society. What are the issues with regard to face mask wearing? In my country, we have people who are very much against face masks, even though it's a public health issue. And we can use facilitation as a way to have the medical profession educate people on both sides of mask wearing or sanitation or distancing, or even now, it's a big issue now here, and I think in India as well. Do we go back to school in person? or do we go online? And there are people who have very strong feelings about each of those issues, and people like you or I can be hired or even volunteer our time to manage that conversation, much like the facilitators are doing who are managing your sessions during this week's conference. Mediation is a bit more formal. An impartial mediator helps the people reach the solution. In a mediation, the goal is always a settlement. Facilitation, it's understanding. We move a little bit more advanced down the scale, and we see that the mediator is designed to help reach the solution. It might be the price of the vehicle. It might be new terms of an employment contract. It could be what the result is of a breach of contract. It could be a dispute between an investor organization and the government on funding a treaty. Anything like that is subject to a dispute and available for an alternative way to resolve that dispute. Conciliation is something that's looked at quite differently in different countries. In India, you have the 1996 Arbitration and Conciliation Act. We won't spend much time talking about this. It's really a way that the act used the court system to have judges mediate or help resolve a dispute that allowed that dispute to become a court judgment. The role of the conciliator is defined in the act, and it has lots of authority and parameter to help resolve that dispute. Arbitration is probably the most formal. In many places, it's called rent a judge. And so what do we as arbitrators do? We are hired by the parties in a dispute who have usually placed in a contract a dispute resolution clause that says, if you and I have a dispute, we're going to use an arbitrator or a panel of arbitrators to resolve it. We're not going to go to court. Why does that happen? It happened because we thought, or people who use arbitration thought that our arbitration panel is going to be faster, cheaper, less expensive, and a less formal way to resolve a dispute. In addition, if there's a dispute cross borders, no one wants to be in the home court system of the other party. You don't want to be, if you're an India company that's doing business with a USA company, you don't want to be in the US courts because you think you may have some kind of bias against you, and the US people don't want to be in the Indian courts because they think there may be a bias against the US company. So they use an arbitration system where perhaps the US party picks an arbitrator, the Indian party picks an arbitrator, usually from their home countries, and then there's a neutral or third party appointed, usually from some country, not the home country of either of the parties. It became a very efficient system. There's a lot of criticisms of it now because it's not so fast and cheap as it was designed to be when it was first instituted. The last is community disputes, and we see different ways of doing this. It can be some fight about a pet. Your dog is barking in the middle of the night and I can't sleep. It can be about an issue between two communities, maybe two different religious communities in a city or a neighborhood. And it can be about very public issues, as you see in the bottom picture on this slide, where there are different views of governmental acts that create conflict between large groups of communities, often protests or very loud and forceful actions by community members. Those disputes are not for everyone to participate in facilitating. And many times the government itself doesn't see the opportunity to use community dispute resolutions to help the government manage policies, or perhaps even formulate policies. So let's look at COVID-19, unless there's any questions at the moment. And if somebody wants to look at the chat box, or you can raise your hand or something, and let's see if there's any questions. If not, let's see how COVID-19 plays into disputes generally, and how we can, looking at all of our obligation to create a sustainable future, how we might be able to look at our disputes in a different way and maybe proactively become a dispute prevention person, where we're looking at the opportunities where disputes can occur, and we intervene early so we don't have litigation or arbitration or community problems. So COVID-19, we can all agree, huge game changer, right? We saw at the beginning of it, and sometimes the beginning is starting again. Even in the city I'm in, we had a return to some of the more stringent lockdown measures, because when some of the bans were released slightly, we saw another uptick in the number of positive cases, and so we're going backwards here. Even last night, I think they added, effective tomorrow, or no, I think maybe Friday, we added some more travel restrictions here that people in my state, I live in the city of Chicago, the state of Illinois, I think we're up to over 20 states where we're asking people to self-quarantine for 14 days if you've been in one of the neighboring states that have very, very high levels of positive cases. So travel bans and restrictions go on a cycle. They go up and down. We saw at the beginning, you didn't have access to justice. The courts were shuttered, maybe significantly scaled back. We're coming back online with some of them now under very restrict measures. Public projects, construction projects in some jurisdictions were stalled or stopped completely. Some allowed to return in small ways. Supply chain, the distribution of goods and products were severely interrupted. So if you or a member of your family, or perhaps if you're an attorney, a client, or maybe you work in one of these businesses that all of a sudden you can't get the goods shipped to your supplier, or from your supplier to you and you to your customers, there's some breach of contract. There's a legal right that somebody has that's been breached by the result of a travel ban or travel delays. And so I can't get my goods here to resell. I owe them to somebody, and so somebody is trying to sue me in Chicago because I can't get the goods that I ordered from India. I was in our Indian neighborhood just a few days ago, and one of the gentlemen in the shop, and they're doing lots of trade now because of the Eid holiday coming up, and he said, I can't get my goods from India because of travel bans. I don't have enough inventory to sell. And so this impacts all of us. The supply chain, construction projects, repairs to our homes, our businesses, our schools, all of this is impacted by travel bans. So it's kind of a perfect storm. It was a storm in March, April, and it seems to be coming back now, where everyone, and if you're the owner of a business, you have employees who are impacted by this too. People who have been put out of work, who may need desperately income to have their families a place to live, food on the table, and all of that. So it seems to be something that doesn't discriminate against kinds or types of people. It hits companies as big as Tata, Apple, Microsoft, and people as small as I, who just a person trying to work in the field that I'm in. And so this virus doesn't discriminate. It doesn't care who you are. It finds a way to impact you in some way. So these are the challenges for what we are now calling dispute management. How do we look at problems, and how do we try to react to them even before they occur? So what we've been advising people in business or in law, or now very important as educational institutions in the US are thinking about reconvening, get ahead of the dispute. Try to develop protocols in advance so that if and when you can reopen your business, your school, your community center, that you have valid protocols in place that comply with local governmental restrictions, and that you've thought about the impact on all the stakeholders in your community. And that may be the customers that come to your store, the employees who work there, the people who come and deliver things to you from other businesses, and the people who you're asked to deliver to as well. So we try to recommend to people that they engage with their contracting parties, set up some kind of a regular meeting with the people you do business with, perhaps your employees who were sent home. Don't just say, sorry, you have to go home, and don't talk to them. Don't tell your faculty members, sorry, we're not going to have you participate in a conversation about how we can reconvene our educational process. Right here in Chicago, for example, we have announced that the schools are going to be back, the public schools in the city, and these are high schools, not the universities, are going to be back in session this year. There's a strong indication from the teachers, no, we're not going to come. I'm not sure that they started that dialogue process early enough to talk with the leadership of the teachers union. We have here a very strong union that speaks for thousands and thousands of teachers who teach in our public school systems. They don't want to be back in school. Here, we're about to look at, in about a month, the first day of school, and probably it's not going to happen. Those conversations should have started a few months ago. The other thing we recommend people is that if you're in a business that has contracts that are being disrupted, look at those contracts, see what the dispute resolution mechanism is, perhaps begin a negotiation early on in advance of someone trying to file a litigation against you, your company, or perhaps that's going to come from a supplier, perhaps it's going to come from an employee. So we look at, if we're in the legal business, we look at the contract clauses that talk about these kinds of issues. We look and see if there's any insurance to manage those kind of disputes. We look at the impact a contract may have on our finance agreements. Perhaps the bank is looking for money and we can't pay them. And we begin to audit each contract to see what are my opportunities to resolve that. If there's no dispute resolution clause, we perhaps can modify that contract to say, let's do a mediation or let's do an arbitration. We have a dispute. Let's not go into court. Court's going to take too long, and we have imminent problems to resolve in our businesses. We can't turn our problem over to a court system that's not functioning at all. So establish early communication. Consider how you're going to develop continuing relationships with the parties that you're in dispute with. And what do we look for in the area of alternative dispute resolution? We always try to identify or find common problems or common issues between the parties in dispute. If there's one thing that COVID-19 does, it creates a common issue between everybody because it impacts everybody. So we can begin our conversations with the people who we are in dispute with, or with whom we anticipate a dispute. We can begin the conversation by saying, we've been impacted, every one of us has been impacted by COVID-19. What can we do together to fix this problem that's affecting our business relationship, our employment relationship, or perhaps our international relations? So we have a common ground at the beginning. And we should, in the field of ADR, alternative dispute resolution, we should be using that common ground as the starting point for conversation. And perhaps send a letter saying, how are we going to manage this together? We'll look for room to negotiate the clauses that are in there that may, some international clauses, contracts have clauses that say if there's some major event that disrupts the ability of the contract to be performed, we can terminate that contract. Many people need contracts terminated as a result of COVID-19. I think you're probably finding the same thing I'm seeing here at home. We have a huge number of businesses closing. And these can be very small businesses. We call mom and pop, which are just the family businesses. You know, perhaps a little shop that's selling goods, or perhaps it's your local place where you get your coffee and a beverage or something like that. And sometimes it's companies as big as international chains that are finding it very difficult to stay in business. And so we look at how we might renegotiate staying open, or how we may renegotiate our closing. And here we have some laws, I think you do as well, that allow for a company to file bankruptcy or get some relief from the government that allows for the non-payment of pending obligations. And so one thing that we learned, and we're doing it today, is that online tools have become the methodology for engaging with other people. Something like the Zoom that we're using now has become used in all of the mechanisms that I talked about earlier: negotiation, mediation, arbitration. Even our court system here are using Zoom calls to allow parties with the judge to have a hearing. And so we encourage people to use online tools to educate themselves in the use of that, and educate all of those people in their business community or stakeholder community to get up to speed. See if you can bring in the technology that you need and your business needs to use online mechanisms, because sitting back and waiting for COVID-19 to end is not effective dispute management. Effective dispute management is addressing the current issues currently, not delaying them, because frankly, nobody I talk to knows when this is ever going to end. We have a view that it certainly is not going to be in the year of 2020. Whether we're going to have some return to the normality that we might have had in January of this year, highly unlikely. So we have to bring our technology up to speed. We have to teach those who we rely upon to manage our businesses and run our operations to use technology. Financially, we have to make a commitment to bring technology into our businesses, our educational institutions to do this. It wasn't too long ago, I was asked to participate in something in India, and the government had shut down the use of Zoom, because you remember early on, Zoom had Zoom bombing, and there were some kids that were invading the Zoom sessions and posting, things that shouldn't be posted in an educational setting. And so the government of India decided that you can't use Zoom for legal related things. And the technology changed a bit. There's a lot of protection measures that were put in place. The fear was real at the beginning until you learned how to use tools such as this to safely manage a conversation or an arbitration or a mediation. And so we want to get away from this. I'm going to just talk for a few more minutes, and then I see some questions, and we'll open up for questions now. But let's think about not having, about not having this thing. And I'm a lawyer by practice, so it may be strange for all of you to hear a lawyer say, let's not talk about the relentless pursuit of litigation. That's how we all pay our bills. That's how we all helped our families get through school and things like that. We use the court system quite effectively. But what we do now as dispute resolution professionals is we talk about dispute management. Let's talk about, instead of litigation, let's talk about options, renegotiation, using mediation, using some form of mediation and arbitration, using other mechanisms that are available to help the parties who are in a conflict in a very dangerous and difficult time design a system to resolve their problem that meets their needs and interests. The court system probably doesn't have the ability to do that, especially when it's not operating at full speed and full capacity. And so let's go to some questions. And so Bodhika, if you have some, if you want to unmute people and have them speak the question, I'm happy. Are you managing this session, I think? I don't mean to give you a job that you don't have.
We have some questions in chat box, sir.
Okay.
We have some questions in chat box.
All right. And so if anybody else wants to speak a question, they can let you know, and then having a conversation works really well for me. I'm going to look back at the chat, and I saw someone say, what is the mechanism I prefer the most? And so for me, mediation is the thing that I prefer the most. And you may as well if I talk about it just a little bit. And so imagine a dispute. I had one here some years ago, and there were two cousins, and they decided to be in the falafel business, right? They claimed they had the best falafel in the world, and they were going to start a series of falafel stores. And of course, as most plans are, the plans don't work out, right? Maybe it wasn't the best falafel, but they had 2 or 3 stores, and cousin A was running one and cousin B was running the other, and it wasn't successful. And I really think cousin A's family said, you got to get out of this because you're not bringing any salary home. And they had a big dispute about how do I get out of this business. And they came to a mediation. There was a litigation pending. They decided to resolve it by mediation, and they didn't speak to each other. They were so angry at each other, they wouldn't even sit in the same room. And as a mediator, that was an opportunity for me to try to help them solve a business dispute, but also a family dispute. We all have families, and we know how difficult it is at times when some business relationship interferes with the operation of the family. The cousins won't both come to the same family parties together anymore because of this business dispute. So in mediation, unlike court or arbitration, we have the opportunity to talk to each of the cousins and perhaps restore the relationship they had with each other, to see if not only we can fix the business problem, but fix the family problem as well. And so that's a good example of why I favor mediation as just for my personal outlook, because I like the idea of restoring relationships. That's kind of what drove me to working in refugee communities. We've done that. I've actually been in Lebanon working in a Palestinian refugee camp there, trying to integrate some of their values into the Lebanese society. We saw that in Greece with the refugee community. There's different issues that we can work on that we try to get people to understand. Perhaps these people of this other religion aren't so bad. Why don't you talk to a few of them? All you've ever done in your life is call them names based upon assumptions. Maybe if you hear their story, maybe you'll understand that they're really like you. They just believe in a different set of religious principles. And so that's my answer to the question about that. So question about digital modes of documentation, really good question. In the legal field, we really worry about how do we transfer documents, cyber issues about hacking, right? All of these things that we can't afford, especially in my world, the legal world. If I'm arbitrating a case and people are sending me documents that are sensitive, confidential, I have to make really sure that my systems are safe, that no one can hack and get access to those confidential documents. Perhaps it's the draft of a decision that I'm writing, and maybe that's of a company that's traded in the stock market. And if somebody sees that, maybe they can buy or sell the stock because they have advance notice of that. So we rely upon multiple layers of security. In most arbitral institutions, we don't exchange documents by mail. They're done through a highly secured portal. You heard in the introduction, FINRA, which is a securities agency. We resolve securities disputes, highly sensitive documents, and we don't email documents to anybody. We only access them through a secured portal that the litigants deposit them in, and the arbitrators get access that way. And there's other security policies that we're designed to make sure that the parties in digital dispute resolution or remote dispute resolution are protected as well.
So we have a participant. You can ask a question, Mr. Gaurav. You may ask a question.
Hello, good evening, sir.
Hello.
This is Gaurav Gulati from Allahabad.
How are you?
I'm fine, sir. How are you?
Good, good.
Sir, I have a question. If a public justice system was technology as a service platform, it would hold potential to greatly improve the access to justice for many people, but it may also yield opposite results. Some people are either unwillingly or unable to interact through technology. Also, we have to take into consideration the digital divide. So, sir, what is your give on this question? How do we make the system more inclusive?
I like that question because I've always looked at online technology as an access to justice tool, that we were enabling people who didn't have perhaps financial access to justice. We were creating a better mechanism for them to resolve a dispute because they didn't want to pay a lawyer. Well, you don't need a lawyer to be in a mediation. You can do it yourself. So we thought that, but we didn't pay as much attention to, do they have the tools? And so we can provide access, but if they don't have a computer or a phone now, we can even use our phones for Zoom and all that. So we have to pay more attention to making sure, especially now when court systems are going digital, we have to pay close attention in designing dispute resolution mechanisms, designing them to take care of the people who normally would walk into court and see the judge and are now told you have to log in on your computer or your phone or something like that. And so this is part of what design means today in the COVID-19 era, is working with technology experts and judicial administration experts to figure out how we create that. And perhaps we do that by having we have a very robust library system in Chicago, for example, is creating in the libraries free computer centers where litigants can use access to those computers for free to log into their cases. Perhaps there are other community centers. Many times we can integrate with religious communities because many of us use religious communities as a daily, weekly, whatever meeting place, and perhaps we can have those centers be points of access for digital access. But Mr. Gulati, we also have to not just think about making the space available, but we have to train those people. One of the slides I didn't get to was we can get ourselves all ready to be online to digitally resolve our dispute. But if our clients don't know how to do it, we failed them. So we have to, as lawyers or professionals, take time to train the clients. So making the space available in a library or mosque, a church or temple, whatever, is fine. But unless there's someone there to train the people how to do that, we failed them. And so we have to think really globally. And I think the pandemic has taught us that our job now is to think bigger than we ever thought. That we can't just think about my world, but we have to think about everybody's world. And we learned about that in making health systems accessible, whether it's vaccines. We have a huge problem coming up with a vaccine. The wealthy countries, are they going to be the ones who can buy the vaccine at the best price? And are they going to be the ones that get it? And the developing countries don't get the vaccine. So as as thinkers, we have to be thinking about the impact of all of this. And I think as bad as this COVID-19 is, it should make us, all of us, no matter who you are in our session today, it should make all of us realize we are all in this together. And COVID-19 made us realize that there can be something that binds, that's common to all of us. But let's not just think about COVID-19 as the thing that binds us, but realize there's people with other issues in life, whether that's community disputes, religious disputes, and think of how we can be of service to each other in whatever our role is, making sure our educational system is accessible to people who can't get there, making sure our teachers are properly trained in delivering online lectures and courses, that we have to teach differently. We have to make it more engaging. We can't just talk for 1 hour and expect students to learn as much as if I was in the classroom. When I can see your eyes, I can see that you're paying attention or not. And so we know about this digital divide. You can guess that I'm on the other side of it. From most of you whose pictures I can see online. I've made it a point, and I'm not really good at it. I practice more at it than most people because I've been training people for 5 years on how to use Zoom in mediation. And so I think I'm better at it than a couple people that I know, but I'm certainly not as good at it as most of you on the call. But what do? I use you to teach me. And so we got to reach out to the technology people. We can't think we're the smartest guy in the room all the time. I just trained in April and May about 20 people who do what I do, senior level people who were told you can't work in an office anymore. And in mediation, we love being in an office. I shake your hand, give you a cookie, here's a tea, and that's how we work. And these guys were shocked. And basically they were told, sorry, you're too old, you're out of business. So we did a training, a Zoom training. How are you going to use Zoom to be a mediator? And I'm telling you, and I won't mention any names, high people, Ivy League, all kinds of education. I'm teaching this one guy Zoom. And I said, well, it should be on your desktop. How to share a screen, it should be on your desktop. And it's kind of funny, but he said, Tom, where's my desktop? And so I'm realizing that there are people who are brilliant, but the digital divide is no better explained than in that example. I can't find my desktop. And so we found his desktop for him, and we got him through the session. But luckily, we then had about 15 or 20 men and women who thought they were out of business, who are now getting quite good at using Zoom in mediations. And so we can all help each other. And that's why I enjoyed and I appreciated the invitation from the college to come and be with you today. I much prefer doing it in person because we have more fun doing it that way. But we try to make it as close to in person as we can. And each time we have a digital interaction, we should be better at it than the last time we are. Because I'm still learning from you. And I'm still learning from all the other people that I get to interact with. And I look forward to doing that. But this technology thing, we have to make sure it's available to everyone. You can't have a justice system that works only for some people. Any other questions?
So we have one more question in the chat box.
Yes. Timely justice, this one, yeah. So the question is, one of the objectives of ADR is timely justice. Most ADR mechanisms are not binding upon the parties, and many times parties prefer to move to court. Really good question. So I'll tell you something funny. When I first came to India in, I think it was almost 10 years ago, we were doing a session in Bhopal on introduction to mediation and running a competition there for students. And one of the young guys said, but sir, why would I take a mediator when I can have a judge? And so I learned, and I don't think it's ever changed much in India over the many years I've been coming there. I think we still have this bias about mediation that it's not the same as court. But I'll share two things, is in terms of timeliness, if two of you had a dispute and you wanted to mediate the dispute, we can do that. I don't know how much more time I have, but we could do it in 20 minutes, let's say, and using an online mechanism. And so the court system is never going to be as efficient to get two people and a mediator together as this voluntary process is. About the usefulness of a mediated settlement. So when we settle a case, it's not a court judgment. And that's always been the bias in India, is people are saying, well, I don't have a piece of paper to go into court and say you owe me money. And the answer to that question is this. When two people voluntarily come to a mediation to settle a dispute, nobody is telling them pay this amount of money. They decide on their own. So the satisfaction rate, or let's say the compliance rate in mediation, how many times do people not comply with the agreement they just made? Less than 1%, because the mediator doesn't tell them the solution, they create their own solution. So they've decided what they're going to do. And so we find the compliance rate much higher than court, because you know the first court judgment, you're going to appeal it. And so we find it very effective. Just this, no, I'm sorry, it was August of last year. I was there in Singapore for international commercial disputes. This issue was so important that people weren't mediating international disputes. So the United Nations Convention on Mediation Settlement Agreements was signed in Singapore. I was lucky enough to be at the signing ceremony there, and it will come into effect in September of this year. There's still the process of people, countries signing on. And so we can take an international mediation settlement agreement and make that an enforceable judgment in any of the signing countries in the world. and so that tells you, I forget who asked the question, Abdullah, that tells you how important your question is even on an international level, it's a concern. And the dispute resolution community is addressing those concerns and trying to provide mechanisms for making mediation more effective and meeting the needs of the parties. I can't answer the question about the exam for class 6.
So please,
you want me to answer that?
No, sir. No, sir.
No, I can give the best answer. No exams any longer. Everybody passes with the highest marks. Is that a good answer? So any other questions or a comment? We have a few minutes left, or Subodhika, tell me what how our timing is.
So
what
we
skills
have
to be an arbitrator? Yeah, let me take this, at least this one more question. So much different than the skills to be a facilitator or a mediator. And so most arbitrators come to arbitration with some background or experience, right? Many times it's a legal background or experience, because oftentimes there's legal issues in an arbitration, but you don't have to be a lawyer. We find that in public projects, construction disputes, many times arbitrators who are asked to be involved in that are engineers or perhaps worked in the construction field. And so in disputes between pharmaceutical companies, sometimes biological or chemical engineers are involved in those because they have background in pharma or some kind of healthcare technology. So the skills can be quite broad, but with regard to actually hearing a case and writing decisions, there are very technical skills that need to be addressed, where arbitrators learn how you take evidence, how you rule on evidentiary matters, how you deal with confidentiality with documents submitted to you, and then actually how you write the award. When we finish a case, we write an award, and how that's prepared has very strict requirements depending upon the jurisdiction you're in and how you address the legal and evidentiary issues that were raised to the tribunal. Many times, a tribunal is usually a panel of 3. Many times they want a lawyer on that, because the lawyer is very effective in writing the awards and making those legal decisions that happen all the way along the way. And so there's lots of training for arbitrators. Most of the law schools in India have some kind of arbitration. There are ADR courses in arbitration, but that's teaching you how to litigate, how to be an attorney in an arbitration. Different skill set than sitting as an arbitrator where you're really a judge. And so the country by country or institution by institution, I'm a FINRA arbitrator. We had a separate training for FINRA cases that was designed to address the securities laws that apply to those cases. Anything else? Any questions from the microphones? So I'll conclude. I think we're at time, right?
Yes, sir. So we have another question in the chat box, if you would like to take it.
Okay, somebody asked me about the Israel-Palestine. So let me explain that. It sounds more important than in description and in the bio than it really is. And so we weren't working on peace between competing nations, right? We were asked to come there, and I was on the exploratory committee. There were 3 of us that went to Israel to talk to members of the Arab Israeli community and the Jewish Israeli community who had fights usually about business matters, and they didn't trust each other. And so we were designing, and Nev Shalom Wahat al Salam is a village, actually a monk, Bruno Hassar, gave this village, gave some land adjoining a monastery to the people of Israel and said, make a village where Palestinians and Israelis, Muslims and Jews would live together, be educated together, and would govern themselves. And so it's a village of not thousands of people, smaller than that. They built a school there. They teach the kids in both languages. There's a school for peace there that does some of the cutting edge work on solving the other problems. But we were designing a system for disputes between those 2 communities to be resolved. They couldn't hire a Jewish mediator because they didn't trust the Jewish mediator. They couldn't hire an Arab mediator because the Jews didn't trust him. And so we designed co-mediation, where they would hire a mediator from each community and resolve that dispute. And that went on and developed to become a community mediation system where they were doing disputes between the communities at the community dispute level, where there might be neighbor disputes or other disputes beside trade disputes between merchants. And so that was in 2009. Actually, that was when I was there. Security was quite challenging. At the very beginning, the bombing had stopped, but there was still a couple bombs that were dropped. We were about halfway between Jerusalem and Tel Aviv, in this village where we stayed, but we did some of our research work in Jerusalem with some ADR professionals there, and in Tel Aviv as well. Very interesting work. The end result of our product was to design a training system for mediators there. But despite, every time somebody sees Arab Palestinian, they think you're resolving the political and the religious dispute between people, but we weren't there working on that. We were working in those communities, but not on that issue, on the smaller issues that face each of them on a day-to-day basis, not the national issues. What if a party wins the arbitration, but the arbitration ignores that? Good question. And so the one first point to make is, in arbitration, there's really rarely an appeal. The only basis in most countries for an appeal is that one of the arbitrators didn't disclose some relationship. I forgot to tell you that I'm Subodika's uncle, and she was the lawyer for one of the parties in the dispute, and I never told anybody that, and you found out, and I should not have taken the case. That there can be some appeal from, but usually there's no appeal. And so if we render an award and it's not paid, that award gets registered with the court and becomes a judgment. And then you go into court and say, here's the arbitrator's award, make it a court judgment, and then you can use the court process to get bank accounts, assets of whoever that party is in the jurisdiction that you're in. It's in my jurisdiction quite easy to do. It's one piece of paper, and they're never denied. It's just filed, it becomes a judgment, and then you can go to the bank with the judgment and say, give me this person's money. Okay. Anything next?
Thank you, sir.
Good. Thanks for the questions. Yeah.
Meeting and enlightening session, sir. I hope all the queries of the participant have been answered now. Now I request the chairperson of this technical session, Professor Shakil Samdani, sir, for his concluding remarks. Shakil, sir, kindly unmute yourself. Shakil Samdani, sir, kindly unmute yourself. Yes.
Am I audible?
Yes, sir, you are audible.
Okay, okay. Thank you very much, Mr. Tom Valenti, for your address. I am sure the participant must have benefited a lot from you. We are grateful for the time and effort you took to share your thoughts and experiences with us. Sir, I would like to add something. And I would like to start by saying a quote of Abraham Lincoln, in which he says, "Discourage litigation, persuade your neighbors to compromise whatever you can, point out to them how the normal winner is often a loser in fees, expenses, cost, and time. Injustice anywhere is the threat to justice everywhere," is rightly quoted. We know that for non-creamy layers or poor people, it is difficult to reach the court of law because of hefty legal charges and complicated procedure. In most of the cases, they prefer an easier way to settle their disputes. This function is well undertaken by ADR, as you have pointed out. This function is well undertaken by ADR. It also reduces the stress from the minds of the disputing parties as they have reached a conclusion. So ADR, or alternate dispute resolution, is a mechanism for solving the disputes outside the court of law. Now I will discuss the background of ADR in India, a historical background of ADR, sir. As far as the historical background of ADR in India is concerned, I would like to say that ADR has existed in ancient India, medieval India, and is still existing in modern India. In ancient India, disputes were peacefully decided by the intervention of kulas, or the family or clan assemblies, serenis, guilds of men following the same occupation, parishas, assemblies of learned men who knew law before the king came to adjudicate the dispute. Although the highest authority was the king, but the people respected the mandate of kulas, serenis, and parishas. When they were not satisfied in that situation, they went to the king. Otherwise, most of the cases were settled by these three categories of persons who were very near and dear to the litigants. Coming to the medieval India, the concept of settling the disputes outside existed. The English meaning of the word tahkim. Tahkim is an Arabic word. At that time in medieval India, there was Mughal rule and Persian and Arabic words were used. The English meaning of the word tahkim is arbitration, and the meaning of the word hakam is arbitrator. An arbitrator was required to possess the qualities essential for a qazi, I mean an official judge presiding over a court of law whose decision was binding on the parties, subject to legality and validity of the award. So the practice of ADR was very much prevalent in medieval India, especially under the Mughal rule. Now, sir, coming to the ladies and gentlemen, my dear participants, coming to the modern India, there are various methods of ADR. There are arbitration, mediation, conciliation, negotiation, and lok adalat. Mr. Tom Valenti, you have rightly discussed them in detail. You have discussed them in detail. I would like to discuss Lok Adalat, which is nowadays prevalent in India. And under the Legal Services Authorities Act 1987, the Lok Adalats have been established throughout the India. These the word Lok Adalat means people's court. This system is based on Gandhian principles and is one of the components of ADR system in India. Lok Adalat provides alternate dispute resolution or the device for expeditious and inexpensive justice. One more thing I would like to tell you, sir, the participants, that in India, since we have a secular democracy, in family matters, the parties, I mean the communities, they are free to decide their matters. So as far as the Muslims in India are concerned, they have used this opportunity, and for the last several decades, they have been using the platform of Darul Qaza. I mean the Shariat court in family matters, dower, divorce, marriage. They have been using it. In every city, you will find 1 or 2 qazis. And in most of the cases, the decision of the qazi, they are accepted by the parties because they have already given their consent that they will accept the verdict of the court, verdict of the Darul Qaza or the qazi. So this system is also very, very well going on in our country. In the last, sir, I would like to say one more thing, that in India, nowadays, due to COVID, this is the best option. You have already told you that for the last, I mean, throughout the world, if you go for the last 7 or 8 months, the entire world is facing COVID, this pandemic. And in the time of pandemic, it is very difficult to go to the court, opening of the court, going to the court. Lawyers, they are unable to attend the courts. So in this situation, it is perhaps the best solution to opt for ADR. This is the best solution. And you have already pointed out about this, because in the current pandemic, this current pandemic has led to a virtual shutdown of the court system. Civil trials have been suspended. Court appearances are not taking place. In essence, cases have been left in limbo, a virtual standstill. So in this situation, I think the ADR is the best solution. And going through your CV, I found one very important thing, that you have also tried to solve the dispute in Middle East. You have been the arbitrator, negotiator. So I would like to tell you one more thing, that under international law, Article 33 of the UN Charter says, Article 33 of the UN Charter says, 33 clause 1, that the parties to any dispute, the continuance of which is likely to endanger the maintenance of international peace and security, shall, first of all, seek a solution by negotiation, inquiry, mediation, conciliation, arbitration, judicial settlement, resort to regional agencies or arrangements or other peaceful means for their own choice. So not only in civil matters, but in international matters, Mr. Tom, this can be very useful. And I am very happy, I am very satisfied that you are trying your best to solve the problem between Arab and Israel. Moreover, sir, I would like to conclude my talk by saying that nowadays, due to the pandemic, the litigants, they are in trouble, because they are not in a position to solve their matters, especially where there are petty fines, where money is involved. At least in Lok Adalats, both the parties are there, and the mediators are also there. They tried their best in accident claims, for example, insurance claims, petty matters. They try their best, maintenance matters. They tried their best to solve the matter by persuading both the parties. So nowadays, for the last 7 or 8 months in our country, for the last 4 or 5 months, we are not in a position to go to the court. So perhaps this is the best option available nowadays. In the last, I would like to once again thank you, sir, for your enlightening speech. And I am also thankful to the college, I mean SS Khanna Girls Degree College, who has organized a very important international e-conference and invited us. I am really thankful to the chief patron, Professor R.R. Tiwari, the Vice Chancellor of Allahabad University, patron Dr. Asha Seth, and there are many persons associated with it. And especially I am thankful to the conference director, whom I am associated, who called me, Professor Lalima Singh. She is the principal of that college. I am very thankful to Professor Lalima Singh Saiba. She invited me, rather she forced me to preside this session. I am very thankful to her. I am also thankful to the coordinator, Dr. Archana Jyoti, Dr. Jyoti Bajaj, Bajal, Dr. Anuradha Singh, Dr. Preeti Singh, Dr. Sippi Singh, Dr. Nishi Seth, Dr. Arifa Begum, she's from Urdu department, and the organizing committee, Dr. Alok Malia, Dr. Shikha Agarwal, Dr. Richa Tandon, Dr. A. Rahman, Dr. Sumita Sahgal, Dr. Subedika Sharma, Dr. Surata Malvi, Dr. Ravi Kant Singh. These are the persons who are the backbone of this seminar. And I have left the names of Garima Gupta, Professor M.L. Nath, Professor Namita Singh, Professor Jagdamba Singh, Professor Ramen K. Singh, Professor R.K. Chaube, Professor Dhananjay Yadav, Professor Ashish Saxena, Professor Ajay Pratap, Professor A.K. Vajpayee, and Dr. Ravi Tandon. These are the persons who are behind this show, behind this 5 days international conference. It is very difficult to organize 5-day conference. I have been also doing for the last 2 months. I organized 5 conferences, 2 international conferences, and 6 conferences, and 4 national conferences. I know what type of labor, what type of pain is involved in it. I am thankful to you, sir, once again for your very enlightening lecture, for your question answer session, and I am once again thankful to the SS Khanna Girls Degree College, to the management, especially the chief patron, and the principal, Professor Lalima Singh, who is the director of the seminar. Thank you very much. Thank you, sir.
Thank you, sir, for your kind words. Now I request to Ms. Shubhodika Sharma to propose formal vote of thanks.
Thank you, ma'am.
Shubhodika, please.
With this, we have reached the end of our session. On behalf of the principal, Professor Lalima Singh, the organizing committee, and the entire SS Khanna family, I would like to express my gratitude to Professor Shakil Samdani for chairing the session. Sir, your presence has truly been very motivating for us. I would also like to thank our distinguished speaker, Mr. Tom Valenti, for being kind enough to accept our humble invitation. Sir, it was truly an informative and enriching session. I would also like to thank Mr. Gaurav Gulati, founder and managing director of Droid Pinel Group Prayagraj, for his constant support and assistance. Also, I would like to thank all the participants who have joined us. Before signing off, I would like to request our chairperson, our speaker, and all the participants to kindly mute themselves, switch on their cameras for a group photograph. Thank you all and have a good day.
Yes, I would like to say a few words, if I'm allowed.
Yes, sir. Yes, sir. Sure, sir.
Are you audible? Am I audible?
Yes, sir.
Yes, sir, you are audible, sir.
I want to say one thing about SS Khanna Degree College. I have been to your college for 2 times in Damodar Shri competition. And I want to tell the audience and the persons who are listening to me that this is perhaps one of the best colleges of UP. I have seen the discipline, the management, and the participation of students. I have been there twice as a judge in the Damodar Shri competition, and I found that the competition was held very fair. It was very fair, and everything is up to mark. On this occasion, I want to congratulate the college administration as well as the management committee for managing the college in such a beautiful way. I am very much impressed, and God willing, if I have some time in life, I got some money, then I will try to establish a girls college at Aligarh. I am really, I want to, मैं चाहता हूँ कि ये कॉलेज बहुत तरक्की करे. इस कॉलेज की इंतज़ामिया बहुत अच्छी है, यहाँ का डिसिप्लिन बहुत अच्छा है, यहाँ का जो मैनेजमेंट है वो बहुत अच्छा है, और जो जजेस साहबान जुड़े हुए हैं उनका तो जवाब नहीं. दे आर सो हम्बल कि मैं उनके बारे में कुछ नहीं कह सकता. ऐसी ह्यूमिलिटी, ऐसी हम्बलनेस मुझे कहीं नहीं मिलती. मैं SS खन्ना डिग्री कॉलेज को तहे दिल से सलाम करता हूँ, उनकी प्रिंसिपल साहिबा को सलाम करता हूँ, उनके मैनेजमेंट को सलाम करता हूँ, और आज उन्होंने मुझे यहाँ पर बुला के जो इज्जत बख्शी है, मैं उसके लिए तहे दिल से शुक्रगुजार हूँ. बस यही मुझे कहना था. बहुत बहुत शुक्रिया.
Thank you, sir.
Thank you, sir. Thank you, sir, for your kind words.
Thank you, madam. Welcome.
Now, sir,
Thank
we
you,
move
sir.
on to the next technical sessions, technical session 4, which is on social sustainability of in the 5-day international e-conference on sustainability challenges and transforming opportunities amidst COVID-19.
Okay, madam. Go on.
In this session, as chair of the session, we have amongst us Professor Ashish Saxena. I welcome you, sir. And as our invited speaker, we have Professor Unnat Pandit from JNU. A warm welcome to you, sir. Professor Ashish Saxena is currently the professor and head department of sociology at University of Allahabad. Earlier, he was an associate professor at University of Jammu and has significant publications on the issues related to social exclusion, social justice, subaltern communities, land reforms, democratic decentralization, identity politics, and globalization in India with specific reference to Uttar Pradesh, Jammu Kashmir state. A recipient of prestigious UGC DAAD Fellowship 2014 under the UGC DAAD Exchange of Scientists Program in Humanities, and a prestigious DP Mukherjee Memorial Award of 2017. He has also been a recipient of Sulabh Swachhata Samman 2016 and a gold medalist for academic contribution in the field of sociology of sanitation and action sociology by Sulabh International. He has also received the prestigious Devang Mehta National Education Award by Business School Affairs, Mumbai, on its 25th Silver Jubilee year in 2017. 17 on 14 September 2017. He has his publications are Shifting Manifestations, Scheduled Castes in Jammu and Kashmir, and 2019. And then he has handled many projects. He has been state coordinator cum consultant for the study of socio-economic assessment of Manrega on financial inclusion, convergence, and awardees district for selected districts in UP. He has held honorary positions and been members of many academic associations. He is a life member of the Indian Sociological Society, and he has been the youngest management committee member of the society till date. He is a life member of NWISA, and he is an external member team of the PRIAS, member of editorial advisory committee of e-journal of Sociological Bulletin, member of Pahadi Advisory Board, Government of J&K, and a member of Community Development Cell, University of Allahabad. He is also executive member to research committees, many research committees, to child and youth studies, and has acted as ICSSR observer for capacity building program for NGK VP Varanasi. Sir, I will accord you a warm welcome and as a chair of this session, and would now invite you to invite the speaker and do the honors now, sir. Ashish, sir.
Especially when we try to speak about the pandemic, many things comes in our mind, especially with regard to the sort of crisis of sustainability which we try to see. And under these situations, if we are trying to handle the academic deliberations, I think it's really wonderful. So I think without wasting much of my time, definitely it's a tight schedule. So virtually I would invite Professor Unnat Pandit sir. I think CV may be read by Dr. Jyoti in the later phase, or maybe subject to the permission of the chair. And also, I think it's really wonderful to have Professor Unnat Pandit sir, who is doing wonders in terms of speaking about Atmanirbhar Bharat. And I think it's basically the AIM which is really working for doing wonders in terms of indigenous innovations. And I am sure that the deliberation by Professor Unnat is going to enlighten us with many other issues which we are ignorant. So I am quickly handing over the mic to Professor Unnat sir for having his deliberation on this particular issue, and followed by my chairperson's comments. So over to Professor Unnat. I think I can see him smiling face, and I think he will be a wonderful speaker to give us the insights about the ups and also the positivity which we have to see with regard to the social context of sustainability. So over to Professor Unnat. I warmly welcome, sir. And I now ask you and request you on behalf of the college to give your deliberation on this particular issue. Professor Unnat, sir.
Thank you, Professor Saxena. Hope I am audible and volume is also reaching to all the participants.
Yeah, it's quite audible. Yeah.
Yes, sir.
This is just to confirm that I am audible and then there is no signaling or transmission related. So it's indeed really a privilege to be with you, and especially I would like to congratulate the organizer from the SS Khanna Girls Degree College, Prayagraj, for organizing such a wonderful and interactive session on sustainability challenges and transforming opportunity amidst COVID-19, and wherein on the theme of socio-economic sustainability. Today, I am going to share my views on Atmanirbhar Bharat, role of youth in societal transformation. So I will be in mostly in a dual language so that message is reaching to majority of the participant. सामाजिक परिवर्तन में युवाओं की भूमिका. This is something which is grossly touching to each section of the society. And youth are pioneering a pillar of this entire structure. So the three qualities which are there in the youth, which can really transform entire society. पवित्रता, धैर्य और दृढ़ता, तीनों सफलताओं के लिए आवश्यक है और इससे सबसे ऊपर है एक दूसरे के साथ मेल जोल, प्यार बढ़ाना. So this is something which is going to be the foundation of entire societal transformation. And I'm trying to correlate the things with the Atmanirbhar Bharat Sankalp, which has been given by our Honorable Prime Minister Shri Narendra Modi ji. And today, I don't know whether the participants have seen the press briefing by the Minister of HRD or not. Minister of HRD has unveiled the National Education Policy. And after more than 30 years, this policy has been redrafted. And in the present context and in the futuristic context, how this is going to impact the education sector. And we all are part of the entire educational system. So how the pedagogy is going to change, how this is going to have a shift. I would like to interlink this concept. So this slide I have prepared just before a few minutes, that how we can have a correlation of this with the Atmanirbhar Bharat concept, and how the societal transformation can be enabled by the youth. So if we want the societal transformation to happen, and we want to achieve the goals of Atmanirbhar Bharat, then it's a high time for all of us to really brainstorm about Atmanirbhar Shiksha and Atmanirbhar Yuva. If we want the transformation in real sense to happen, not just a societal, it's a holistic transformation. If we are seeing and we want all the societal challenges are to be addressed, then we would like to have a transformation in the education pedagogy. And that's what is the visionary National Education Policy has been released by the Union HRD Ministry. And this is something which is going to ensure that education is encouraged by the society, and societal needs are being fulfilled through the education. And of course, the value component and the inclusiveness component are also being ensured. Education should be stress-free and increase the knowledge and skill of the students and the youth. And definitely it should be encouraging or inculcating the patriotic nation building among the youth and the students. And if we want to achieve the Vishwa Gurutva, then the education system and the transformation which has been proposed will definitely ensure that the outcome of this entire initiative will put our nation into the world superpower. And that's the ultimate goal, which can be achieved through the deployment of this policy. Now, I am trying to again correlate the contextual aspect. Currently, in the previous speaker has also mentioned about the kind of challenges which are being faced. So we are passing through a change, transformation, and Earlier, in a normal days, we used to go into the mall and we used to have a chilling out with the family and with the people. Now, during all of a sudden, which we may not have even foreseen, that the society is going to have such a pandemic situation. And we may not have imagined that 70% of the world population is going to be at a risk of the COVID-19. And midst of the COVID-19, we have also got the message of us thinking, envisioning about the Atmanirbharta, self-reliance. And that's what we all have realized, that on the things where we are self-reliant, then we are much more sustainable and we have a defined growth parameters achieved as an outcome. Now, on an education field, the time is for recovering the education, which for almost more than 100 days, the entire system was just locked down. Nobody was going out. And now slowly and steadily, entire education system has transformed into the online mode. We may not have thought in December that we are going to have such a international high quality interactive sessions and enjoying this knowledge sharing in our laptops or on our mobile. And this is again, the COVID-19 era has given all of us this opportunity. It's a crisis and it's a change, but change brings the transformation also. And the education has this quality wherein we can achieve this. I would like to quote here one of the slok from Srimad Bhagavad Gita in the 3rd Adhyay, 20th Pad, 20th sloka is Karmanaivahi Sadsiddhi Masmitaha Janakadayaha Loka Sangraha Mevapi Sampashyan Kurtam Mahansi. That means karma, जो क्रिया है वो क्रिया ही रामबाण है, उत्कृष्टता एकमात्र उपाय है. यहाँ तक कि राजा जनक राजा was highly visionary and with full of administrative qualities which is inbuilt into him. Then even with this kind of a visible karma, that karma is required to achieve the excellence. माध्यम से ही लक्ष्य को हासिल किया. In that sense, समाज के कल्याण के लिए कर्म अनिवार्य है. तो युवाओं को भी कर्म के आधार पर भारत के निर्माण में, समाज व्यवस्था के निर्माण में अपनी भूमिका अदा करनी होगी. This is something which requires the societal transformation. Education is the foundation, where I would like to here present the Pancharatna. I have made these slides in Hindi and I am going to discuss in English so that the message is reaching to each and every participants into this group. The youth has a power. Youth has the energy, and how youth can really bring us the future citizen, which is thinking about the education, which is the fundamental of any human being and its growth and development. So education and this current new education policy, I am sure tomorrow and today after this session, you are going to see so many articles, so many write-ups, so many news, so much of into the social media about this. But entire thing is required to have a brainstorm. And maybe it's a high time for deciding something on this line to have a brainstorming among this and organizing national, international brainstorming session on national education policy as well. The policy again emphasizing on knowledge, utilization of a knowledge for the societal benefit. And yes, the futuristic technology, knowledge, research, and the innovation are going to give us the superpower or position to place Bharat among the superpower. And that is something which is useful. Intellectual development is again an important aspect. युवाओं को समसामयिक विषयों पर बौद्धिक संवाद एवं चर्चा करना. Currently, the topics which are there for the discussion, brainstorming, I think it's a high time for encouraging the students and youth towards getting engaged into such a development. And definitely the Anushasana and the discipline is going to build their entire character, their aspect towards the society, and definitely it is going to show them the path of progress and the success. And that's what is going to ensure that they are achieving the goals of Atmanirbhar Bharat. And if at all we want that India should lead the world, and technology and innovation is going to help in building the self-reliant Bharat. This is what the official graphics of the PIB, which I have utilized in my presentation. Atmanirbhar Bharat, the road ahead, is describing the 5 pillars. And these 5 pillars can be achieved if we have all the information, data, knowledge, and the wisdom in place. 135 crore of a population is generating data at each second. And this data is required to be organized in the form of a meaningful information and contextual knowledge, which will open up the wisdom and applied approach towards building the societal useful knowledge. And that's what is going to build the entire nation. And across entire value chain in the 5 pillars, which has been described in this concept called Atmanirbhar Bharat, the holistic development can be achieved if we have a technology and innovation in sync with the societal relevance, cost effectiveness, citizen-centric, and relevant to the need. And we have major goals which is required to be achieved, which is a sustainable development goal. And if we want the societal transformation to happen, we have to encourage the youth towards exploring the new technological advancement in this sustainable development goal. India is negatively listed in among the majority of this, around 8 to 10 SDGs, wherein we are negatively among the top 5 or 10 countries. So it's a high time for exploring the solutions and offering something which is useful. And if we want to build the knowledge economy for the self-reliance and societal need, the youth can utilize their inner talent and knowledge and effectively manage both of them for the benefit of people, for the benefit of new processes, novel technologies, and advancement. Such practice, improvement, insightful thinking, and the information will lead us towards building the knowledge economy. And while we are envisaging, the role of youth is important for the societal transformation. It's a time for empowering the community to ideate and bring the change. We all must know what we have and how we can utilize this knowledge towards betterment of the next generation. and rejuvenate the inherent culture of society towards adopting the new innovative mindset. Majority of the startups and entrepreneurs are failing. These are the few rationals why they are failing. But in general, these are the fear in the youth also. If we address this fear, then it's a time to overcome with this, and we can build the new inspiring youth with the skill of tomorrow's education and the innovation need. And education and innovation can really play a pivotal role in the societal transformation and ultimately reaching to the goals of Atmanirbhar Bharat. Youth has to be an omniscience approach. Omniscience approach means grab the knowledge wherever and anywhere which is available with you. Learn to utilize the knowledge and apply into the developmental leap. Build the entrepreneurial mindset in the youth so that they are right from the beginning. They are thinking about the job creation. And there might be 10 to 20% students who can create the job, but they are going to cater the need of entire youth who are going to create the foundation for the Atmanirbharta. Tell them to really have a vision of managing the bottom lines. Drive through data and the facts rather than driving through the hypothesis. This is something which, if such qualities are getting nurtured into the youth and the students, then we can easily see the societal transformation, which is the important pillar of Atmanirbhar Bharat. What innovation can do for future, that's again important, because most of the time when we are thinking about the societal transformation, we are eliminating how these societal challenges can be addressed. Technology and innovation has the power to address this challenge, and that can be achieved by reaching out to the new partners, exploring the external innovation and power of doing it yourself. It's a high time for all of us to nurture the youth towards the societal transformation and utilize their strength and power, and which is going to be the game changer strategy for Atmanirbhar Bharat. Innovations are important enabler, and for the emergence of powerful regime, Bharat need to encourage the young and dynamic aspiring innovators, entrepreneurs. And that's what is being done by Government of India through various initiatives. Startup India, Stand Up India, and empowering the youth to skill India. These are the inherent skill building, inherent capacity building initiative. Bharat is a young country, but Bharat really need to be much more progressive. The baby step which we are taking towards the self-reliant Bharatiya culture is not going to be sufficient enough. It's a time for a pole vault jump, which we have to take up and build the Atmanirbharta, creating the center of gravity to be in the global power center. And that's what is visible even during the COVID-19 time and pre-COVID-19 era as well. The international position of Bharat has gained, and we are already establishing ourselves into the power center. And the presence of Bharat has been noticed by all the important economy. And that's what it can be realized if we use the hotbed sectors offered to the startups, innovation, and the entrepreneurship sector. Time for building the new approaches into the progress and growth of youth. Time to strengthen the innovation ecosystem in Bharat and ensure that all the transformatory solutions are reaching to each and every mile of the country and the globe. And in order to do it so, it's a high time that students and the youth are enabling the startup ecosystem, and education which we are imparting to the youth should be transforming the global trend and the trade. This is something which is important, where in highly personalized experience we have to give to the technology users. It's a high time for scaling up the unimaginable way through creativity and innovation. Nation curriculum building, which has been the major recommendation of the National Education Policy. An entire education curriculum is going to be redesigned as per the societal need, and which is also, I have covered it here. It's a time for being a part of a solution, or unless we can also go into the survival mode. These are the hotbed sector which are going to be useful for the youth and the students for the societal transformation. On education sector, naturally we all are associated with the education system. So we all are encouraging the online connectivity and imparting or sharing the knowledge among the students and youth. But on various aspects of the online education, the startups and technology developers have already offered their solution. And this is the power of offering the new and futuristic platforms in imparting the education and setting the new pedagogy for the education. I have also specifically highlighted 2 startups here, which are the Indian startup, among the many other which is mentioned in this particular slide. EMLI, which is just near to the JNU logo. This is something, the startup whom I have met during my interaction at Atal Innovation Mission Niti Aayog. They have developed something which is a cutting-edge holistic solution, which takes care of the admission to the placement. So student taking admission even at a KG nursery level, do the PhD, postdoctoral, and the placement aspect. So entire value chain of any youth coming out of the education system can be managed through this. Purchase to write-off. Any academic institution, this is important component. And recruitment to the retirement. So this is something, the system platform which is offering the holistic solution. Anything and everything which is a part of the education system can be managed through this. And really, I'm proud to mentor them. When they met me, they are currently offering this solution to one of the French West African country. And entire university administration, school administration, child's holistic progress, growth monitoring is being done on a single platform. Entire country's education system is being managed by this platform. It's a proud that this is the Indian startup, and this is the power of Indian offering. If now honestly answer yourself, I don't need any answer. How many of us are putting the trust on the youth and the startups who are developing such a useful solutions? How many such startups have approached us, or if we not, then how many startups we have approached them? OneForm.in is one of such startup also. India's portal for online admission. So students and the parents don't need to go each and every college. One single application form, and the platform will do entire admission process. multiple colleges, multiple university, multiple curriculum can really manage through this portal. And in new education policy also, there are various recommendation and cross functional approaches are encouraged. That means a student who is studying in physics, chemistry, or biology and has an interest with the music, so he can also opt the music as one of the subject in the formal education. This is something which is a dynamic, and such dynamic admission process is also required to be managed. And this can be managed by through such startup. 3,500 institutions got affiliated with this particular platform, and time is changing. Education is the most powerful weapon which we can use to change the world. And if we want to build the self-reliant Bharatiya education and innovation ecosystem, the youth and the students has the power to transform this. We all are bound with the various kind of responsibilities. But the quote again, I would like to mention it into the Hindi. जिस दिन हमारे सिग्नेचर ऑटोग्राफ में बदल जाएंगे, उस दिन मान लीजिए कि हम कामयाब हो गए. Students and youth should think about the success in all walks of the entire value chain and the growth plant. सर्वोत्तम व्यक्ति वो नहीं है जिन्होंने अवसरों का इंतजार किया, बल्कि वे हैं जिन्होंने अवसरों को अपनाया, जीता और सफल बने. And this is something which is the remarkable revolutionary thought by our honorable former president, late Shri Dr. APJ Abdul Kalam. And that's what is the foundation. If we want a societal transformation to happen, youth has to play a catalytic role and in achieve the Atmanirbhar Bharat. We all are bound with the various kinds of struggling, or we are found with the chaotic situation and yes, confused during the lockdown phase. The only way to predict the future is to create it. And this gives us the opportunity to catalyze the youth towards the societal transformation and ensure that we are achieving the all goals of nurturing them and promoting them towards the societal transformation. Thank you very much.
Thank you, sir, for your illustrative talk and making us aware of the initiatives that Indian youth are doing from here at various corners of the country. Now I would request Ms. Subodhika to proceed with the question and answer session. Subodhika?
Yes, thank you, ma'am. We have a participant, Mr. Gaurav Gulati, who would like to ask a question.
Good evening to Ashish sir and Unnat sir. I have a question for Unnat sir. Sir, what are your opinions on the opinions that the best way that the youth of the country can contribute in nation building?
See, youth has to think about.
So you are not audible.
Hello, am I audible? It is showing me the green sign in the microphone.
Yes, sir. Now you are audible.
I am audible.
You are audible, sir. You are audible.
Yeah. So, if youth wants to be part of the nation growing and nation building activity, it's a high time for youth to utilize their knowledge. For so many years, they have gained the knowledge and they have gained the education, but they have hardly learned how this knowledge can be utilized for the society, how they can utilize their knowledge towards addressing the societal challenges. And this is something which, if they are doing, then this can be the revolutionary contribution for the students and youth towards the nation building.
So we have another question in the chat box. It says, we are always talking about sustainability, but we never reach there. What are the problems we have faced to reach the goals?
See, in my opinion, again, this is my view, sustainability is the continuous process. Of course, as soon as we are achieving the sustainability at a X level, but under that circumstances and that condition, sustainability bar will go little upside. So we have to make another effort towards achieving this. And at each level into the society and across the various sections of the society, the sustainability parameters are different. And while we are thinking of achieving for one section of the society the sustainability, the bar will go up by we achieve this. And then we have to make an extra effort to achieve the new level of this. When we achieve the new level, the bar will again go upside. And that's the level of growth and progress for the society and growth and progress of the country, which is inherent part of the sustainability all across the individual and the country as well.
So Bodhika, I would like to pose a question to sir. Rather, it is a request, not a question. Sir, would you throw some light on an establishment of an incubation center in educational institutions such as ours, All Women's College? So I would request you to throw some light on it, sir.
The incubation is again an important aspect. How we can nurture the students and youth towards innovating, and once they have whatsoever they have innovated, how we can ensure that their idea, their concepts are reaching to the society. And that's what can be achieved by if we handhold them, if we support them, and if we mentor them. And that's again, mentoring is a national mentoring program is also announced into the new education policy. And new education policy is also encouraging the students and youth towards adopting that technology and utilizing their inherent creative and innovative skills towards addressing the societal challenges. And this can be offered if we create the specific infrastructure, specific facility for them. And this facility is called the incubation center. And very much there is a all woman and incubation center is already there in Rajasthan, Banasthali University. And I'm sure you should have the collaboration or MOU signed with this incubation center and get your iCell, eCell incubation center established with their mentorship, so that your sustainability, your incubation centers access and operations are seamless, and learning from the experienced person is important. So I would recommend that you should be exploring the MOUs with this Banasthali and also All Women Incubation Center in Hyderabad. And they can really guide, mentor you towards establishing the incubation center in your college.
Thank you, sir, for your insight into this issue. And Mr. Bodhika, you can pose questions if we have any in the chat box.
Yes, ma'am. There was a question in the YouTube chat box. I'd like to ask that. It says, how can we bring the kids' positive attitude towards technology?
See, kids are much more smarter and adaptability is much higher. So technology adoption, which is there in kids, we should be thrilled and we should be satisfied with the level. Now the students and the kids in the class 2 and 3, they are operating the Apple phone, they are opening, they have their own choice and selection, what to view, what not to view. And for those who have small kids nearby to their houses, they or in their house, they can see and they can check with this behavioral change. And this is the spirit of Bharat, wherein the technology adoption is all-time high and it is growing. And that's what is going to help all of us in encouraging their adaptation with respect to the new and novel approaches, including the online education. And this is all going to increase, and it has been witnessed. The way teachers are teaching in online mode, which are pretty mature teacher, and they know how to, they have seen entire telecom revolution, right from booking the trunk call from home to the STD, ISD connectivity in mobile, and also the Facebook and WhatsApp chatting and all. But still they are facing to get adaptation about the online education, connecting through Zoom, teaching the mathematical equations, utilizing the online platform. Without student, it's difficult for teachers to teach. That's the level, but students have adopted. They are quite comfortable learning, sitting in home, and they have quite adopted it well. So this is going to increase, and such hybrid education system is going to build the new normal in entire education academic pedagogy.
So there's one more question from Mr. Yogesh Mishra. Till when can India achieve the Atmanirbhar status?
See, Atmanirbhar Bharat is not a new concept. This is the concept which has been defined in the present context. And it's a continuous moment. It's a goal, like similarly to the sustainability. But yes, holistically, all inclusive, Atmanirbharta is the ultimate goal which we should be achieving. Defining into the particular time frame, 135 crore population will ensure that they will be working towards a single goal and achieving this goal. It's next to impossible, the time, harmony, and energy which they are going to put it. Now, in scientific term, when all the three major parameters are not in harmony, how can we define the time frame? Maybe a physics professor can explain this phenomenon, but it is difficult to define the time frame. Collectively, all 135 crore population of Bharat have to work in harmony, in sync with the thought process. Then I think we would be in position to achieve the Atmanirbharta. But it is not an impossible task. The classic the classic example is the Janata curfew, a call which has been given by our Honorable PM. No matter whether people are accepting or they are not accepting this thought line of thought, but for the betterment of each citizen, for the betterment of India, Indian communities' health, everybody were there at their home. And they have obeyed the Swayambhu curfew, Janata curfew. That's the spirit, and gradually also the lockdown situation. we can't imagine that for more than 100 days we were into the home. Just think when you were into this situation in past. We don't have that history, but still we have adopted this. We have found the new ways to get connected with each other. We have found new ways to share the knowledge. And that's the spirit of our Bharat. And this spirit will remain as always.
Subodhika, are there any other questions, or should we go towards the
we can move
concluding remarks?
Yes,
Yeah, I have also another commitment. So
Yes, sir. Yes, sir. Yes, sir. So before we move on to the concluding remarks, I highly appreciate the presence of Dr. Ravi Tandon with us all through the session. Good evening, Ravi, sir. Now we move towards the conclusion of this session, and I would request the chair of the session, Professor Ashish Saxena, sir, to kindly give the concluding remarks.
Thank you, Dr. Jyoti. I think it's really wonderful listening to Professor Unnat. I think the name itself has a meaning, and which of course is reflected through how we can speak about the visionary speaking on this very sensitive issue. So Unnat definitely trying to see in terms of transformation. And I think the dialogue which has been raised, I think they are quite enriching, especially when we try to see it in terms of either the question of pavitrita, dhairya, and we try to speak about dृढ़ता की बात हम लोग करते हैं. और इसके साथ साथ जो एक इम्पोर्टेंट चीज है वो ये है कि हम लोग अपने आप के लिए ना करके बल्कि हम लोग एक विश्व स्तर पर जो ग्रोथ की बात है वो लगता है हमें करने की जरूरत है, और कहीं ना कहीं नीड ऑफ द आवर वही है उस तरह से. इसके साथ साथ जो एक अनुशासन की बात हम लोग करते हैं, आई थिंक बहुत सिग्निफिकेंट पॉइंट्स हैं जिनको सर ने रेज किया. बट बियॉन्ड दैट ऑफ कोर्स आई थिंक कुछ क्वेश्चंस का जो एक फॉर्मेट आया था, समवेयर वी हैव टू सी दैट Atmanirbhar may be seen as a sort of an ideal type, which of course may be having certain parameters, and definitely they can be contextual for a specific period of time, and it has to change. I think we have to believe on the dynamism. If it is not there, so then there is no need for the youth. So we have to think in that context that how through time we have to have certain amount of changeology with regard to the transformation. And I think one wonder which has been done through this pandemic is that putting the youth at the ICT, and somewhere I will just try to put it as e-youth. So I think that is the broader terminology which is trying to unite us all together. But beyond that, we have to see it in terms of the equity, or we have to see it in terms of capability building, which of course is the need of the hour. Either it's in the term of entrepreneurship, or it is to be seen in terms of the new Atmanirbhar Bharat. So I think these are the things which we have to take care for this particular moment. But beyond that, I think as has been rightly pointed out by Professor Unnat, that we have to have certain amount of convergence in terms of education. Like we try to speak about the single application form, which can have the common, what you can say, unification across the universities or the centers. I think that, of course, is the requirement. And we have to see that youth has to be seen not only in terms of a homogeneous category, rather we have to see the potentiality which has to be enhanced. And here I would like to just try to put that we have to see certain reversal which has to take place, because this is the time when we have to start from the zero, and then we can have the new build up. So I think rural definitely has a good potential in terms of contributing towards the nation. And I think that is going to be more effective, that we have to have an eye on how the youth of rural can bring about a change in the nation, in the urban. I think that, of course, is the requirement. And somewhere we have to stop this brain drain, which of course was happening. And I think pandemic definitely has stopped that by force. And now I think we have our potential very much in-house, and definitely challenges which are coming from China, how we try to overthrow them, and then try to have, under these critical situations, the advent of the Atmanirbhar. So I think the crisis has made us more stronger, more visionary, and in that way, I think many challenges are there which we can face. So we are the believer of the population definitely is huge, but I think we can have the population with potential which can do wonders. And I think many things are to be shared, but nicely has been drafted and documented by Professor Unnat. So I'm really thankful for a wonderful deliberations that we had, and also to the distinguished audience who put such a vibrant questions. And beyond that, of course, I think I would like to thank the college who always believe on doing something great in terms of having certain academic collaboration. And I'm hopeful that under the leadership of Professor Lalima, new things will be coming up. So I am thankful to all the distinguished co-hosts and also the delegates who are part and parcel of this. So with these kind words, I will be happy to say my final vote of thanks on my behalf as the chairperson, and over to Dr. Jyoti for further comments.
Thank you, sir, for your motivating words. Now I would request Dr. Subodhika to kindly propose the vote of thanks.
Thank you, ma'am.
And just after the a little interruption, I would say, I would request all, just after the vote of thanks, we'll mute ourselves and switch on our videos for a group photograph. Subodhika, now you can propose the vote of thanks.
I would like to record my gratitude on behalf of the principal, Professor Lalima Singh, the organizing committee, and the entire SS Khanna family. I would like to express my gratitude to Professor Ashish Saxena for chairing the session. Sir, your presence has been truly motivating for us. I would like to thank our distinguished speaker, Professor Unnat Pandit, for accepting our invitation. Sir, it was truly an informative and enriching session, and I hope and I'm sure that it has added to the knowledge of the participants. I would also like to thank all our participants who have joined us. And before signing off, I would like to request you all to mute yourselves and switch on your cameras for a group photograph. Thank you all and have a good day.
And with this, we come to the end of the technical sessions for the 4th day of this e-conference. And tomorrow, the last day, we'll be meeting. The first technical session will begin at 10 a.m. It is on the sub-theme of legal sustainability. So till then, goodbye to all of you and hope to meet you again tomorrow morning.
Goodbye, everyone. I'm ending the meeting.